round of applause for organizers. So it is my pleasure to introduce our first speaker, who happens to be my postdoc. I told him he could not introduce himself. Yan, yeah, he's a postdoc in my lab. That's who he is. And he did his PhD at Stanford with Dominique Bergman. And he's going to be talking today about decoding the cellular and genetic basis of nectary development and evolution in Mimulus. So thank you.
Thank you, Elena. Yeah, all right. Everybody can hear me okay? All right, thank you very much. I have to confess I cheated, so I'll put myself first so I can, enjoy the rest of the talk later. So bear with me. So you should be short. So I might cut it short because of the timing today. All right, so I stole this slide from my friend Minya, and I just show you the amazing flowers. And everybody loves flowers. But for me, the structure that I'm very interested in are not those showy petals or the sepals, but the structure that actually feed the pollinators. That's the nectaries that Clay already introduced you yesterday. So the nectaries are very important in our daily life because it gives rise to honeys. And then during the last 100 years of horticultural breedings, we have breeded like horticultural plants and crops to make more nectar. It's also a very interesting system in terms of evolutionarily because this is a phylogeny I'm showing you here in part of the core eudicus, and you can see that each column is a different floral organ that nectar can be associated with. And then during this evolutionary time, we can see this very mobile for the nectar structure, and then it has independently evolved almost more than 100 times. And it can dynamically gain and loss very quickly in the specific lineages, and suggesting that this structure can be easily achieved through genetic modification, in gaining and loss. At the same time, it's also very diverse in terms of the way you can transport the sugar out of the cell. So you have different ways to secrete the nectar, and using different layers of nectary cells to secrete those nectars. But for me, as a developmental biologist, I'm really interested in the developmental question behind nectary development. So no matter what kind of nectary plant develop, it always goes through 3 phases of developmental stages. From phase specification, that a small group of the cells will gain the identity and become nectary, to elaboration that they recruit specialized cell types like stomata and things that we'll talk about a little later today, and as well as they have very coordinated activities at the final stage of nectar secretion, as Clay has alluded to yesterday. It's a very short developmental window when the flower opens that they are allowed to, they will release the nectar. And I mentioned before that you can have nectar associated with any of the floral organ. And most of our research has gained from work from Clay's lab and many other labs that's from model plant Arabidopsis, right? So Clay already introduced to you yesterday that the nectar of Arabidopsis develop at the base of the stamen and between stamens. And then those tiny little structures will give rise to the nectary cells and secrete nectar. And then developmental research has generated quite a lot of data, but one of the most important one has been found is this Krebs claw transcription factor, the YAB family transcription factor that originally involving a daxial or baxial polarity of the carpel. And then when we mutated this, not me, but yeah, when the Krebs claw mutant has been generated, they completely lost the nectar. And on those small, tiny nectaries, we can always see stomata, and that's one of the hallmark of Arabidopsis type of nectar. And based on the work of Clay's lab, and we have now known that there's basically a metabolic pathways have been shown to be associated with this type of nectar, involve the synthesis of sucrose, transporting of sucrose, and hydrolysis of the sucrose to make nectar to come out of the tissue. But also, as I mentioned to you earlier, that nectar is has evolved more than 100 times during angiosperm evolution, and this only represents a small lineage. Arabidopsis only represents a small lineage of the angiosperms. At the same time, Clay also showed you yesterday that it's a very small flower, and then nectar is even smaller, sometimes less than 100 cells. I think Luke is going to tell you some exciting story today about how we can use single cell to allude to this. But if we want to use a biochemistry assay to really get to the function of some of those proteins, this might not be a great system to understand nectar development. At the same time, Arabidopsis is also largely reported to be self-pollinated, so the nectary is not really necessary for its reproduction. So to really understand nectary function in the ecological context, we also needed more models. Because of that, during my postdoc, I developed 2 more nectary development models. One is Mimulus, one is Aquilegia. They represent a different lineage of the angiosperm evolution, and also they use very different nectar secretion method to allow the nectar to come out. And then I was very interested in 3 major questions, both all related to the development of the nectary. And then today I'm going to talk about something I got really excited last year that we started a year ago, and then specifically using Mimulus to ask how is the nectary structure, the fate specifying, and also how does the structure change over time in terms of the location, and to facilitate different pollinator interactions. So Mimulus has been already set up a model to understand floral evolution and pollinator interactions, based on John Willis and then all the people from his lab. And then it's very attractive to me because similar to Arabidopsis, Mimulus is very easily transformable. So some of the species you can use floral dipping to make stable transgenic lines. At the same time, they all make pretty large nectaries, and I can associate it with multiple different type of pollinators. So when I first started, I was very curious where is the nectary and what type of nectary that Mimulus develop. And then we first look at SEM, and we've shown that the nectary is actually formed at the base of the carpel. But interestingly, it's only on one side of the carpel. And on this carpel, if we look at the epidermis, we can see a lot of stomata forming on this epidermis, suggesting they might use a similar nectary secretion method as Arabidopsis. And to validate this, we have looked at the transcriptome, and then as shown here, it's the 3 major sugar secretion pathways all being elevated in nectary in the late development stage, suggesting it's really using the similar type of secretion method as Arabidopsis as well. So this is very exciting to me because this also represents one of the most common nectary type in angiosperms. And I was very excited about this, and I decided to look further in terms of molecular mechanism that controlling the nectary differentiation. And then we first turned our eye to Crab's claw and using in situ hybridization. So now we're looking at the side of the carpel and doing longitudinal section. We can see at different development stages, we can see faintly Crab's claw expression on the epidermis on both sides at the very early stages, but with the developmental stage going on, when flowers start to mature, the expression of Crab's claw is sculpted to be only the future part of the nectary. And I was also very excited to see this expression pattern because the location of the Crab's claw expression and then the nectary development is actually the intersection of multiple different polarity axes of the organ. That's most developmental biologists are very interested in. So what I mean by polarity axis, so any lateral organ of plants would intrinsically have 2 axes of polarity. So we have the distal to proximal axis, basically. The distal side represents the growing tip of the organ. And then based on the orientation related to the apical meristem, we also have a adaxial and a abaxial surface. This can be mostly viewed as a leaf because when the leaf first emerges, it's close to hugging to the meristem. And once it opens up, the top of the leaf we call adaxial surface. And then the bottom of the side is abaxial. And then Mimulus flower is actually interesting because beside these 2 major axes, you also have a zygomorphic axis because the flower is zygomorphic. It's not radiosymmetric anymore, so you have the dorsal and the ventral side. So if we take a Mimulus flower and then turn to the side and only look at the carpel, we can see that the side close to the inflorescence is the dorsal side. The side further away where the nectary develop, it will be the ventral side. And then we have the distal and proximal axis here. And it's generally accepted that the outside layer of the carpel is a abaxial surface, and the inside is a adaxial surface. And if we put this together with our in situ, we can see that nectary exactly form at the proximal, ventral, and abaxial, the intersection point. So for me, this represents potentially these 3 polarity axes generate a 3-dimensional grid, and it can really help in pinpoint where the nectary is going to form. And we want to see whether this is true molecularly, but at the same time, it's also very important to notice that this could potentially also play a very important role in guiding the pollinator for specifically where to get the nectar. So here I'm showing the hummingbird pollinated species. Here you can really see that it does have a dorsal-ventral axis besides the carpal and also in terms of all the other structures orientation. And having the nectar only on the one side can directly guide them to know where to get the nectar and then allow the head of the hummingbird to touch the pollen and transfer the pollen to the next flower's stigma. Right? So this is very great, but like how exactly is it controlled in terms of this polarity axis? And then team up with Minya, who is the previous graduate in our lab, and then now she's opening her lab in UIUC, we decided to look at one of the polarity axis, the zygomorphic axis. And we can see that the cross-angiosperm, we know that cycloidea transcription factor has been repeatedly recruited to control the zygomorphic feature of the flower. And in Mimulus, it's also the case. If we overexpress cycloidea, everything is converted to the ventral side. So I'm very excited to using this to see whether that affects the nectar development in this case. But before that, we first look at the expression pattern of both Krebs claw and cycloidea. We see that if we take a cross-section of carpal, now we see that nectar forming at the bottom. Krebs claw is nicely expressed mostly on the ventral side, where it's cycloidea using the ruby reporter, the pink reporter. We can see that's mostly on the dorsal side. They kind of mutually exclusive based on the expression pattern, and I want to see whether they genetically interact. So if we overexpress cycloidea using this line already exists, and using DR5 reporter as a reporter for nectar fate, and we can see that in Y-type cases we have a nectar nicely formed, but when we overexpress cycloidea, there's no more oxygen maxima anymore, and then there's also no nectar forming anymore, suggesting cycloidea could act upstream of Krebs claw directly or indirectly to inhibit its function. And we move this further, that we actually knock it down cycloidea using RNI line, and we see something weird happening. That instead of having a ring of nectar, it's actually now we have two little bump potentially become nectar. At the same time, they have some fate problem. That normally we don't form those trichomes on the nectar cells, but now both sides would have trichome forming, suggesting cycloidea potentially not only regulating where the Krebs claw is expression, but also could play some role in the fate determinants of nectar. But at the same time, what this really intrigued me that it didn't form a ring like a lot of the species that we see. I'm very curious how is that controlled in the future. But one thing I also want to mention to you today is that the reason we're interested in this zygomorphic axis is also because the natural variation of Mimulus of nectar size lie on this axis. Here I'm showing you the small clade of Mimulus we work with. So they can interact with different pollinators, and they irradiated about 3 million years ago. If we take a cross-section of two really closely related species, Mimulus parisia, which is a sow-pollinated species, and then the Cornelius, the hummingbird pollinated species I showed you before. And we can see that they have very distinct nectar size. In the parisia carpal, the nectar is very small. It's mostly concentrated on the ventral side. But in the Cornelius species, the nectar largely expanded into the dorsal side. And then this nectar resize has been proposed to be associated with how much nectar it actually can generate as well. And to understand what is controlling this variation into the species, we decided to take a QTL mapping approach. With a very talented undergrad, Ray Sakamoto, we used the F2 population already generated by Yao Wu, and then we look at the variation in terms of nectar size in this population. We can see that if we take cross-section as we shown here in F2s, that we do see the nectar size variation in the F2s as shown in the circle region. And we can also generate a quantification in terms of the nectar size based on the proportion of the epidermis that's actually occupied by the nectar in terms of the whole, in the ratio to the whole carpal parenchyma. We can see that Mimulus cornelius have a much larger number, close to 1, and the parisia is close to 0, and the F2 is nicely lying between. And we look at the preliminary about 93 samples, different F2s, And to my surprise, we actually get a very almost pretty normal distribution in terms of the nectar size, suggesting there are multiple, at least two or three genetic locus actually controlling this factor. In Norwegian soy also just only one. And we decided to really map this trait. And when we got the result back 1 month ago, I was so impatient. Instead of doing the mapping, I decided to take some candidate-looked approach. We basically bin the F2 based on different, which locus you inherit for Krebs-Scott locus, whether they got a homozygous Parisi locus, or they got heterozygous or homozygous cardinalis locus. We can see that based on this binning, we already see a nice differences in terms of Krebs-Scott locus, suggesting that the Krebs-Scott locus is likely one of the influential factor of nectar size. But when we look at cycloidea, we didn't see anything really surprising. So indicating Krebs-Scott might be one of the controlling factors. And together with Ben Stone, who just started his lab at Mississippi State, we really mapped the trait. This is our preliminary mapping. You can see still some problems. We try to resolve it. But I was very excited because it shows one of the peak is actually underlying Krebs-Scott, and then there is a novel one on chromosome 4 that we're still trying to figure out what that is. And then just to quickly summarize here, so hopefully I've shown you that Mimulus is a great model to understand nectar development. It can give us a lot of cells to understand the biochemical property of this, and also the polarity controlling of the nectar expression is very exciting in terms of understanding developmental mechanism underlying nectar development and things. And in the future, I would like to continue this work and expand our work in nectaries. And then before that, I would like to thank everybody who has helped me in this project. Particularly Minya with a collaboration with our cycloidea work, and Ray, the talented undergrad, did most of the section of the F2 work. And also my great collaborator I generated in the past 10 years. And also I want to mention that in July, in July, I will be moving to UC Berkeley as an assistant professor, and I will be hiring across different levels. If you know anybody interested in interested in nectar development evolution, please tell them I exist. I really appreciate to have more evolution people in my lab, because I feel like I'm still learning on that front. And with that, I'm happy to take any questions. Thank you.
Okay, I think we have time for one question. If anyone wants to ask a question. Any questions? All right. I'm going to let Luke ask a question, because you have to get up anyway.
Trichomes on the nectary, the RNA line, is that a homeotic transformation? Is there similar looking trichomes on other organs?
Oh yeah, so Mimulus is annoyingly covered with trichomes. So yeah, it's the same type of trichomes, and but usually not forming in carpel. I think there were some potentially some regulation in terms of the receptacle and where the nectar has been miswired. That's why we're seeing them showing up on the nectar.
All right. Thank you very much, Ian. Okay.
I got it.
Oh, you got it? All right. He's got it. Okay. So, Philippe. Our next speaker is Philippe Amorin, who is joining us from Sao Paulo University, State University. And he's going to be telling us about the sweet link, ecology and natural history of nectar in plant-animal interactions. He's got some very cool videos.
Great. Good morning, everybody. And thank you very much. Before starting, I must admit, with my poor English, I cannot truly express how happy and honored that I am to be here. So all I can say is that I think actually that even Portuguese, I wouldn't be able to do this. So even Portuguese. So all I can say is thank you very much for this incredible opportunity. And thank the organizing committee of this meeting. Thank everyone who's attended this today. Thank you so much for having me. So today, I'm going to present you a few studies that we have been conducting in my laboratory at São Paulo State University in Brazil. Our campus is located at the countryside of the São Paulo State in a small city called Botucatu. Botucatu, in the local language, Tupi-Guarani, mean good airs. So it's a very nice place that we've been living for the last 13 years. So, to start, nectar has played a central role in some of the most influential books from Charles Darwin. So Darwin has cited nectar many times in some of these books. As you can see here, in chapter 4 of The Origin of Species, titled Natural Selection, in order to make clear how Darwin believed that natural selection acts, he begged permission to give two imaginary illustrations. And he started giving an example about a wolf and its prey. However, the next example, Darwin talked about nectar. Certain plants secrete a sweet juice, a little sweet juice or nectar which is secreted by plants, and insects come to the flowers to get this nectar, to feed on this nectar, and then they get dust with pollen. And Darwin continued by saying that those individual flowers that have the largest nectaries producing the most nectar also attract most insects and have advantages by gaining more outcrossings. However, the most remarkable example of nectar and nectary involving the evolutionary theory developed by Charles Darwin was published in 1862 in the book on the various contrivances by which British and foreign orchids are fertilized by insects. Darwin was writing this book when he received a box containing the flowers of Angrecum sesquipedale, an orchid species endemic to Madagascar. And when Darwin received this flower, he, in a letter to Joseph Hooker, famously wondered, good heavens, what insect can suck it? Then Darwin asked what may be the use of such a disproportional length of a nectar spore. Then Darwin thought that the fertilization of this orchid would depend on the length of this nectary, this floral spore, and also would depend on the nectar contained only in the very tip in this spore. Then Darwin hypothesized that in Madagascar there should be a moth with a proboscis long enough to reach the nectar hidden inside the bottom of this floral spore. And it occurred in 1862. And only 41 years later, a moth was discovered in Madagascar and was historically named as Xanthompa morgani predicta, since its existence was predicted by Charles Darwin more than 4 decades earlier of its discovery. And all of this because of a nectar in a nectary. So it's amazing. So nectar is really ubiquitous among flower plants. It's widespread in many plant lineages and many ecosystems, and make a very important link between plants and their pollinators. And most plants, we know, rely on pollinators for sexual reproduction. However, until less than 15 years ago, we didn't know how many flower plants were pollinated by animals. However, Jeffrey Allerton and his colleagues provided us with this estimate in 2011, and they showed that about 87% of all angiosperms globally, they are pollinated by animals. Also, he showed that there is a latitudinal trend where the proportion of animal pollinated plants increased towards the tropics. And since the publication of this study by Jeff Allerton and colleagues, I was very curious, and this study also inspired me a lot because you know the importance of nectar, but we didn't know how many flowering plants pollinated by animals were nectar producing, because we have lots of different kinds of floral rewards produced by plants. But since nectar seems to be so important, how many flowering plants are pollinated by animals produce nectar? And I was very curious about this because many authors along the last decade have repeatedly claimed that most insects visiting flowers, they are looking for nectar, often concluding that most flowering plants were nectar producing. However, we don't know, we didn't have this estimate. And to answer this question, it started in a classroom. Together with some friends, we teach a course titled Foundations and Frontiers in Pollination Ecology. So we used to teach every year before the pandemics. And in this course, at the end of the course, the professors provide the students some suggestions, some questions related to the subjects covered during the course. And in that year, I suggested the students to try to analyze how many flower plants were nectar producing. Because the final assessment of this course, the students need to write an essay, a paper, or a review in one of these subjects. In this course, despite being a graduate course, many undergraduate students, many undergrad students are allowed to take part of this course. And in that year, Caio Ballarin, which is here today, and today is my postdoc, but at that time he was an undergrad student, he chose this subject, and he accepted the challenge of trying estimating how many flowering plants were nectar producing. So, together with some lab mates, some students from my laboratory, Caio not only delivered the final assessment, but they also continued working on this subject along the following years, even during Caio's PhD, and then they provided us with this estimation. So, we made a compilation of studies indicating floral nectar production in animal pollinated plants. We use different repositories to do this, and it totally 977 studies, which included 162 communities all over the world. And as you can see here, these communities included 322 families, which is about 77% of all angiosperms families. That also include 7,621 species, which is equivalent to 2.2% of all angiosperms. As you can see here, we were able to sample at least 1% of all species within each plant family, even those species very rich, and those families very rich, like Lamiaceae, Rubiaceae, Fabaceae, Orchidaceae, and Asteraceae. So, our estimate, we showed that 6% of all angiosperms globally produce nectar. it represents about 74% of all animal-pollinated angiosperms. And similar to Jeff Allerton, we also found a latitudinal trend. However, this trend moves to the opposite direction, since the proportion of plants producing nectar as floral reward decrease towards the tropical regions rather than to the temperate regions. And as you can see here, the proportion of animal-pollinated plants increased to the tropical region. So, to better understand this pattern, we used generalized additive mixed models and model selection, and we found that latitude and altitude were the variables which best explained the distributions of nectar-producing plants around the globe. And we saw that at higher latitudes and higher altitudes, the proportion of nectar-producing plants increase, and at lower latitudes and altitudes, this proportion decrease. And by now, you may be wondering that this pattern seems a bit counterintuitive. So what should explain this pattern? So Caio had a very nice idea to get a subsample of those communities and analyze the diversity of floral rewards. And then we found that at the tropical region, where we have the higher diversity of plants, we also have the higher diversity of different floral rewards. In the tropical region, for example, we have much more plant producing pollen, producing oils, producing perfumes, producing resins, tissues, and other kind of different resources. So, since the diversity of the floral resource are higher in the tropical region, nectar loses its predominance in these regions. However, in the higher latitudes, the predominance of nectar increases only because of the lower diversity of floral rewards. So this explains this interesting pattern that we have found. So to conclude this study, we found that 6.6% of all angiosperms are nectar producing, and it represents 74% of all animal pollinated angiosperm, and there is a very interesting latitudinal trend. So it's an interesting macroecological pattern, and I really enjoy studying process and patterns from a macroecological perspective. However, at heart, I'm a naturalist. And as Edward Wilson once said, biodiversity research requires more boots on the ground. So, quoting Charles Darwin, I must beg permission to give you 3 empirical illustrations to make it clear how I believe natural history studies can contribute for a better understanding of ecology and evolution of plant pollinator interactions mediated by nectar. So, let's go to the first example. If you are a pollination biologist, I bet that if you stumble on a plant like this, you would wonder, good heavens, what animal can pollinate it? So, meet Cibalium fungiforme. Cibalium fungiforme. It's a fungus-like holoparasitic plant from the family Balanophoracea. And this plant is monoecious. It's a monoecious species which produces male and female flowers in different inflorescences, and each plant can harbor many inflorescences, and each inflorescence can secrete over 12 milliliters of nectar. 12 milliliters. Look, this astonishing amount of nectar, 12 milliliters in only a few days. It's astonishing, the amount of nectar that this plant produces. And to continue this story, I also need to say that this story also began in a classroom. 10 years ago, I created in my department an undergraduate course called Integrated Projects in Ecology. It's a course on project-based learning in ecology where students develop a small project, a small hypothesis-driven project, and then we go to the field to test their hypothesis. Then I present to the students this plant, and they need to study pollination syndromes. And so, after doing this in the classroom and write a small project, they hypothesized that this plant would rely on rodents or non-flying mammals as the main pollinators of this plant. So, to test this hypothesis, we bought on the internet second-hand old cameras equipped with infrared lights for night vision to leave the cameras overnight in the field to try to observe the potentially nocturnal visitors. We also used empty plastic ice cream containers to cover the cameras and protect it against the humidity over the night. And early next morning, we went to the field and start make our observations for diurnal visitors. And then we had our first surprise when we saw hummingbirds coming to the ground to drink the nectar. And we never imagined that hummingbirds could behave like this and come to the ground. Together with hummingbirds, lots of insects like these wasps, they also came to drink nectar and also to eat pollen. However, while we were doing the field observations in the forest, some students stayed at the laboratory to observe the videos recorded last night, and then we got our best surprise. We found that opossums came to drink the nectar of this plant. So you can see these shiny drops here. All these shiny drops are nectar, and the opossums came to drink this nectar. And we got really excited by seeing this. We also saw some rodents, but opossums were the most frequent visitors during the night, stepping on the flowers, licking the flowers to get some nectar. And it was very interesting. We were pretty sure that we had found something very new, and I was very excited, and I started sharing this find with my colleagues in Brazil, and then I learned that my colleague Patricia Morelato had studied this plant 30 years before our observations. However, she didn't see any visitors in that time. And then I sent her the videos, and she replied me with a voice message as excited as we were when we were in the field. And then she also sent me her data in a drawing that she commissioned almost 30 years ago to represent what she imagined would be might be opossum visiting this plant. Then we published this study together with our undergrad students. Anna was on vacations and came to the field to help us also. So we published this study together with our students. And what was very exciting as well was that as soon as the study was published, it called the attention of the scientific media all over the world. And here, I couldn't help but mention that this study was conducted during Bolsonaro's government, a dark period in Brazil where our universities were under attack, during which our Minister of Education was accusing our universities and our students of causing disturbance. So, while the Brazilian government was treating our students as troublemakers, the scientific community was calling them Brazilian scientists. And it was very beautiful. It gave us a lot of hope. Unfortunately, this dark period has gone, and hopefully this dark period will never happen again in our country. So, moving forward, I will present to you the second example. As you can see here, this fungus-like holoparasitic plant, the plant is covered, inflorescence is covered with scale-like bracts. So during the first study, we hypothesized that the uncovering of the inflorescence would depend on a mechanical removal made by opossums. So we hypothesized it in the first study. It was published. And to test this new hypothesis, we went to the field during the pandemics. We went to the field and we got new cameras, Bushnell Nature View cams, and we could leave these cameras in the field over several weeks. And then we took these cameras to analyze if the opossums were able to remove these scale-like bracts, exposing the flowers. And yes, opossums again came over, and as you can see here, with their nose, opossums did remove the scale-like bracts. But we also had another interesting surprise, because the ruby-crowned tanager also was able to remove the bracts, less efficiently, but they also removed these bracts. But now where it gets really interesting, because also after the bracts are removed and the inflorescences are uncovered and nectar are exposed because of the removal made by opossums and birds, we got our hugest surprise because we saw these guys. It's the ruby tanager, crowned tanager, but you saw these guys showing up to drink the nectar. The Pallas long-tongued bats come to the ground, land on this plant to enjoy the abundant nectar. So this is the only case outside New Zealand where you can see a bat come to the ground to drink nectar in a ground flowering plant. So it was really amazing, especially because the bats, they land on this plant and lick the nectar, eat also pollen, and when they land on this plant, they get covered by pollen like this. So bats may act as a very good long-distance pollen dispersal among different populations inside the forest. So what we found here is that opossums and birds remove this inflorescence, allowing the bats also to visit the nectar, the plant to drink nectar. So what we are seeing here is a kind of facilitation process involving pollinators that share a common plant. Then we published this scientific naturalist paper titled "Opossums and Birds Facilitate the Unexpected Bat Visitation to the Ground Flower Cibalium Fungiform." So we have here a unique case of facilitation involving pollinators sharing a common plant. And going to the end, let's go to the third example. After the plant gets pollinated, it stops producing nectar. What is quite normal, quite expected. It stopped producing nectar. However, what we did not expect is that when fruits are matured, the infructescence is starting producing nectar all over again. what may be the use of this nectar in the fruit essence, you may ask? We cut the fruit essence, and as we learned yesterday in Clay's talk, all of these black dots are starch grains that originate nectar, this abundant nectar. But by cutting the infructescence, also the inflorescence, we discovered that the whole structure is a massive nectary. So you may be looking at what may be the largest nectary in plant kingdom. So it's really huge, a huge structure. Another very interesting thing is that when fruits are mature and ready for dispensation, they burst out from the infructescence. And when they are bursting out from the infructescence, they get covered by nectar and maybe some kind of mucilage, what make the fruit quite sticky. And then we also left cameras over there. Where's the video? It's not playing. Unfortunately, it's not playing. Okay, it's okay. So the ruby crowned tanager also come back to this infructescence to eat the abundant nectar produced by the infructescence. And while drinking this nectar, the fruits get attached to the bill of these birds. Then when they fly into the forest, they may disperse these fruits. Here, there was a very nice video with a Brazilian gracile opossum also visiting this plant. And together with opossums, they also come to the plant to drink the nectar, and then they remove the seeds. And here, there was a video with a mom mice with its puppy drinking the nectar all together, and also they may remove the seeds and spread across the forest. At the end of the reproductive season, we can see in the ground of the forest the plants scraped just like this, what means that a lot of animals came to drink the nectar and remove the fruits. However, we have a lot to learn yet with this plant, and now my master's student, Arthur Gasparindo, is helping us to unveil the next mysteries about Cymbalio fungiform. So, in conclusion, more than 100 years ago, Charles Darwin realized that a little sweet juice secreted by plants could trigger a cascade of evolutionary chains shaping not only the flowers that produce nectar, but also the animals that feed on it. Nectar is a key ecological link between most angiosperms and their pollinators, sustaining interactions from individual organisms to community and ecosystems. And we still have much to learn from this sweet link. So thank you very much, or as we would say in Brazil, muito obrigado. Muito obrigado.
Oh, thank you so much for that beautiful and inspiring talk. We have time for a couple questions. Anybody have questions? Yeah.
Clay.
Yes, beautiful talk. Out of
Thank you.
curiosity, do these flowers have any volatiles, like for the nighttime visitation? Do you smell these at night?
Yes. Lots of volatiles. We are analyzing the volatiles yet. We don't have, but it has like a fruit odor, which attract opossums, but also attract bats because they come about the smell, the volatiles. Thank you.
I guess to follow up on that, like, are they mimicking fallen fruit?
No, definitely not. Definitely not. I don't think so.
How many taxa did you count? Was there others?
It's a very good question. Thank you. So we counted so far more than 20 vertebrate species visiting these plants, including carnivores like the Eira barbara, which are carnivores that come to these plants to drink the nectar. So a lot of animals, and all of them are able to do this after the bracts are removed by opossums and birds. Sorry? No one else could remove? No, no one else can remove. Only opossums and that specific bird species. So, but the two opossum species that we recorded with this plant can remove, and only the ruby-crowned tanager are able to remove, and no other species are able to do this.
Perfect.
That's
Thank
all.
you again. Oh, was there another question? Oh, yeah. Sorry, Kanji.
Ganji, thank you.
After different visitors to the flower, which animal is more efficient in pollination?
It's a very nice question, Gandhi, because we have done some exclusion experiments to try to understand if diurnal or nocturnal visitors who are the most efficient pollinators. And we saw that the nocturnal visitors are most efficient. However, without the day visitors, without birds, for example, in places where opossums are not that common, only birds remove the flowers, allowing bats to come and to do a very good job by pollinating these plants. So they are complementary.
I'm just curious about the biology of the plant. Is it an specialist, or is it always linked to a tree in specific?
All of this are yes.
It's not specialist. It can use the roots of several plants in the forest. Some trees, but most of them are lianas, huge lianas in the forest. So the most abundant and frequent host are lianas in the forest.
Fantastic. Thank you so much. This is
Thank
really
you, thank you.
yes. Okay. Oh, yeah. Oh, good. Yes. So our next speaker today is an old friend, Luke Nikolov, who was here as an undergraduate and a PhD student. He is now at the Indiana University Bloomington, and he's going to be talking to us today about floral nectary development and diversity through a single-cell lens. So thank you, Luke.
Thank you, Elena, and thank you to the organizers for bringing us all together. I'm really delighted to be here. Of course, I'll be talking about floronectaries, and they're quite diverse, as we all know. Many different kinds of structures, not homologous to one another. So the floronectary is really a functional definition, and this is what unites all of them, these sweet droplets of nectar that we saw all about this. So from Jan and Irene's review, we know that the nectaries could be associated with different floral organs. We have them on the perianth, on the gynoecium stamens, on the receptacle where all floral organs come along, and they're organized in different ways. Some of them are epithelial, so they're only a single cell layer that would be secreting all the rewards. Some are much deeper, they're mesophiliary, so they contain many cell layers, and they come out, and also the mode of secretion is very different. So if we map this diversity, or more like eyeball it on a tree of the angiosperms, we can start to recognize some traits, some trends that exist. For example, in the anagrade here, we have mostly perianth or staminal nectaries. In the monocots, most of the nectaries are associated with the carpel. In this group of plants, the eudicots, we see a lot of receptacular nectaries. So there are some patterns here that are very interesting. But because they're organs that are associated with different floral organs, they have different organization, we can hypothesize that they are independently derived, or they are not homologous to one another. And just to orient you here with the tree, here is Aquilegia, the sister to the eudicots, with a nectary here, at the tip of the spur. And we'll spend a lot of time talking about the mustards here that Clay so beautifully introduced us to, that have mesophiliary nectaries at the receptacle, at the base of the flower. And here, again, these structures in Arabidopsis, there are two kinds of structures. So there's this dimorphism that exists, larger lateral nectaries here along the lateral axis, and median nectaries along the medial axis here. Different numbers. They appear to be functionally differentiated. And most of the work is done by the lateral nectaries. So we're developmental biologists, and we're interested in the developmental events that are involved in specifying these organs and the cell types that are associated with them, also the elaboration of these organs, and ultimately how they end up doing their function. All of these processes are developmentally regulated. And the way to look into these processes, we've taken advantage of a revolutionary new technique that allows us to study the transcriptomes of individual cells. So the way that works is we take flowers, we soak them in an enzymatic mix that would break the cell wall and the middle lamella. When that's gone, the cells, they take spherical shape, like droplets of water in space. And then we take this suspension of cells that we call protoplast. We flow them on a microfluidic device here that would unite each protoplast with a barcoded gel bead into these gems. Then we lyse the protoplast. That releases all the RNA that's within them, gets soaked up by the beads. Library preparation takes place. And that's how we can capture the transcriptomes of thousands of different cells. So we use this technique as an in-way to study nectary development, but because this is not a targeted approach, this is one of the reasons why we used it. We've actually used it to generate a whole floral atlas of all the different cell types and cell states that build up the flower. And this is how these atlases look like. So each dot here is a cell, and cells are colored according to their similarity of transcriptional profiles. So we here have about 90,000 dots, 90,000 different cells that correspond to cells collected from different floral organs, different stages of development. Because we've done different libraries, we have that information pretty much nailed down so we can connect everything together. And at that level of resolution, we have about 88 different cell populations which correspond to different cell types or cell states. So to understand which are the different cell types and cell states, here on this atlas, basically connect the transcriptional profiles with what we can see under the microscope. We've used these three complementary approaches. We've used marker genes that define specific cell types. For example, here histone H3 would be marking the cells that divide very actively. So we know that this population 2 here is dividing cells, and there are a few different loci, a few different regions in this map that are dividing cells. Then we've also used marker genes, newly derived marker genes that characterize these different cell populations, like this particular reporter here, a picture I've taken here at the Arnold Arboretum, the middle cell layer in the anther cell wall. And finally, a technique that is based on in situ hybridization, multiplex in situ hybridization called Murfish. In the case of the nectary, we used the complementary fourth technique that allows us to identify where is this population. And we've taken advantage of the Krabs claw mutant, this gene that Jan was talking about. It lags the nectary. So here the nectary in the wild type. Here you see it on this X-ray computed tomography image. And here they're gone. They don't exist in the Krabs claw mutant. So if we generate transcriptional spaces or transcriptional libraries from both the wild type and the mutant, and we align them, we overlay them on top of one another, we can see that the blue cells, the mutant cells here, they're on top of the yellow cells here, the mutant, are on top of the blue cells. So they align pretty well. So they're pretty much similar. We have the same cell types and cell states in the mutant and the wild type, with one exception. And this is this lonely island here that's present only in the wild type cells, but not in the mutant cells. And when we do derive markers of this population and generate reporters for this particular population, we see reporter activity in the nectary. So this is the nectary cell transcriptome. We have nectary cell transcriptomes, not just the transcriptional profile of the nectary as an organ, but of hundreds of individual cells. And when you have that many cells, well, you might want to ask how different are they from one another. You want to compare the different profiles. Here we've used a computational approach that would allow us to arrange these cells in a progression, diffusion map approach. So that's basically a way to see how similar are these different transcriptional profiles to a root we have assigned. So that is our measure of how far away are the different cells from the root. So the further away they are, the more different their transcriptional profiles. So the further away along a pseudo-time coordinate they are. And that's how this space look like. If you do diffusion mapping of the transcriptional profiles of the nectary. So potentially here on this pseudo-time plot, we have precursors and then a very narrow isthmus that connects it to the nectary. But remember, I have libraries of different stages of development of the flower. So we can, because we have the identity of these different cells, now we can align this, overlay absolute time with this. And here you see cells from stage 10, 11 flower, 11, 12, 12, 13. So this is not just a pseudo-time progression. This is an actual developmental time progression, which is very valuable to us. So now we can assign absolute time to it. So here is the expression of Crab's claw in this particular plot. We can see the narrow isthmus that connects it. And here is the spatial transcriptomics platform that we use for Crab's claw. We use the reporter that's in the context of a bacterial artificial chromosome that will capture all the regulatory elements that are involved in the expression of this gene, and studied the expression of this reporter using confocal microscopy. And you can see that these three cells here, they're destined to become the nectary. And that decision takes place very early in development. Like soon after the stamens are actually formed. Then at later stages of development, we start to see that the reporter is actually not expressed as in interrupted pockets like you would typically see them in the SCM, but it's actually an interrupted ring of expression that is outside of the stamens. And at later stages, even further, when we have an nearly open flower, this is how the reporter shows expression. Practically marks the nectaries. It's almost like a circle. Well, two hemicircles that are associated with one another, and they have these protrusions at the two planes, one in the lateral, where the larger lateral nectaries would be formed, and one in the medial, where the four smaller medial nectaries would be formed. So I really like Clay's definition of a nectarium. So these are not isolated glands. This is actually what appears an independent world that forms in the flower. And we consistently see that in our imaging. So how is that world formed? So each one of those hemicircles is a rather complex three-dimensional shape. It's almost horseshoe shape, but the edges of the horseshoe kind of like diverge out, and that's what forms the individual medial nectaries. So how is this formed? How is the identity of this world formed? In a way, well, a good starting point is to look at the floral organ identity genes. And this is a known information. C-class genes are involved in specifying nectary identity. And if we map the expression of the three C-class genes, the gamma shutterproof 1 and 2 here in the nectary, they're expressed in the nectary. We see that. Then we did the same thing for the B-class genes, and one of them, AP3, is expressed in the nectary throughout development, but very surprisingly, the other, Pistillata, appears to be specifically excluded from the nectary. And this was very interesting to us. because the ABC model of floral development, perhaps a cliche among this audience, is actually a combinatorial code that specifies the identity of the different floral organs. And here we have the B-class activity that specifies petal and stamen development. It is a result of a heterodimerization event between two partners, AP3 and pistillata, and that's what actually gives rise to the B-class activity. Well, if you don't have B-class, if you don't have pistillata expression, you compromise B-class identity, and that actually is effectively creating another developmental domain that interrupts the B-class identity, and based on this SEM, that's how you see it between the petal here and the stamen. So that is where the nectary is. So we would argue that this is a different floral category that is outside of the canonical floral organs, and we're currently studying, investigating this model further using some overexpression studies we can chat about later. Okay, the second stage, the overexpression, the elaboration of this structure. I already showed you the horseshoe here. This is an SEM of the same top view here, a side view. So here's the lateral nectary. So how do you get this complex structure? It's a structure that develops very quickly during development. So nectar secretion appears to be very tight window, very tightly regulated window. Well, development itself appears to be also quite fast. Within 72 hours in the lateral plane, we start from a flat surface to a nectary, to a fully developed nectary with stomata here ready to secrete. And Andrea Gomez, a postdoctoral fellow in the lab, was very interested to study this process in real time. So she used time-lapse imaging. The way she's done it is she dissects an inflorescence like this until she reaches to a point where the nectar is about to be initiated. So here's one of those flowers. She removes one of the sepals, and here is the stamen, and here's where the nectar is going to happen, where it would form. Then she mounts it in a media, and then takes images under the confocal over real time, and that allows us to follow the whole developmental process from a flat surface to a fully developed organ, and that is how it looks like. So that is the same gland, medial gland in this particular case that we followed, and here is a stomata that has formed already. So this has been really cool to see, and also quite insightful, because yes, the nectaries in Arabidopsis are very small, but this, in this particular case, is an advantage because we can actually see inside them. We can see in the functional part of the gland. So this cannot happen with large nectaries. So that is actually a benefit here. So here is on the surface, on the maximum projections, here a section, so you can actually see inside these cells, and we can actually follow individual cells. We can follow lineages of cells because it is basically the same gland. We can see how many times it's divided, in what orientation, how much it grew. And here is another example that actually, because we can look into the gland, we are not just looking at the outside, but we can see on the inside, and we can see that actually here, two different cell types that make up the nectary, the epidermis on the outside, the L1 layer, and the actual nectary parenchyma, they're rather different. They're qualitatively different patterns of growth associated with the two processes. For example, in the epidermis here, we see this very precise and very regulated process of cell volume increase along a circumferential band that would be lifting up the structure up. But if you look underneath, it's significantly more regular. The cells are smaller, but they seem to be increasing in volume much more regularly. So if you imagine this process is a process of like not just lifting, but expansion from the inside. And we can correlate this with the number of divisions that have happened. Divisions in the nectary epidermis seem to cease earlier than within the nectary parenchyma. So that is this explosive growth, as I was mentioning initially. So rather coordinated process of cell division and self-expansion that would actually sculpt the gland. So who's responsible for all of this? Well, remember we have hundreds and hundreds of nectary cells at different stages of development, and when you have hundreds of cells, you can study gene co-expression. So, and here I'm showing the co-expression between two genes in individual cells, Krabs claw and another gene, STM, an important regulator of growth. And each one of those dots corresponds to the expression of these two genes in individual cells. If you see a cloud like this, we refer to this as co-expression when we represent it as two nodes connected by an edge. So that's the co-expression relationship. And if we repeat this process iteratively over, let's say, all differentially expressed transcription factors in the gland, we end up with a network like this. Here's the connection that I showed you. But this is a gene co-expression network of the secreting nectary, including all transcription factors and including genes that are known for nectary regulation, like Krabs claw here showed you shatterproof, AP3, MIP57, a gene that's important for nectar secretion, as Clay has shown in the past. New genes, unexpected, YABI5, the circadian regulator CCA1 that's involved in nectar production as well. So here's shoot meristemless on the developmental trajectory, and that's the correlation with Krabs claw. So ideally, we would like to study what shoot meristemless does in the nectary. So how does it sculpt growth? But this is not exactly a trivial task because it's called shoot meristemless for a reason. The mutant lacks shoot meristem. It gets exhausted during development. And this is how some of our CRISPR mutants look like. They just don't have a meristem. If they don't have a meristem, they don't have flowers. We cannot study the nectaries in this particular case. So we need another approach to perturb gene expression in this particular case. And we've taken advantage of another CRISPR strategy, but this is a tissue-specific CRISPR strategy. So just to develop this technique, this technology, we're using this particular construct here. We are using a fragment from the promoter of Krabs claw, a 3.2 kb region that that basically recapitulates the same expression we see with the bacterial artificial chromosome here. So here's a reporter. Then we have a fluorescently tagged Cas9 nuclease. So that is an enzyme that will come and break specific regions of the DNA. Because it's fluorescently tagged, we can follow this during development. We can see it, and you can see it expressed here under this particular promoter at a very specific place. So it's not throughout the entire flower. It's not going to make mutants everywhere. It's going to make mutants only in the nectary. And finally, we use functional guide RNAs, in this particular case as a control, just targeting the ones that would go towards the nectary gene Krabs claw. So these are guide RNAs that would specifically target the Cas9 to the endogenous Krabs claw locus, break it there. And how does it look like? We've tested the efficiency in regular CRISPR in this particular case, and they look like the CRISPR mutants there. Okay, so here's the wild type. Here's the Krabs claw mutant. How do the tissue-specific CRISPRs look like? Well, they phenocopy the Krabs claw mutant, so we've achieved a pretty efficient knockout in this specific developmental domain. The nectaries are small. They're here at the base. You cannot quite see them. But if you use actually a reporter that would be marking this developmental domain, well, you can see that the domain might be formed, but there is no outgrowth. So if we break Krabs claw, the nectary domain might be forming. There is no outgrowth. Well, occasionally there is, because that's a somatic knockout. So you would have some level of leakiness here. But nonetheless, this thing works very efficiently. We can get biallelic knockouts in the first transgenic generation. So that's great. We can use that technique to see what STM is doing. So if we have guide RNAs targeting STM, and indeed, if you do this, yeah, the domain is formed by the reporter, but there is no outgrowth. So this is a powerful way to study and perturb gene expression in a specific developmental domain, and a very good way to study genes that are essential for development, also genes that are very broadly expressed. If you want to know what this gene is doing specifically in this cell population, you just drive the Cas9 in this developmental domain, and that's how you can obtain functional information. Finally, with the nectar secretion, we don't study nectar secretion specifically, but well, we really like structure. We like anatomy and morphology. So we've looked at the cell changes that happen with transition from pre-secretory to a secretory gland. And this is the project of a graduate student in the lab, Michael Kwaki. So from a pre-secretory stage, we have these cells. Here is the nucleus. So here are the starch grains that Clay and Jan talked about. And here is the secretory stage, cells that have not increased that dramatically in size, but what you've seen is this a rather dramatic secretory compartment, or we hypothesize the secretory compartment here. So we've used block phase SEM to reconstruct the architecture of these secretory cells, and they have a lot of plastids. For cells that don't actively photosynthesize, they sure do have a lot of plastids here in green, but particularly this large vacuolar vesicular compartment. And you can see from this very busy cells, they're really packed with stuff. They're packed with plastids. They're packed with mitochondrial peroxisomes. But particularly this yellow compartment here, which we don't know what it is, but it's likely involved in the secretory process. So this is very exciting. My starting point for all of this study has always been diversity. I'm interested in diversity, and I worked on diversity in the mustard family, Brassicaceae. Brassicaceae are kind of known for having a lot of leaf diversity and fruit diversity, significantly less floral diversity. Well, I don't think that's the case. If you look at these X-rays here, there is quite a lot of diversity in these flowers, but architecturally they're rather conserved, as we can say. So if we study the nectaries of these different plants, well, we see quite a lot of diversity there. The canonical floral organs, they're conserved, the architecture is conserved, but the number of nectaries, their orientation, their overall morphology varies quite a lot. So ultimately, we would like to get an insight into this diversity. So how does that happen? And our approach to that has again been to use single-cell transcriptional profiling, this time phylogenetically informed throughout the family. We've included several different samples. So here are the different spaces. This is work done by Ricardo Chavez, who in addition to working in the lab, also has a sidekick job at South Park. But he's mostly computational, and that has been a major challenge. So how do we integrate single-cell transcriptomes from different species that have diverged millions of years ago? So there is certain phylogenetic distance between them. Well, you need a common ground to compare apples and oranges. And our way to do this kind of analysis been to use an orthogroup approach. So when we study gene expression, instead of looking at the expression of specific genes, we look at orthogroups that basically allow to make a comparison between different species, not only diploid species, where establishing one-to-one-to-one orthology is relatively straightforward, but also genes, species that have higher ploidy, where establishing this kind of one-to-one relationship is difficult. And Irene, who was a postdoc in the lab, made a lot of inway into figuring out what's the best way to build these data sets of orthologous genes in the Brassicaceae. So when you take the single-cell, single-species transcriptomes and integrate them using traditional methods for integration, this is what you get. So you have the comparable base, the orthologs to compare, but ultimately the single-cell transcriptomes do not blend, do not integrate together. So they seem to be separating according to the different species, and that would suggest that you have unique cell types and cell states within Brassicaceae, a family that nonetheless shares a common ancestor and not that many million years ago. So it's quite unlikely that we can hypothesize that there are unique cell types in these different species. So obviously phylogenetic distance here is playing a game with us and doesn't allow to create a full integration. So we need another step that would be basically extracting this phylogenetic or subtracting this phylogenetic signal, which manifests as noise in our data set. That's why we use a neural network or an encoder of the neural network to actually integrate these spaces and allow us to align them the same way we aligned the mutant and the wild type in the previous example. And that's how these integrated spaces look like. Now, it's much easier to identify which are the populations that are homologous to one another and to start to look at differences. So what makes the nectary of class B panicras different from the nectary of Capsella, for example, or Arabidopsis. So here is the nectary population. I'm looking at different species here, not only Arabidopsis that don't have the same level of functional genetic resources that are available for Arabidopsis, but because I can do this kind of integration, I can do label transfer and say, oh, well, this is the same population where the Arabidopsis nectary cells are. That must be the nectary of Capsella, grandiflora, rubella, brassica, tlaspi, and camelina. So basically, this is an aligned space, and that just highlights the different cells from the different species. When you do single-cell sequencing, it's a bit of a random process of what you can capture. So we've captured quite a lot of cells from all of these species, not that many from camelina, just the way it happened. And then when we use these spaces, we can start to look at it, like what are the markers that define these different nectary populations from the different species. And it turns out that from all of these species, there are about 730 different marker genes, strongly differentially expressed genes. They seem to be shared between these species. Then in the next category, we have about 300 markers that are shared by all except for camelina. That would suggest that maybe something is different with the camelina nectary. Well, not really, because if you look at the number of cells that we've captured, only 25 cells, it's not. So we probably just don't have the power to recognize this. But I still want to highlight that 25 cells of camelina are enough to still recover about 730 markers that are shared by all of this. So this is remarkably powerful computationally. Because we have all of these spaces now, we can map gene expression of specific genes to these different populations. And here's our favorite gene, crab's claw. So you can see it's conserved, it's expressed throughout the nectaries of the different species. So that's a shared marker. Then we can look at markers that are not really shared. So for example, this SNM1 specifically expressed in the Capsella grandiflora nectaries, not expressed in the Capsella rubella nectaries, for example. So that might be one of the genes that determines the differences between these two species. Here we have TOL3, which is a repressor of agamas, specifically expressed in the Capsella nectaries. Here we have this gene LSH4, a boundary gene that's specifically excluded from the Capsella nectaries. So we have all these lists of genes that we can really pinpoint that determine expression differences between species and allow us to have a handle, an anchor to do more thorough functional studies, to understand what are the genes that determine differences between species and what is the balance between conservation and divergence in these different cell populations. In the remaining 3 minutes, I'll show you another group of plants that also develop nectaries. They belong to a different clade. These are the monocots. And they have mostly nectaries that are associated with the carpel. They're septonectaries, so the 3 carpels of the monocots, when they fuse, they create a nectary at the fusion site, and you see it here in this septonectary. So likely independently derived, they appear to be derived from the epidermis, so the L1 layer, in contrast to the different nectaries that I showed you. So what is the genetic basis of their formation? How do they form? Well, we've looked at banana as a model for this because it has the genomic resources, and this is a project that Irene initiated and Ricardo and Michael are working on. So basically, banana has pistillate and staminate flowers. It first forms these pistillate flowers. That would be the bunches of banana that we all recognize. And then all these covered bracts would be opening, revealing the staminate flowers here. So nectaries are present in both the pistillate and the staminate flowers. But the staminate flowers, they seem to have like a floral part. That's the top where the nectar is. And this base, which is devoid of nectary cells. Then the pistillate flowers, which are much more elaborate, larger, the gynoecium is larger. They have nectary cells or septonectaries throughout. So we've generated single-cell transcriptomes from pistillate flowers, from staminate and top and bottom parts, and we used our integration approach to bring them all together. So that's how this integrated space looks like. So now we can highlight from which sample these different parts came from. So how would we find where the nectary is? The nectary is supposedly not crab's claw controlled, so we cannot pick a marker gene that would allow us to highlight this particular population. Well, we're looking for some population that is present in the pistillate sample and the staminate top sample, but not at the base. We can use some candidate genes to try and reveal some of the identity of these different populations. And here we can see there's the pedicel parenchyma, some ovovascular parenchyma, the phloem. But the population that seems to be absent from the base but present in these two is this inner epidermis population. So that's likely where the nectary is. Nothing screams as nectary. It's not a separate island as the mesophyllary one that I showed you. It has more of a feature of an epidermis. We've picked plants and flowers that are mature, they're anthetic, they're supposedly secreting nectar, but nonetheless, we cannot find really distinct transcriptional features that would tell us, oh, this is unequivocally the nectar. It just behaves as an epidermis. But to try and figure out, well, maybe there is something special, that would be a way to identify these marker genes. Let's align the networks of the inner epidermis and the outer epidermis. They will be the most similar ones using the same network approach I showed earlier. And maybe that would allow us to highlight what is specific to the inner versus the outer. Well, nothing screams out. There are a few shared nodes by MIP transcription factors shared by the outer and the inner epidermis. Other than that, there are a few other motifs here that we can find present in the inner epidermis, but not in the outer epidermis. This is just something that is starting as a project, but still a lot to learn. Bottom line is, it is different. It's not like what we've learned from the Brassicaceae, and I suspect that this would be the case over and over and over again if you look at throughout the angiosperms. So certainly very exciting times around expecting us. So we'll have a lot to learn here. With this, a brief summary. I showed you our single-cell floral atlas and some multiplex spatial transcriptomics. I think we're all convinced that the nectary cell type is a great model to study cell fate acquisition and differentiation. And you can use functional perturbation using tissue-specific CRISPR to study what these genes are doing in this specific developmental domain. And I also showed you our tabulae florum, the conservation and divergence of different cell types throughout the mustard, but also other clades. With this, let me thank members of our lab. collaborators, and thank you. I'll be happy to take questions.
I think we can take one or two questions right before our break. Does anybody have a question? Luke, maybe in the back there?
Great talk. I was curious if you have thoughts, so I guess the current model for the entry origin information is that it has just CDs, right? Or I guess lost the PBI, it's lost the B set.
That is what we were proposing here. Well, it's lost B-class activity, and only one of the B-class genes is expressed there.
I see. Oh, so it's just a subset. So I guess like my question is, I guess what do you think is driving the difference between the stages and just this subclass?
Well, there must be another factors that are creating this combinatorial panoply of options, so very similar to the different axes here. And I think with looking at these different cell types, I think that's a way to look into it and to start and figure out what are the different combinations that would give you this versus that versus, yeah, we don't know overall, yeah.
Jan, do you want to?
Yeah, just a quick question about the time lapse, because I feel like that's a great system to really answer the question, is nectar stomata, right?
Uh-huh.
Yeah. Does it actually cycle, like in terms of does it do asymmetric cell divisions, and do you actually see those things come?
Yeah? Yeah.
Here we go.
Can I see your spot? Oh.
Yeah, you are the first, right? Yeah, because otherwise the online people can't hear you.
Oh, cool. Wait, I think I pushed a button. It's okay? Really? Okay. Cool. Okay.
All right, folks, thank you all for coming back. I hope you are well caffeinated and sugar spiked, less in liquid form, more in solid buttery form. Before we get started, I do want to also give a huge thank you for Wendy and Nicole for doing all the organizing. Amazing, everybody's schedule and travel and food and everything else. So if you see them out there, they're hovering by the tag table. Give them a big thank you. This would not happen without them. All right, so session 2, we are very excited. I am thrilled to be introducing Carrie Wessinger. She is an assistant professor at the University of South Carolina. She's a talented evolutionary genomicist who has truly turned Pestamine into a beautiful model of genetic, genomic, and ecological study. She takes an integrative approach to understand the genetic and ecological processes governing adaptation, mostly in the context of pollination biology. And I think one of the most amazing things about Carrie is that she studies this real plant that's real hard to work with, and yet makes it seem so easy.
It's not easy.
It's not easy, but the results are like, how does she do that? So I'm very excited for Carrie's talk.
Thanks, Robin. All right, thanks so much to the organizers. Again, this is a really fun symposium. All right. I just have to turn it on. All right, do it this way. All right, so evolutionary shifts from bee to hummingbird pollination are common in North American flora, promoted by the efficient pollinating ability of hummingbirds, of course. And these transitions involve the convergent evolution of these bright red, narrowly tubular flowers that produce lots of nectar. And this set of traits is the hummingbird syndrome. And I think that these shifts to hummingbird pollination are a really nice way to study complex adaptation because these require the coordinated change in multiple floral traits. Yet, these transitions have occurred many times across different lineages, allowing us to make fun comparisons across taxa. And also we can usually find really closely related pairs of species that show alternate floral syndromes, like bee versus hummingbird syndrome, which means that these evolve rapidly and we can kind of address the genetics of these complex shifts. And so the genus Penstemon really exemplifies this pattern of convergence. So this is a large North American perennial wildflower genus. It has almost 300 species and it's rapidly diversified. Most of the diversity is in the western United States. And it's diversified in a lot of different ecological traits, but a really characteristic pattern is these parallel evolutionary transitions in pollination syndrome. So most species are adapted to be pollinated by bees, but there have been many evolutionary transitions to these hummingbird syndrome flowers. And to sort of visualize this pattern of repeated evolution, I'm showing you a phylogeny that sampled about a third of the total species diversity in this group. And I've marked the hummingbird pollinated lineages with red dots, and you can see that there are like lots of shifts to hummingbird pollination that are distributed across many separate lineages. And they're often sister to bee-pollinated species, suggesting that these transitions in Penstemon have occurred really rapidly. And so these features make this genus a really nice system to ask some questions about the genomic basis of complex adaptations. So some of the questions that me and my lab mates and collaborators are interested in is, what is the genetic architecture of these evolutionary shifts? How are these divergent adaptations maintained in the face of gene flow, which could be relevant for closely related pairs of species that might co-occur? And then what's the source of genetic variation for these repeated shifts? And here I'm showing, this is like a kind of a cartoon phylogeny that shows like a focal clade that my lab's really focused on that includes 4 separate origins of hummingbird pollination. And I'm gonna start with a story about a focal species pair that I've been working with for quite a while now. And this work has been collaborative with these lovely people, Lena Heilman and John Kelly at the University of Kansas. Okay, so these species are Penstemon barbatus and Penstemon virgatus. They're sister species. They have different floral syndromes, and they have overlapping distributions in the Southern Rockies and Sky Islands in Arizona and New Mexico. They can occur sympatrically in some places, but obvious hybrids that show intermediate phenotypes are super rare. So for a long time, we've known some basic features about the genetic architecture of this floral syndrome shift. So we previously crossed these species and mapped QTLs for floral syndrome traits. We found that nectar volume had a single QTL of major effect. We also found a single QTL for flower color. And so both of these are located on the same chromosome, chromosome 1. And of course, we know the gene for flower color because it's kind of the low-hanging fruit of plant evolutionary genetics. So this flower color shifts from bluish to red flowers in Penstemon involve a shift in the type of anthocyanin pigment that's produced. So this is a very simplified version of this anthocyanin pathway that shows just a key enzymatic reaction. So these purple flowered species produce delphinidin-based pigments, this bluish pigment. Hummingbird pollinated species tend to produce this red pelargonidin-based pigment. And this gene right here is our flower color gene. Basically, this gene has been inactivated through loss of function mutations to the coding sequence. And some work quite a while ago now showed that this type of loss of function mutation is a really common route to red flowers in penstemon. So we saw that in 12 other red hummingbird pollinated species, we see a variety of loss of function mutations to this key enzyme. So a variety of inactivating mutations, and in many cases, this gene is like extensively degraded. And more than that, in every red flowered population that we've looked at, we often see different loss of function mutations like segregating within a population. So perhaps it's not surprising that these loss of function mutations are underlying this common route to red flowers in this group, because there's many ways to break a gene. But this large mutational target size for this type of gene might facilitate rapid evolutionary transitions. So why am I talking about flower color? Well, in a moment, I'm going to tell you about a potential candidate gene for some of the other floral traits. And it remains an open question whether mutational target size is also sort of facilitating rapid evolutionary transitions in these other floral traits. So coming back to these QTL results we found, in addition to our major effect QTL for nectar production and flower color, we also found a major QTL for flower width. And these three traits really sort of critically define the hummingbird syndrome in this group. So having these bright red, narrowly tubular flowers that produce lots of nectar really gets you a lot of the way there. And so the fact that these have perhaps only involved large effect alleles could perhaps have streamlined the rapid assembly of this syndrome in this hummingbird pollinated species. So I'm going to come back to these QTLs in a moment, but I want to point out that we did measure some other floral syndrome traits that didn't have a large major effect QTL. And so here I'm just showing an example LOD trace for style length. Okay, so some aspects of floral syndrome divergence involve major effect alleles, and maybe others are much more polygenic. But how are these complex differences maintained in the face of gene flow that could potentially like break apart and recombine these combinations of alleles? And this is a real potential issue for closely related species that may co-occur in secondary contact. Now, one way to avoid this is if all of your alleles for hummingbird syndrome happen to be sort of bundled up in the same genomic region in tight linkage, then this would allow those alleles for hummingbird pollination to be sort of jointly inherited during a hybridization event. But if we come back to our QTL results, we get a sense that this doesn't seem to be the case, right? These QTLs aren't all sort of like right on top of each other. And to get a better view of this, here I'm showing a recombination map across our 8 chromosomes in penstemon. And so this green trace here is showing genetic distance in centimorgans against physical distance in megabases. And so we see on each chromosome there's a region of low recombination. These are these plateaus, and then there are these steeper bits are regions of high recombination, and I've overlaid the peak LOD positions for our floral QTLs onto this plot. And we see that these QTLs are like sort of sprinkled throughout the genome in these high recombination regions. Maybe there's a little bit of pileup in some key regions, but it suggests that things may in fact get quite scrambled if these two species were in secondary contact and able to hybridize. So to kind of look at patterns of genomic divergence between these two species, I've sampled now many populations across their range. I think I'm up to over like 50 populations now. And we've begun generating whole genome data. And so we call genotypes, and we're interested in well, we started by looking at pairwise genetic distance between these individuals to understand how much variation is sorted out between these two species. And we found that these two species form this species complex with porous boundaries. So here I'm showing you pairwise genetic distance, or DXY, between populations as a neighbor joining tree and as a multidimensional scaling plot. And on this plot, each point represents a population, and the distance between points reflects average genetic distance between these populations. And we see this interesting pattern where our Penstemon barbatus populations located in the eastern portion of the range in Colorado and New Mexico are more closely related to nearby Virgatus populations, the bee-pollinated species, than they are to other populations of barbatus out west in Arizona. And so it seems like there's been a lot of mixing between these species, at least at a regional scale, despite them having these really strongly divergent floral adaptations and having these distinct pollinators. And so we'd like to know sort of where in the genome are these two species differentiated, and in other words, like what's holding Penstemon barbatus together as like this really recognizable species. And it turns out that there's actually very few fixed differences between these species, at least at a range-wide scale. So we decided to take this genome sequence data and conduct a GWAS, basically treating pollination syndrome, or like species identity, just as a binary trait in a case control framework. And here are these SNP associations. So we see that there are just these small localized regions of the genome that are like strongly diverged between these two species. So these are SNPs that are strongly associated with us saying, okay, this is Penstemon barbatus versus this is Penstemon virgatus, just based on their floral syndrome differences. And again, I've put the floral QTL mapping results. So I've overlaid these peak LOD positions for floral trait QTLs onto this plot with these colored lines. And we see that there are some cases where really strong SNP associations are like right on top of our floral trait QTLs. And so let's zoom in on a couple key regions. So here on the left here, I have the region that includes the flower color gene and our nectar volume QTL. And then here on the right, I have the region of our flower width QTL. And we see these nice narrow chimneys of SNP associations that are really close to these inferred peak LOD positions. Now, genome-wide linkage disequilibrium is pretty low, especially considering that these individuals represent different species. So linkage disequilibrium decays to below an R-squared of 0.05 within 10 kb. So basically, we have these like narrow genomic regions that are strongly associated with these species differences, and they're in strong LD with each other, as you would expect if they were like really tagging the causal genes underlying floral syndrome. But they're like embedded in these super high recombination regions, which actually is pretty convenient for our efforts to actually localize the genes responsible for these differences. And so if we zoom in even further, we begin to resolve these peaks at the scale of a gene or two. So on these zoomed in plots, if you can see these light gray shaded areas, these are annotated genes, individual annotated genes. And so at our flower color gene, comfortingly, we find strong SNP associations right next to the causal gene, sort of a proof of concept here. Our nectar, the SNP associations at our nectar volume QTL localize to a gene that's annotated, well, actually, it was an uncharacterized protein in the annotation. And we blasted it, and it comes out as a lipid transport protein. So penstemon nectaries are a patch of glandular trichomes on the stamen filament tissue. And so Lena Heilman, my collaborator, former postdoc advisor, thinks that this protein could generally be involved in secretion processes, even though it's called, from our blast result, a lipid transport protein. But curious to hear what the experts think about that hypothesis. And to kind of complete the story here, flower width, our SNP associations at this flower width QTL region localized near the apetalate-2 transcription factor, which has a known important role on petal development. So we think that's a really nice candidate gene for our flower width QTL. Okay, so to sort of take a step back here, our population genomic data describes this really sort of surprising pattern. These two species are really unmistakable in the field. They have this constellation of floral differences that immediately diagnose species. But if you look at the genetic data, it looks like a single well-mixed species, and you almost have to be like right on top of a causal gene to really distinguish them using genomic data. And so we have a lot of remaining questions. So there must be strong selection acting on these combinations of traits combined with assortative mating conferred by the distinct pollinators. So we're curious, so how much of this is shared ancestral variation that's still sorting out? How much is being shared through current gene flow? And to begin to understand the evolutionary history of these specific alleles, I've begun inferring ancestral recombination graphs. So what is the ancestral recombination graph? So this is a representation of the evolutionary history for a sample of sequences. So it includes all of the coalescent events, recombination events, and mutation events. And we can think of the ARG as a set of trees that are highly correlated with neighboring trees, separated by a single recombination event, so that the genealogy changes as you move along the sequence. And knowing these genealogies it is pretty powerful. So we can ask, what's the age of an allele of interest? Is it experiencing a selective sweep? Or just some more general questions like, what's the demographic history of our population? Can we estimate recombination maps or mutation rates? So I'm using a software called ArgWeaver that attempts to infer the ARG from sequence data, and it yields estimates of local trees along a sequence, along with the recombination events. And so I've started small here. So I'm taking our 3 focal regions, including 5 kb upstream, 5 kb downstream, and I've taken just a subset of my individuals that really like samples a lot of the diversity within this species complex. And so just as a reminder, this is our genome-wide population tree kind of turned on its side here, showing this relatively deep divergence between the eastern clade of our Barbados sequences and the western clade. And what the concept here is, I'm sort of asking, are there regions near these key genes where we see all of these Barbados sequences are monophyletic and they share a recent coalescent event? In other words, a small time to the most recent common ancestor. So something that looks sort of like this, right? That would look kind of sweepy. So I'm going to walk through the results for this lipid transport protein that we think may play a role in nectar reward. So here is the output of ArgWeaver, and what is plotted here is the time to most recent common ancestor as you move along this sequence. And it looks like a step function because it's basically broken up by recombination events. The red line is showing the time to most recent common ancestor for the Barbados sequences, And the purple line is the time to most recent common ancestor for all of the Virgatus sequences, the bee-pollinated species. There's actually a black line on here that is usually behind the blue line. That's the time to most recent common ancestor for all of the sequences in this sample. And this shaded gold area demarcates regions where all of the Barbados sequences are monophyletic to the exclusion of the bee-pollinated species. So at this lipid transport protein, we basically see this region of monophyly for the Barbados sequences that is also a region where this time to most recent common ancestor for the Barbados sequences is like pulled down. And the interval is fairly narrow. It's just a couple kilobases. So let's look at a couple representative trees. So outside this region of monophyly, the sequences, the genealogy for these individuals is all mixed up, which is consistent with everything I've just told you about how mixed up this species complex is. But in this region of monophyly, we see a super recent time to most recent common ancestor for all these Barbados sequences. And it looks kind of sweepy. Okay, so to just show you what's going on at these two other regions, we did the same thing for this apetalate2 gene, but notice that the region of monophyly, at least in the coding sequence, is really narrow here. The local tree that's estimated here also shows a recent common ancestor for the Barbados sequences, but perhaps it looks like a little bit more of a gentle sweep. And then finally, at our favorite flower color gene, this is interesting. There's actually no region of monophyly for all the Barbados sequences in the coding region. And I mentioned to you that sometimes there can be multiple distinct loss of function mutations that are segregating even within populations. So perhaps we're not surprised by this. But a little ways upstream, there is a region of monophyly that's about 1 kilobase upstream. And so we can look at the local trees here. So, sorry, in the coding region, it's all mixed up. But in this region of monophyly upstream, we see Barbados sequences form these two lineages that have a pretty deep divergence from each other. And so, you know, this ancestral recombination graph approach seems really promising for kind of disentangling the evolutionary history of some of these alleles and explaining how we find this really interesting pattern of genetic variation in this system. Okay, so it seems possible that in our Barbados system there have been these separate selective sweeps that have occurred at many different loci. Our next question is, what is the source of genetic variation for repeated shifts? And I mentioned that Penstemon barbatus is embedded in this clade that includes 3 other origins of hummingbird pollination. Are these due to de novo mutation events, or has there been any role for pre-existing variation driving some of these repeated shifts to hummingbird pollination? A question we're interested in is whether there's any evidence that these hummingbird adapted alleles have moved between lineages due to adaptive integration. And this was actually a model that Stebbins actually hypothesized could help explain these repeated origins of hummingbird pollination in Penstemon and other Western North American genera that have multiple origins of this syndrome. So Luke Wheeler, a former postdoc in the lab, used whole genome resequencing data to infer a tree for our focal clade, and we're currently working on a phylogenomic analysis to quantify integration between these 4 different hummingbird pollinated species. So stay tuned on that. But in the meantime, we can at least take a peek at the gene tree for our 3 favorite genes that are the stars of this story. And look for evidence of integration. So what would that look like? Integration would be implicated if we saw really close relatedness of sequences from different hummingbird pollinated species, perhaps even monophyletic. So here are the gene trees for our 3 genes of interest, and in a nutshell, we see no evidence of super close relatedness of sequences from these different species. Each seems to form its own diverged lineage at each of these genes. But it is, of course, possible that de novo mutations have repeatedly targeted the same gene. In fact, we know that's probably true for our flower color gene. But for these other candidate genes and our other traits that make up this floral syndrome, to know whether these particular genes have been the target of selection in our other 3 lineages, we really need some sort of genetic mapping data, QTL mapping data, or GWAS data, and that's what we're kind of currently working on. And so I think in my last couple of minutes, I'm going to quickly tell a short vignette about one of these other hummingbird pollinated species in this clade. This is Penstemon eatonii, which has a broad distribution in the southwest. Its sister species is Penstemon laevis that has a narrow distribution right here on the Arizona Strip. And we've been sampling populations in this area of geographic overlap and resequencing genomes, and we can first look at just the genome-wide landscape of divergence between these species. So these samples were taken from this area of sympatry. And we see that there are, so this is showing the landscape of divergence between these species. So the peaks are showing regions that seem differentiated between them, and the valleys are showing regions where gene flow is apparently homogenizing genetic variation between these species. And so the first thing to ask is, what's going on at our 3 candidate genes? Are they differentiated in this species pair? So we find differentiated SNPs at our flower color gene, suggesting that the 2 species are diverged at this gene, but we don't find any SNP associations at our lipid transport protein or the apetalate-2 candidate genes, suggesting that these probably are not explaining floral differences between these species. Very unlike our first species pair, these 2 species form these spectacular hybrid zones where they co-occur. And in the past few years, we've been sampling hundreds of individuals in these canyon systems on the Arizona Strip. And within these hybrid zones, we see a ton of variation in floral traits and in hybrid ancestry. And so we're using an admixture mapping approach to map the genetic loci underlying this trait variation. We're also studying patterns of selection on these hybrids. And this has been a big collaborative project in the lab with major contributions by Josh Stevens, who just got his PhD, Hoyt Williams, a former technician, and before that, an undergrad in the lab, and then our former postdoc working with us, Luke Wheeler. So very quickly, we've looked at genetic associations for flower color. So in this case, floral hue, which was measured from reflectance spectroscopy on petal tissue. So this is the degree flowers are reddish to purple. Here we find major genetic effects on flower color at F3', 5'H. No surprise there. We probably didn't need to do this to know this. But we also see a role for another pathway gene in the anthocyanin pathway, flavonoid 3' hydroxylase, which maps to this broad low recombination region. And I'll show you one last result. So this is a GWAS for flower width. This is a polygenic trait in this hybrid zone with genetic effects found on 5 of our 8 chromosomes. And there's 2 interesting things here. So first, we find genetic associations that are very close to F3', 5'H, suggesting that there could be linked genetic variation affecting both tube width and flower color. We find no evidence of genetic effects near the AP2, apetalate-2 region. What about nectar production? Well, it turns out it's very difficult to measure nectar production in the high desert plants. We've tried really hard to do this. So right now what we're doing is trying to validate using a proxy trait, specifically the size of the nectary, and that is something that's really accessible for us to do out in the field. Okay, so to sum up really quickly, we find that there may be some genetic features that might enable rapid assembly of this hummingbird syndrome, which include alleles with relatively large effect size and perhaps an interesting role for these loss of function mutations facilitating rapid evolutionary shifts. We find that even though these are polygenic adaptations, they seem to be maintained despite gene flows, suggesting correlated selection acting on a bunch of different loci. And then finally, we actually have found no evidence for adaptive introgression fueling these repeated shifts. It seems to be mostly involving de novo mutations at this point. But there's still a lot more to learn here. So with that, happy to take any questions. Patrick.
Amazing talk. This is really fun. I have kind of a methodological question, which is about your art results, and I'm curious about whether you alternatively also try doing sliding window FSC or something, which in principle feels like it's doing the same reciprocal monophily thing, and if so, whether you saw different results, the art was highlighting regions that you wouldn't have seen if you were doing FSC?
That's a great question, and we have done sliding windows, but I don't think we've done it at narrow enough intervals to really capture this fine-grained view. But yeah, we should do that again with a smaller window size. But I think we would see a similar pattern. Yeah. Yes.
Great talk.
Thanks.
I really enjoyed that. It seems like if the loss of function precedes the evolution of the other traits, at least I'm wondering, that's kind of the question, but sort of the second follow-up. If you knock it out and then make a common garden, would you see a rapid evolution of these other traits? Do you have any guesses? I don't suspect you've done it, but
I would love to do that. That sounds like a really fun project. And I don't know what the order of trait evolution would be. I would think that nectar production would be one of the earliest traits to shift, because I don't know if hummingbirds are going to reliably visit flowers that aren't producing that large nectar reward. But it's certainly true that these loss of function mutations might mean that this can happen pretty rapidly for flower color. And the ancestral recombination graph approach, we're hoping that this might allow us to approximately date some of the ages of these alleles, but there's a lot of technical complexities to using those approaches. But yeah, so the short answer is I don't know.
All right, I think we're going to move on in the interest of time, but please grab Carrie at lunch or one of the other breaks. Next up, we have Tadashi Fukami, who comes to us as a professor from Stanford University. And he is an insightful community ecologist who asks some of the most important and fundamental questions in community ecology in some of the most interesting contexts, one of which is inside the flower. And so we're very excited to hear his work looking at community dynamics in these sweet little pools of nectar, broadening our ideas of plant-pollinator interactions to include fungi and microbes and bacteria. So thank you so much for joining us.
Thank you very much. I really appreciate the invitation to be part of this very nice symposium. And as Robin said, I wanted to invite you to think about now microorganisms in this plant rewards questions. And so what I wanted to do is to look at nectar bacteria, fungi, and how they might be related, linked to plant-pollinator interactions. And then to do that, I'm going to stick with hummingbird pollinated plants, continuing the theme that the Carrie talked about. And so in my case, I'm going to talk about one story, and that's come from California. We have, this used to be a mimulus species. It's Diplolecus orientaicus. It used to be called Diplolecus orientaicus in the 80s, and it was changed to Mimulus orientaicus, and now recently it's changed back to Diplolecus orientaicus, but it's still and so it's a hummingbird pollinated plants, and as we now know, this plant has a nectar, and inside you can see often this nectar specialist yeast and bacteria, and it doesn't have to be this particular species as long as you have nectar in flowers. Often you see these bacteria and yeast living in nectar, and what happens is that if hummingbirds come to go to old flower with lots of bacteria or yeast growing, next time the hummingbirds go to another flower to get more rewards, plant rewards, then the hummingbirds can carry from old flower these microbes, the bacteria and yeast, to get to inoculate new flower. That's initially, we know that they are microbe-free. When the new flower opens, it's kind of free of microbes. But because of the hummingbirds inoculating new flowers with bacteria and yeast, we have now a primary succession that will happen. The microbial community is developing within nectar. And this is a nice system, it turned out, because as I mentioned, it's initially it comes out as just a free micro-free system, and most of the species of bacteria and yeast that we see in flowers, they are found mostly just in nectar or pollinators, in our case hummingbirds or insects that come to flower. And so you can think of each of the flowers as almost like a habitat islands where you can study primary succession. And we have many flowers. Then it's a very well replicated system. And another thing that's really convenient from the community ecology perspective is that unlike most other microbial systems, which are really species rich, which is why microbes are very interesting. But at the same time, if you're trying to think about species interaction, understanding community ecology questions, it becomes really daunting if you have a really diverse system. In this case, unlike most other microbial systems, it's a relatively simple system because it's only a handful of fungi and bacteria that seems to have evolved to withstand a lot of challenging factors that are associated with the nectar. For example, there's a lot of sugar. That's why we're talking about sweet habitat. But you can have too much of that. And so the osmotic pressure can be really challenging. And so it seems like just a handful of species of bacteria and fungi have evolved to be able to grow despite that challenging condition. Another thing I'm going to talk about more is amino acids. As yesterday, Clay talked to us about different chemicals in nectar. One of them is amino acids in addition to sugar. So amino acids, there's the diversity of amino acids in nectar, but they can be very limiting. And so the nitrogen availability is another thing that seems to have restricted the number of species of microbes that can live in nectar. And so this is a really simple system that's kind of convenient for ecology questions. And what I wanted to do today is to I'm a PC person, so this is a little unfamiliar. You have to tap. What I wanted to do today is to have an ecological story to tell you that between yeast and bacteria, there can be what's called priority effects, where when you have yeast growing, then they will make the low amino acids low, and then that will keep the flowers as yeast dominated. And on the other hand, if you have a bacteria come into flowers, then they will become dominant, and that will, because of something related to pH, and that will become bacteria. The flower will stay bacteria dominated, and this kind of history-dependent interactions, we call it priority effects. So I'll tell you more about that. But before getting to that, I wanted to also kind of repeat the theme that we now have. Felipe talked a really nice talk about the importance of undergraduate teaching. And I have this kind of similar story. I didn't know anything about yeast. I never worked on yeast before I moved to Stanford. That was 16 years ago. So, but when I arrived, I was part of biology department, and they already had a good class for undergraduate students, lab exercise in genetics and cell biology, where they were using Baker's yeast as a model organisms to study genetics and so other kind of cell biology. And so biology department had this idea that it would be nice to have an ecology class where we use yeast. And because then the undergraduate students who've taken genetics class with the yeast would have this method familiar to them. And so it would be nice to do it in ecology as well. And there was also Jasper Ridge Biological Preserve that's close to the Stanford main campus. And so we wanted to do that class with students there. And so that was the idea. And then if you wanted to look for yeast in the field, then you have to have a lot of sugar. And it turned out that the flowers would be a good one. So we started doing this as a teaching material. But when I arrived at Stanford, one of the other things that I learned is that before going to Stanford, I was at University of Hawaii. That was my first assistant professorship position. I was struggling because teaching takes a long time. And you never think that you can be a perfect teacher because there's just so much to learn. And also, to be a good teacher, and also you want to spend a lot of time with students. And as a result, I think many of you experience that. You find very little time for research. And so when I moved to Stanford, one of the pieces of advice that was given to us, new professors during the faculty orientation, was that teaching and research doesn't have to be separated. You can combine them. And actually, when you do that, both can be actually better, synergistic. And so I had that in mind because of my struggle in my previous position. And so that's why, as we did this teaching, when I had Melinda as a PhD student at that time, and Kabir was my postdoc, we thought that it would be interesting to look at these nectar microbes that we were using for teaching. Maybe we can do research with that. We didn't have any idea about anything ecologically interesting about that. So Melinda went out to our local field site, Jasper Ridge, to do just simple sampling of nectar and then see what you can get with Sanger sequencing. And so this is an initial list of, for example, fungi from flower and then also hummingbird. And you can see that the list is not that long. And what we decided to do is to do a simple priority effect experiment where you just look to see whether if you try to assemble your community, whether the introduction order, the order in which you introduce species might change the community. And it was a little difficult to do that in the field, so we had an artificial hummingbird, which is pipette, and then do it in the lab. And so I'll skip all the details for now, but what we found with this nectar yeast species the competition experiment, so it was just the 2 species at a time, just the pairwise interactions for now. If you look at whether there is a strong priority effect where the first species will always be the winner, and then second species will lose, we see that when you have a closely related species of yeast, and if you have a distantly related species, then we don't see priority effects. So negative, strong negative, the inhibition, if you have a closely related species, we don't see that in a distance related species. That doesn't necessarily mean that the interactions are weak when you have a distant related species. We still saw a really strong inhibitory effects of first or second species, but it didn't matter which order you use if you have a distantly related species, because they are just overwhelmingly one of the species is always competitive regardless of the order. But only when you have a closely related species, presumably they are ecologically similar, then you have a very strong priority effect. So with this priority effect thing was always conceptually my overarching research questions before we started this. And so as we found these interesting things happening in nectar yeast, now this seems like a good research material in addition to teaching material. So we decided to submit an NSF proposal some time ago now, a long time ago now, and it was rejected. Eventually, we were successful in getting the support from NSF. But the first time we submitted the proposal, one of the helpful comments that we got from the reviewers was that, so it seems like a good system. They were convinced, but isn't it too simple? In flowers, if you just look at nectar yeast, we have only often no species of yeast, and often we have one species, and at most maybe 2 species. There are other fungal species too, but they are not very active. And so if you have just the one species most of the time, you can't really think of that as a community ecology study system. And so the reviewers said that why not look at bacteria, because they must be bacteria. Once you have bacteria, then it becomes more like a community, ecological community. So we had, with that comment, Marie at that time was a really good technician that we worked with, and Rachel, who is sitting there, she was, some time ago, she used to be a postdoc with me, the excellent postdoc. And so together with them, we followed the reviewers' comment to see whether bacteria, we can find bacteria in our sticky monkey flower nectar. And sure enough, we're finding, in addition to yeast, we often see bacteria. One of the genera that we find is called Neocomagatea. We also find another genus called Acinetobacter. Those two are the most common species that we are finding in our nectar. And that's about it. And so it's also simple for bacteria. But once we have bacteria, Marie was really interested in thinking about not being just a microbial ecologist, but also see whether there's an effect on plants and pollinators. And so we did a simple experiment where we inoculated new flowers with isolates of bacterial yeast and see what happens to the pollination and seed set. And monkey flowers, those of you who are mimulus researchers know that there's a stigma is open when you have a new flower, but closed when you have a pollen deposited by hummingbirds or whatever pollinators you have. And so you can actually see the flower and use stigma closure to have some idea of whether it's been pollinated. And what we found is that only when we have bacteria, Neocomagatea in our case, we have a negative effect of that inoculation compared to control. We didn't see that difference in yeast. And the same thing when we looked at the number of seeds produced from these flowers, there was a negative effect of bacteria. There were fewer number of seeds produced if you have a bacteria inoculated into nectar, but not when we didn't detect difference when we have a yeast inoculated flowers. Another thing we did some time ago was to do another simple experiment where we prepare nectar with microbial yeast and then see whether we see any hummingbird response to that. And so we have a PCR tube in it with those nectar samples in it, and we had an artificial flower experiment to look at hummingbird preference. And so what we did was just to wait for a few hours or so and then just let them come and choose, and kind of a cafeteria style experiment to see whether nectar consumption was different depending on the microbes. And we see the same thing, similar thing compared to the flowers. And so for some reason, hummingbird didn't like bacteria inoculated nectar. But no difference when you have a yeast inoculated, despite the fact that the yeast was also growing really vigorously. And so now we have this bacteria in yeast and wanted to connect them into the priority effect questions now. And so at that time, Caroline Taka was a visiting PhD student from University of Toronto. She came to Stanford for a few months, and she was interested in looking at bacteria and yeast interactions. And so this was an experiment where we had 4 species of bacteria, 4 species of microbes, 2 species of yeast, 2 species of bacteria, and all of them were isolated from our monkey flower plants from a field site at Jasper Ridge. And in this experiment, this is just another simple experiment where we just flip the order of introduction. On day 0, we could have 2 species of yeast introduced in the lab conditions, and then waited for 2 days, which is somewhat comparable to the kind of interval that you see if you look at how frequently hummingbirds are coming to flowers in the field. You can wait for 2 days and to introduce bacteria and see how abundant each of the species were. You can do just old-fashioned plating because they are distinguishable morphologically. And then you just flip the order to see what happens if you have this order now. Or we also tried simultaneous introduction. And so that was a 4-day experiment. What you can do is to get a little bit of the nectar after 4 days and then transfer to a fresh nectar to simulate the hummingbird dispersal. And so that's why we have about a month of the experiment here that Caroline did. And what we found is that if you have yeast introduced, the first one, all of the species introduced together, 2 of the 4 species were persisting, and Meshinicobrae leucophyae and then Neocomagatea, and that made sense to us because those are the most common ones that we often see in the field, and we have the other ones, Stamarilla and Asaya, but they're not that common, so that's kind of expected result. But what we found is, as you can see in the middle and the bottom plots there, if you have yeast introduced first, and then only the Meshinicobrae leucophyae was the one that was persisted, and if the bacteria was were the ones to be introduced first, it was just Neocomagatea. So already we are seeing this very strong priority effect, history-dependent outcome. And if you don't know, if you didn't know what you did in the first 2 days, what happens over this month, there's no way to tell. You really need to know what order you use to assemble this community. And we wanted to also see whether the result was the same if you have a fluctuating temperature, because in California, there's a lot of daily fluctuation in temperature. And so there's some subtle differences, but the overall picture is the same. We still have a really strong priority effect. And so with this Caroline's really what we thought was an interesting result, we realized that this is all lab experiments. We wanted to go out in the field and then just to do a more basic observation. So with Manpreet, who was also a former postdoc, she's now back in New Zealand doing a good work there as a researcher there in New Zealand. When she was with us as a postdoc, we went to different places around Stanford, about 200 kilometers along the coast, to get a lot of nectar samples and as much nectar samples as we could, as many as we could, with undergraduate students who helped us, who worked with us during one summer. And we were just doing the simple plating. And what you see here is that each dot represents a nectar sample from an individual flower. And so what you can see is that if you have a flower where you got a lot of yeast, then you don't see bacteria. If you have a lot of bacteria growing, then you don't see it. And so first time this undergraduate student made this plot, I didn't really think it was real. They are all great, but sometimes they make mistakes. But this plot turned out to be actually true. And so what you can see is that you can kind of simply try to categorize these flowers into bacteria-dominated nectar, yeast-dominated nectar, and the point is that the co-dominance seems to be rare. There are a lot of course, flowers with too few microbes to do the categorization, and then there's also a lot of differences, variation among these 12 sites that we went to. Each one has 96 flowers sampled, but across these different sites, the co-dominance is rare, and you can actually do just statistically confirm that it's either yeast or bacteria most of the time. And so this doesn't really prove that the reason behind this is priority effects. There are other potential reasons, but it's at least consistent with the idea that there's this mutual history-dependent mutual exclusion happening between bacteria and yeast. And then as we do this research, we kept this teaching this undergraduate class where we go to Jasper Ridge to do the nectar research and then nectar microbe research. And then occasionally, or often, these undergraduate students who are just taking the class for just 1 quarter, we have a quarter system rather than semester, some of them get really interested in, and they give us some ideas that we didn't think about. And one of them, some students said that why not use these pH strip, because they should be really easy. And all you have to do is probably just to put a drop of nectar and then see whether we see anything interesting. And so we thought it was interesting, and so with Rachel's involved in it, and we did the experiment where we inoculated, once again, just a bacterial yeast into the fresh nectar in the lab, and then see, using just pH strip, whether there's any difference. And you can see that there's a huge difference there, that the fresh nectar, so none, you can see the 7.5, that seems to be the nectar starts out with. And if you have yeast growing, they're growing really well. it reduces pH to 5.5, around 5.5, and if you have this Neocomagatria growing in nectar, it goes to 2.5. And later, we also confirmed that with another genus, Acinetobacter, which is also common in nectar, and they do the similar things, about 2.5. So it's a huge difference. And so we thought it was interesting, and that was just a lab experiment. And so we decided that we would go out to the field, back to the field, and then do this experiment where we just get a little flower and then put the nectar drop into this pH strip and then do as many as we can. A simple thing that even I can do. And so here we went to the two sites, Jasper Ridge, our main site, but also about 20 or 30 kilometers away, there's another site we went to, San Gregorio. And I'm going to show you the histogram here. And I'm dividing the data set into visited and non-visited using the stigma status that I mentioned. And what you can see is there seems to be different peaks. And what I wanted to do is to kind of overlay these numbers, 7.5, 5.5, 2.5, into this field observations. It's not really completely clear cut, but what I'm seeing to see here is that there is these three values seems to more or less correspond, roughly correspond to the peaks that we see. And when you have non-visited flowers, then the high peak, the 7.5, seems to be the highest peak, and so on. And then depending on whether you go to Jasper Ridge or San Gregorio, if you look at the visited flowers, in San Gregorio, there is an intermediate peak there, around 5.5, and we don't see that at Jasper Ridge, possibly because the bacteria were more common at Jasper Ridge, at least during this time, and then at San Gregorio, maybe it was the site where the yeast was growing much better. Another thing we did was to do some more experiment with simple inoculation in the field, and this is the data that Rachel produced, and what you see is that this is an amino acid measurement. And in this ordination, what this ordination is doing is to summarize all of these. So all of these arrows represent different amino acids that Rachel was able to measure. And if you have these arrows going to directions where they were more abundant in an ordination plot. And so what you can see is that if you have flowers inoculated with yeast, then all of the flowers are showing overall reduction in amino acids compared to control or bacteria inoculated flowers. And so amino acids seems to show differences in between bacteria and yeast inoculated flowers. And later, Manpreet, who was, as I mentioned, she was a postdoc, she was very interested in genomics. And so this Mescherincovia recoffea nectar yeast, this is by far the most common dominant species that we see, not just in this particular plant species we have, but across the world. And it turned out there was no genome available. And so she did the genome sequencing herself. And one of the things that she found is that there's extensive gene duplication in different amino acid transporter genes. And then PAD4, for example, is a proline permease gene. And so maybe connecting to what Claire was saying yesterday, proline seems to be one of the representative amino acids that we see in nectar. And they seems to be, the nectar yeast seems to have evolved to become, have more genes compared to baker yeast, for example. And so this made sense to us because they seems to be really good at taking up amino acids in nectar. Maybe that's adaptive if you are nectar yeast and if you want to grow very rapidly with a limited amount of amino acids. GAP is the general amino acid permease, I think. And so all of that leads to this hypothesis that we have, a simple hypothesis, to explain why we have this priority effects between bacteria and yeast. And we think that it has to do with amino acids and nectar pH. And so fresh nectar, if you measure amino acids and pH, that's where fresh nectar sits. And when we combine all of the data we collected, it seems like the yeast can grow in this region. They are fine with low amino acid availability, but not when the pH is low. On the other hand, the bacteria like Neocomagatria, Acinetobacter, they can grow even if the pH is low, but they're not as good compared to nectar yeast in growing when amino acid availability is low. And on top of that, this is the change. If you put yeast into nectar, this is the change that yeast causes because of the amino acid uptake, taking up of amino acids, and this is the change that the bacteria make. So basically, the idea is simple to understand, I think, that each of them is making nectar condition more favorable to themselves compared to others, and that's why we think that there's a really strong priority effects happening. I wanted to also briefly mention an experiment that Kelly, who just recently graduated, a PhD student, is now staying at Stanford as a postdoc in another lab. She was interested in rapid evolution because in ecology, as you know, the eco-evolutionary dynamics where rapid evolution drive population changes was becoming kind of a hot topic, and we thought that it would be interesting to also consider that in our nectar yeast. And so what she did was a simple growth to prepare different strains, evolved strains. So it was just a 6-day growth, simple growth. Every other day she was just transferring, growing the nectar yeast and transferring. And in 60 days, the reason why she did this for 60 days, which was a long time, was because that roughly corresponds to the flowering season of monkey flower, at least at the local site at Jasper Ridge. And she did this for the same thing under 3 different conditions. One, normal nectar. We had a good sugar, a high sugar, and an amino acid, and then pH is high, and simulating fresh nectar. But she also had another treatment where she did the same thing, except that this same nectar, normal nectar, was preconditioned by growing bacteria. Acinetobacter nectaris was growing in nectar, and we filtered out this bacteria and used that for yeast growth. We also had a low pH nectar as a third factor. And the motivation behind this was this getting back to this site variation. If you go to one site, you have a lot of yeast growing. Another site, you might have more bacteria growing. So we wanted to simulate these different sites where the yeast might be more frequently confronted by some challenging condition made by bacteria in one site, some sites, but no other, where the yeast was dominating. And so after resequencing, so that's why we know that they have evolved. When we look at the genome resequencing data that Kelly had, together with Manpreet, we see some genetic changes after 60 days. And we just wanted to see whether there's any changes in growth. And so what I'm showing you here is that the original is the one where we have an original strain that we used as an ancestral strain, isolate. And then normal nectar, what I'm showing you there is that these are the strains that grew in normal nectar for 60 days. And we were saving them in the freezer. And then when we wanted to do this monoculture growth experiment, we revived them and then just see whether they grow as well as the original. And they were. But the strains, the isolates that were growing under low pH nectar was a little bit slow in growth and so on. And we now compare this to what happened if you put them together with bacteria, Acinetobacter. So it was as expected. We saw reduced growth rate compared to the monoculture growth rate. And that was, we think it was because of this pH thing, the priority effect. But what was interesting to us is that when we look at the difference between the monoculture and the mixed culture growth, that difference was smaller if you have strains that evolved in low pH and bacteria conditioned nectar. So one interpretation, possible interpretation of this result is that after 6 days, which is not long for us humans, but for microbes that can be a long time, many generations, that seems long enough for them to evolve to maybe better at resisting priority effect against bacteria and compared to the normal and then original strains. So because of that, we wanted to go and then do more genetic study on genetic variation. So Kelly and Manpreet did more resequencing of different strains, about 100 strains isolated from different places across these sites that we went to. And we see high genetic variation within this nectar yeast, even though it's a species poor system. And when you look at intraspecific variation, there's a lot of genetic variation. And what we did, using these strains, isolates that we we had from the field, we did some more growth experiments in the lab. And what we found is this pattern where the strain, so each dot is a strain isolate. And there seems to be a trade-off where some strains seems to be very good at growing if under good conditions, but they are not good at growing when they are being challenged by harsh conditions. And then other strains seems to be better at growing when they are in a difficult time, but they are not when they have a good condition. So this trade-off. Now I'm just speculating, but this kind of trade-off among these strains might be one reason why the genetic diversity is maintained across different in this landscape. I'll try to show you a few more results, and I'll be done in a few minutes. But here, getting back to this pH amino acid question, we wanted to do some more, with the undergraduate students taking this class, we wanted to do more experiments and then just to see whether we can actually tease apart the effect of amino acids and pH. And it turned out that the reason why the hummingbird didn't seem to like bacteria inoculated nectar seems to be about the pH. So if you look at this, pH seems to have an effect on the nectar consumption by hummingbirds, but amino acid didn't seem to have an effect. One more thing that I wanted to mention is that in addition to inoculation experiment with a single flower level, one time we decided that we go to the field, back to the field, and inoculate all of the flowers in a given plant. And so we went to one plant and then did the yeast inoculation in all of the flowers that were on that plant. And we went to another plant and we inoculated those flowers with bacteria. And we had water as a control. And we waited for 3 weeks and went back to these plants and sampled nectar from these plants. And when you wait for 3 weeks, it's roughly 3 floral generations away, because each flower lasts roughly a week. And so if you wait for 3 weeks, you're not, and when you're collecting nectar from these flowers, you're not collecting nectar from the flowers that you did the inoculation. But despite that, when we do the metabarcoding and look at the bacterial species, species composition using this kind of a PICO-A, one of the usual ordination that we use to characterize communities, we still saw differences between bacteria and yeast inoculated plants. And so what this shows is that there might be effect of priority effect, not just at the single flower scale, but also whole plant scale. So Lucas Nell, who was a postdoc, and he's now moved on to Cornell to do his second postdoc, he did a modeling to see what happens if you have this kind of situation. And what he found with this modeling is that yeast and bacteria can exclude each other at the plant scale, but when you have a different multiple plants in a landscape, they can coexist because one plant can be yeast dominated, another one bacteria dominated. But the necessary condition was you need to assume in the model that the hummingbirds show this preference for yeast. And so what I did here to kind of wrap up is that we have this priority effects, and with this, the microbial communities are not just living there, and they can have an effect on plant pollinator mutualism. We knew from the beginning that there's a positive effect of flowers and hummingbirds, the pollinators, into the microbes, because flowers provide a habitat to the microbes, and then hummingbirds provide dispersal. So that positive effect was, from the beginning, it was more or less clear. But what was interesting to us is that there can be also the feedback. And so if you have a bacteria, there can be a negative effect on plant pollinator mutualism. And if you have a negative effect, even if yeast might not have a direct effect, they can have an indirect effect, because yeast can protect nectar against bacterial degradation because of this priority effect. And then this can scale up to affect what we might call meta-community collection of flowers on a plant because of the hummingbird dispersal. And then that can feed back to local community. And also we see genetic variation across this region. And because each strains may seem to have a different priority effect strengths with bacteria, that can affect what happens in local community. And they might have also a feedback into region. And so what we are trying to do is to understand more about this multiple spatial scale phenomena where the priority effect that happens within a flower, how that can scale up and how there may be a feedback between small scale and large scale. So I'll stop there and thank you very much for your attention.
Just come back to the alcohol since we were talking about yesterday. I was just wondering, like, could that be another positive effect for the pollination? Because when they come in will get drunk, they got slide.
Yes. Yeah, thank you for that question. We try to measure alcohol as thank you. So the question was, is the alcohol, ethanol, does that have any effect on as part of the microbial effect on the plant pollinator interactions? And that's a good question. And we try to measure ethanol, alcohol content, and how that might be affected by yeast in nectar. And so far, we couldn't detect any. But so we kind of gave up on that idea. But then last week I was visiting UC Berkeley, and then some of the people there, they were measuring ethanol in, and then they were able to measure. And then, as expected, yeast seems to increase the ethanol concentration in nectar. And they did a little bit of calculation to see whether, even though the concentration may be small, if you look at the total amount of alcohol that hummingbirds can get when they were always going to this nectar to get nectar, it turned out, I think, that they were getting more alcohol than maybe average human. And so there might be an effect, but we don't know. We haven't really looked into the effect on hummingbird behavior yet, but we need to look at that. Yeah.
Okay, last up before lunch. It is my great pleasure to introduce Avery Russell, who I understand is a newly minted tenured professor now at Missouri State. I'm sorry. Woo! So that's awesome. Big congrats on that. We are excited that you are here. So Avery is a creative and punny behavioral ecologist, if anybody knows him. He is always quite witty. And his work on bees, and particularly buzz pollination, has really shaken the way we think about, sorry, I had to, thank you. The way we think about animal behavior in response to getting rewards from plants. And so I'm really excited to hear some of your work.
Thank you, appreciate that. Should I put this mic on as well?
Yes. Yeah, sorry.
All right, it's a pleasure to be here, and thank you so much for inviting me as well. I really appreciate the support that Harvard has given, especially at a difficult time. I don't know if this is on. Is this on? Okay, sweet. All right, so yes, I'm going to be talking about not birds. I'm not going to be talking about nectar. I'm going to be talking about pollen today. And will that still play? Fantastic. Already got video challenges. So I'm going to be presenting some work here on, basically that's a culmination of years of work across multiple labs and collaborations, lots of co-authors who you see cited at the end in the relevant section. So I hope you're super excited, because I am too, to present this. And I guess I don't have any working videos. Oh wait, there goes, yay, excellent. So if you'd ever like to collaborate on a grant to bring you to Missouri to do a postdoc, or you want to work on a master's with me, or you've got some undergrads that are wanting to collaborate, please contact us. I'm always happy to collaborate. All the work here that I'm going to present is collaborative work, and that's been the basis of my career. So super excited to do that kind of thing, although you may have to endure severe punishment. So with that, let's carpe diem and get to the science. So foraging behavior is frequently implicated in driving evolutionary patterns in both animals and their biotic resources. For instance, herbivory often drives the evolution of plant defenses, and such as shell thickness in acorns and the number and size of thorns in acacia. In turn, this often results in reciprocal morphological evolution of the herbivores, such as increased snout length of the acorn weevils to drill into the acorn and thickened lips of things like giraffes that offer protection from acacia thorns. And in fact, the ecological and evolutionary consequences of animal foraging have been particularly well studied in pollination mutualisms, of course. For example, pollinators are thought to drive the evolution of nectar spur length, as we talked about earlier, mechanical levers that limit access to floral nutrients, petal surfaces that allow pollinators to hold on, and of course, other things like nectaries. And altogether, in turn, these floral traits are thought to drive the reciprocal evolution of pollinator morphology. So things like tongue length, body size, the shape of hairs that hold on to pollen, for instance, are all reflect adaptations that enhance collection of floral nutrients by pollinators. However, despite over a century of attention to reciprocal evolution of plant and pollinator morphology, we lack a really clear understanding of whether floral traits can drive the evolution of pollinator behavior and the distribution of pollinator taxa, and also how behavior can directly drive floral trait evolution. So I'm going to talk a little bit more about that today. So to help answer this question, we're going to examine the ecological and evolutionary consequences of an incredibly widespread plant-bee mutualism, which is called buzz pollination. And this is all just an avenue to show you cool videos. So when you have Disney photographers in the lab, you want to show videos, and I don't know why this one isn't playing either, unfortunately. I guess this is the Mac to PC transition. So buzz pollination is thought to be an ancient mutualism between bees capable of buzzing flowers to extract pollen, which are their primary source of protein, and flowers with specialized tube-like porocidal morphology that requires buzzing for their pollen to be removed. And if this video were playing, you would see a bee vibrating the anthers and pollen just absolutely dosing the abdomen here. If you want to see those videos later on, I got them all on my laptop, so you're welcome to them. And we call this a totally tubular mutualism. Okay. So flowers with porocidal morphology are thought to nearly be exclusively pollinated by buzzing bees, which use their indirect flight muscles to vibrate the pollen out of the flowers. And again, I assume you're here for the video, so hopefully this one will play and will leave you buzzing with excitement. Not going to play for me. All right, so what you would see would be a bee vibrating the anthers here, and then pollen shooting out onto its abdomen. I guess I can try. Oh, yes. Aha. Ah, boo. Okay. Well, there'd be lots of high-speed buzzing. You'd hear it, and I could imitate that for you later on if you'd like, especially after a couple drinks. And it turns out that the ability to buzz flowers to extract their pollen is really quite widespread in bees. So using the taxonomic list from Discover Life and a comprehensive literature search, we found that bees that buzz flowers are found in all 7 bee families and about 17% of bee genera. And so we can estimate overall that upwards of 57% of bee species are capable of buzzing to extract pollen based on those numbers. So we might ask, what are the evolutionary implications of this behavior for bees then? So here we see a time and fossil calibrated bee phylogeny in which we constrained individual genera to be buzzing or non-buzzing. And I want to emphasize this is really preliminary work. We don't have a lot of records of bee buzzing in the world. There are about 500, 600 species that have been recorded to buzz out of the 21,000 species. So that's why we make estimates at the genus level rather than at a species level. So if you want to do that work, get to listening outside, do some recordings. Especially in the global south, we have almost no records. So this phylogeny represents results of a maximum clade credibility tree with an unconstrained root and a Bayesian uncorrelated relaxed clock model. And that allows us to estimate the ages of lineages. And here we find that buzzing behavior is truly ancient. At the base of the bees appears to have evolved in the early Cretaceous. So presumably the ancestors of the bees also possess the capability of buzzing flowers for pollen. Really weird to us, we didn't expect that. And this buzzing behavior is quite evolutionarily labile. It's been gained around 45 times and lost a further 66 times across the bees. And again, this is at the genus level, so who knows what the lability is at the level of within genera. And finally, branching out, we found that buzzing bees, or buzzing behavior, may be a key driver of bee diversification. So in the clades indicated with the yellow stars, buzzing is significantly associated with greater diversification. However, there's a lot more work that needs to be done to document the occurrence of buzzing, especially in the global south, as I mentioned, in order to determine whether the association of buzzing and increased diversification is widespread across the bees. We basically have no information across most of the bee groups. Okay, so given that buzzing behavior directly affects access to pollen, which is an essential food for bees from a broad range of plant species, buzzing behavior might also influence bee biogeography. So we therefore examined how the biogeography of buzzing and non-buzzing species differed, and to accomplish this, we used the Discover Life Bee World Checklist to link buzzing and non-buzzing bee species to administrative areas. And then we use state province level information for most areas via the Global Administration Areas database and generated about 136,000 links between bee groups and where they're found. And in this figure, you see the richness of all bees, so buzzing and non-buzzing bees, across all areas of the world, where redder areas are richer and bluer areas are less rich. So I want you to dedicate this to memory because the next slide is going to show you how this differs compared to buzzing lineages. So here we see that patterns of buzzing species depart dramatically from the overall pattern of bee richness. So overall, a greater proportion of buzzing bee species are found in Eastern and Central America, as well as in Europe, the Middle East, South Africa, and Northeast Asia, and parts of South America. And these are the regions marked in redder areas. Conversely, there's a reduced proportion of buzzing bee species found in parts of the Southern and Southeast Asia and most of Australia and Central Africa, and these are the regions marked in bluer tones. Okay, so this is our global perspective. So let's take a little bit closer look at each of the individual bee families. So these patterns actually differ quite substantially among bee families, and here I'm going to show you results for 4 of the 7 bee families in the interest of time, and also because some of the bee families have so few species, it's impossible to actually get good geographic mapping. So here again, redder areas are going to indicate increased proportion of buzzing, and bluer is going to indicate decreased proportion of buzzing species. So in the Andrenids, the patterns in North America and Europe are similar, although there's a greater proportion of buzzing bee species found in South America, whereas a much smaller proportion are found in East Asia or North Africa. In Coletidae, the patterns are quite similar between buzzing and non-buzzing species, except in Europe, where there's a greater proportion of bees that buzz. And then conversely, in Apidae, which of course have your honeybees and your bumblebees and really diverse lineages, there are patterns that are overall similar in most regions, but a greater proportion of species buzz in Northeast Asia and fewer species buzz in parts of Southern and Southeast Asia. And then finally, Halictidae, which are your metallic and green bees or sweat bees. These also show similar patterns, although fewer species buzz in Western South America and Southeast Asia. So patterns differ quite a bit across different regions for each family. Okay. So given that there are large differences in the biogeography of the buzzing and non-buzzing bees, we wanted to chart the ecological drivers of these differences. So we consider 53 different variables here as potential drivers of bee diversity and buzzing, which you can see here. These are going to be on the test, so please start memorizing. I teach a lot, so you know. And these variables include different elements of climate, which are highlighted in blue here, as climate and its facets are likely primary drivers of buzzing. Think of this thing as a behavior that heats up bees and will cook them if they are getting a little too hot, and maybe it influences how plants are distributed as well. So we have to also look at impacts on plants, and so we included plant richness overall as well as poricidal plant richness specifically as potential drivers. These are those plants that are thought to be associated primarily with buzzing bees, and they're highlighted in green. So to analyze the effects of potential drivers, we ran regressions with each variable and then used these to make a best model by using initially the variables with the strongest relationships and then sequentially removing variables that were not significant in the model until we had all the variables that were significant. And examining these same 4 bee families again, we find that the top driver of percentage of buzzing species for a given region of 3 of the 4 families is, can I get a drum roll? A little drum roll, excellent. Or a buzz roll, I don't know, is poricidal plant richness. So furthermore, poricidal plant richness did not have a major influence on overall bee richness, highlighting that this driver is uniquely associated with buzzing lineages. And according to this, or adding to this, other lesser drivers like temperature and moisture and latitude also differed for the non-buzzers versus the buzzing lineages. So pretty significant differences across the board. And at last, but not least, if you squint your eyes, you can see that the hotspots of buzzing bee and poricidal plant species biodiversity largely overlap, though there are some pretty significant departures, and I'll explain that in a moment. For instance, many poricidal plant species occur where buzzing bee species have yet to be announced or discovered or recorded. So given that buzzing bees are closely associated with pollination of poricidal plant species, this suggests that there's a lot of work left to be done to document buzzing bee biogeography. Specifically, you notice in Brazil. So we need a lot more scientists doing this kind of work, more funding as well. Okay, so far we've focused on the bee side of the story, but we've neglected to examine the evolutionary history of the poricidal plant species that are thought to require pollination by buzzing bees. And I wish that video had played because it is literally a pollen waterfall coming out of this exocum flower. Again, I'm happy to show you videos later on. So flowers that conceal their pollen using poricidal morphology are very taxonomically widespread. So using taxonomic lists such as the 2013 plant list and a comprehensive literature review, we find that poricidal taxa comprise 22% of flowering plant families, 5% of plant genera, and then of course we can estimate from that that it's around 10% of flowering plant species. These are plant species that don't offer nectar, sorry y'all, but they offer primarily pollen as a reward. So here we've illustrated the diverse ways to build a poricidal flower. False colors indicate the structure or the tissue involved, with the corolla indicated in purple, filament indicated in orange, and anther indicated in blue. And you can see that it is fairly diverse. Most of them are doing this with their anthers, but there are also those that do it with their corolla or staminodes and so on. So I would say that poricidal floral morphology in general is wildly diverse. Okay, maybe not that wild. Hopefully you appreciate my Apollon sense of humor by now. Okay, so using a hidden character state dependent speciation and extinction model, a HISI model, we find that poricidal morphology is incredibly evolutionarily labile. There are at least 205 independent gains and over 215 independent losses across the angiosperms. And we did these at a species level analysis, but again, there's very little documentation at the species level, so we coded genera as all poricidal or all non-poricidal for this region, and there's usually monotypic genera, so most genera are monotypic for poricidal or non-poricidal. So you can assume that this is a really conservative estimate for this reason, and much more work needs to be done. Okay, so poricidal flowers appear to have evolved around 150 million years ago, which is also really close to the origin of the earliest hypothesized buzz pollination or buzz pollinating bees. Remember, they evolved around 130 million years ago or so. So lineages that evolved poricidal flowers then appear to remain in this state with little change to the present. So once they evolve poricidality, they don't really change back to non-poricidality. Interestingly, we also find that net diversification rate of poricidal genera is often, but not always, lower than non-poricidal taxa. And this might suggest that specialization via poricidal anthers is a dead end, but we find a different answer, or anther. So when we examine transition rates, we find that transition back to the non-poricidal state occur much more rapidly than to the poricidal state, suggesting that it's possible for flowers to re-evolve non-poricidal floral morphology relatively easily. Okay, so pulling all of this together, yeah, I know. I've shown you that buzzing is an ancient pollen collection behavior that has evolved numerous times. I've given you evidence that the biogeography of buzzing bees differs from non-buzzing species, and that the top driver of buzzing bee prevalence, and the proportion of buzzing bees in a given area, is poricidal species richness. And likewise, we've demonstrated that porous phytomorphology is incredibly prevalent and can result in reduced diversification, but plants can still escape this specialization. So with that, I'll thank all my co-authors and all the advice I've received, all the collaborations, a bit of our funding, and I want to mention we always work on native traditional land at Kikipoisio and Osage. We're always working to give land back in areas or money when we can. So with that, I'd like to thank you for your time, for all the stuff that's kept pedal to the metal, and with that, I might have some answers for you.
You guys were warned. You were warned. Okay, we'll take 1 or 2 questions and then break for lunch.
Sure.
This was super fun. Thank you. Something you mentioned a couple times was sort of a lack of resolution below the genus level and sort of understanding of whether bees are buzzing or not. But for the porcidal plants, you mentioned that they tend to be maybe monotypic.
Yeah, yeah.
Level. I'm wondering if you have a sense for that of like genera that are well sampled in bees. Are they typically monotypic? And if so, have you tried to like, I'm thinking about some of these like kind of undersampled like geographic regions and wondering if you can kind of impute like candidate buzzing bees or something in these regions.
Yeah. Yeah, great question. I feel like I'm not well suited to some of these answers here. We know that there are a lot of bee genera that are not monotypic. So like even within, so for instance, in Apidae, we know Bombus, monotypic. Every bumblebee except for the kleptoparasites buzzes. But if you were to look at, for instance, Halicta or Andrena, we know that there's some bees that have never been reported to buzz flowers. And whether that's because nobody's ever looked or that's because they are truly not buzzing is up as an open question. Megachile, for instance, your leafcutter bees, they are definitely diverse in whether they buzz or don't buzz. Again, very few people have really looked at this, and also the phylogeny for bees is a total mess. So plants are great. They have amazing phylogenies. You can get resolution down to the species. For bees, it's like just a giant cluster. So, yeah. Thank you.
I have 2 sort of related questions here. One of which is how much do bees get better at this with experience? Do you have a sense of that?
Yes.
And my second question is, I see what's in it for the plants, but what's in it for the bees? Is it just more pollen than these bees could get going to other types of flowers?
Yeah, that's a great question.
This clearly works.
Right. Yeah, yeah. So I'll talk about the experience thing. So most of my work is actually cognition, learning, and memory stuff with bees. They can't learn this behavior. This is innate. It's something they do on their very first visit within the very first milliseconds of visiting a flower, and they don't get better at buzzing. So they can't modify the frequency of their buzz. They can't modify other aspects of their buzz. They can modify how they handle the flower. So it's just like a toolkit. It's like their little Swiss Army knife that they can stick in there and use. The second part of that question was...
What's in it for them? Do these buzz-pollinated flowers make much more pollen than plants are?
Yeah, you hit it on the head. So these flowers produce on the order of 2 milligrams of pollen compared to, like, if you think about Mimulus, they're producing like 0.2 milligrams of pollen. So it's an order of magnitude or sometimes multiple orders of magnitude more pollen. It's often considered to be more protein-rich pollen, and there might be other attributes that we really don't know a lot about. There have been folks that have tried to explore this for the last like 20 years, but very little experimental work. So yeah, super cool. And of course, you can imagine that is a resource that no other pollinator can get access to. If you're a bee and you're foraging on flowers, this is the pollen source that nobody else is going to access.
All right, let's do one more quick thank you.
I think he said Vinette.
Vinette, okay.
Vinette.
Yes.
That's what I said, yeah.
Perfect.
I've been there. Yes, I was just gonna
check off.
was fun, of course. No complaints. Or whatever. You don't have to.
Okay, so Ned Friedman is going to give us a little bit of an overview of the wonderful facility we're at today at the Arnold Arboretum, and then we will get started with our next scientific talk at 2 o'clock.
Do I need to wear a microphone or not?
You can if you want
no,
to.
I mean for the video.
Yeah, for the video, yes, yes. It sounds like.
I assume it's this one, I don't know. Okay. All right, well, welcome everyone. For those of you who just went on a walk, you've seen the Arnold Arboretum. For those of you who didn't, I hope to convince you should see the Arnold Arboretum. So what I want to talk about today is just a really quick overview of the Arnold Arboretum. And to begin with, we are 281 acres, so we're a mile long, a half mile wide. There are roughly 16,000 curated wood plant accessions, roughly 2,100 species, incredibly biodiverse. We have an herbarium of 1.5 billion specimens with our great strength in Asia. A mile from here are archives and library, all kinds of important images in the early 20th century. Our collecting expeditions, there are about 70 people from the Arnold Arboretum of Harvard University that work here. But here's where we start. We got to start at the beginning with the origin story. The land came to Harvard in 1842 when a very wealthy person who went to Harvard College died, and his name was Benjamin Bussey. He wanted to establish an agricultural institution, which he did, and the land came with it. It sat for 30 years until a whaling merchant in New Bedford, Massachusetts, who wanted to build an arboretum, died, and he didn't go to Harvard, but trustees did, and they knew this place. And they partnered the money from James Arnold, hence Arnold Arboretum, to the land and created the Arnold Arboretum in 1872, first public arboretum in the New World. First director was appointed in 1873. I'm the 8th director. It does not turn over very often. Now, the first director was really interested not only in having a great scientific collection, he wanted an extraordinarily beautiful landscape and a public space. And actually, he turned to Frederick Law Olmsted, who designed Central Park, but this is 20 years later, and he's much better than he was when he did Central Park. And they did something amazing. They convinced Harvard to give the land for free to the city of Boston so it would be incorporated into the park system. And then we would lease it back. And the lease we have, the indenture with the city of Boston is for 1,000 years. It's renewable once, and we send a check downtown for $1 every year. We control the property. There are no city employees here. There's no city funding. But this is really essentially forever. It's a 2010-year institution. So we take care of everything, we grow everything, we collect everything. But Harvard, the city of Boston spent quite a lot of time building the carriage roads, the walls, the gates, everything was on their dime. And that took almost 20 years. So I say that we're actually a co-production of Harvard University and the city of Boston. And for those of you, I'm a professor of organismic and evolutionary biology, like Robin and Elena and others here, and I run a city park. And I actually have half my time is spent dealing with city. It's a very complicated place. But this is the thing I want you to remember. That's the minimum run of show is till the year 3882. So who are we? Well, we're one of Frederick Law Olmsted's greatest designs. We're a cherished public park in Boston's Emerald Necklace. If we do our horticulture right, children will do cartwheels, right? We're home and host to the vibrant human diversity of Boston because we are free and open to all every day. That makes us actually an agent of democracy. That means we are bringing people out of their bubbles to make eye contact, to say good morning, to interact. This is incredibly important in the day and age that we live in. We are, of course, Harvard's Museum of Trees. These are living objects that are meant to be studied. And about right now, there are probably 2,000 or 3,000 different organisms that are being studied by someone from around the world or Harvard. We're clearly, I opened up this research building, it was empty, there was a single chair in here. I hired my first person, Faye Ruzine, who runs the research operation here, and we're now full, as you'll see. And we have incredible equipment, growth facilities, greenhouses, you name it, microscopy, obviously genomics. I want to let you know, that we have a number of endowed funds that are for research, particularly for young scholars, and they are not for Harvard people. These are for anyone in the world. If you can think of something to do in this collection, you should be going online. These are up to $25,000 kinds of grants. So make sure you get to our grants page. And as I mentioned, I'm one of 3 faculty based here, along with Robin Hopkins and along with Ben Taylor. So we all 3 have our labs here and our research groups here. We are a collection of trees that are working to help us understand the effects of global change. There's quite a bit of work in this area. We're also an urban biodiversity hotspot. There was a college thesis that documented over 70 native species of bees. We're working with Tufts University with acoustic monitors to document our bats. And we have microbiome work going on here. You can think of it, we can get it done. We have the largest solar panel system at Harvard, and out there is the first native plants meadow in the Commonwealth of Massachusetts. There are over 50 species of herbaceous material under those panels, all collected locally, all to promote insect health. It's not about the plants. We are friends and colleagues with the peoples and the cultures of the world. We've been circling the globe collecting for a very long time. There's one of our 3-year expeditions to China in the early 20th century. Here I am welcoming my Chinese colleagues on an expedition we did in the Appalachians to bring North American material to China. I am right now going on my way in a few weeks to Korea. We're about to sign some new agreements. I've been working 8 years on it to collect in Korea, but it's a wonderful set of relationships. We don't go and grab plants. We go into the field with our colleagues who are in place, and we build friendships. And this has made us a group of intrepid plant explorers. You can see by the size of the dot, we mostly go to China because that's where the most species are of temperate woody plants. But Japan, Korea, obviously southeastern US. Occasionally we drop over to Europe. There's not so much stuff there. We're home to 16,000 organisms with provenance. We know where every plant came from. This dawn redwood was collected in 1947. It was received as a seed in 1948. Collected in the wild in China. We know whose hands it passed through. That's provenance. That's giving an organism standing. That is really important to our culture here at the Arnold Arboretum. We're a safe harbor for species that are extinct in the wild. By the way, that's Franklinia. We're a force for conservation and fighting extinction around the world. We're not local. We go to China to work on paperbark maples, and we bring the germplasm and get it into gardens, so it's ex situ conservation. It's a very big priority for us. And I spent a few years after I got here, we weren't actually collecting regularly when we got here, and we decided we would really map out what we need to support our research mission and our conservation mission, as well as our aesthetic mission in horticulture. And we made a list of 400 things, taxa, that we wanted, that we thought we could improve the collections with. And we put that down on paper, and just to give you an idea of how we changed the collections, after we launched this campaign, this is the Plants of Conservation Concern, 2017, 3.3% of the taxa, and almost 8% of the plants. It's doubled since we really got serious about conservation. And it will double again in the next 6 or 7 years. This is where we park, and we don't put it in a seed vault, we grow it so you can come and study it. That's what we do. And actually, as you walk around, of the 16,000 dots, those 2,700 up there are all members of a species that's threatened. So in 2022, we hit our 150th birthday, and I sat down and asked, again, 2010-year institution, where are we going for the next 150? And I decided that we would really go back to our founding public promise of what's now called environmental justice. Olmsted would have said something like the mixing of the classes and democracy. It's environmental justice is what we call it now. We do it, but are we doing it well enough? And the answer is no. And we're going to double down on it. And of course, through the sciences and through our conservation work, we were going to get really doubling down on fighting global change and extinction. Just going to talk about environmental justice for the last few minutes because it's pretty much what I'm doing full time. So what's our plan of action? I told you the city owns the hardscape. They built it. Those gates you walk through are city property. That's how an environmental justice neighborhood with public housing comes to the Arnold Arboretum, meaning they don't. There's drug use, there's garbage. This is how cities really look when you start to interrogate access and equity. And this is a really, really crushing thing for me. It took me 10 years because they had to build this stuff to get the city, and this is what it looks like as of 2 weeks ago it opened. All right? This is how we provide access. It has to be respectful, clean, and safe. And in fact, we have a grant. I have a grant from the school, with the people in the school of the Kennedy School of Government, the School of Public Health, and we're actually working with experts in equity and green space equity to get to know the people in public housing, to build relationships, not just open a portal. Build relationships and do longitudinal surveys so that we can see the effects of having equal access to green space. So it's a really big thing. This was my starting point for what I would call radical accessibility. We have 14 entrances. We are completely porous. Now, none of them are as bad as what I just showed you, but all of them are pretty shabby. Many of the gates are welded shut because the city didn't fix them. So you have iron bars with no signs. How does that read to someone who doesn't have privilege or access? And we actually had a design competition. We're working with Michael Van Valkenburg Associates. They did the Obama Center. They're one of the premier design firms in the world. And we are going to rebuild and take over the city's entrances because we know they won't do it. So we started with those that are closest to the environmental justice neighborhood. So you're looking at an entrance with bars, welded shut, asphalt, and actually it's going to open in a month. The plantings are going in. The gates will be rehung and open. There'll be plants tumbling out. It will be done. Every single part of that thing was taken apart, numbered, rebuilt, and it's done. That's about a $2.3 million project, all raised from philanthropy. The next entrance is across from the subway station, which is right around the corner. That's about getting people from across the city, 17 bus lines and a subway. Well, there's no way anyone at that station would know that they're staring at the Arnold Arboretum. But we've just started construction on this entrance. This is about a $2.7 million project. All the money is in hand. And by the end of the fall, we will have a new entrance, and it will be beautiful. So I just want to ask a simple question. So what is the Arnold Arboretum? Well, of course, we're here in our scientific capacity. You get outside. It's actually public health that we're serving up. We're making you live longer just by being here. You will certainly feel better. It's a really special place. It's a place of immense beauty. It's a place where dove trees, now in flower, can lift our souls, Davidia from China. It's a place where friendships thrive. It's a place where families thrive. It's a place where love thrives. So I just want to let you know, you're in a world that is filled with hatred. This is a space that is defined by love. Thanks. We have time for a couple of questions, or none. Yes?
Thank you. This is wonderful, and it's great to see the work that you're doing and the way that you're still thinking about access and accessibility. This is a downer question. I'm just curious, when you think about, like you talked about all the collections that you want, all the things you want to collect and bring here, how do you think about mitigating the potential that something's going to go invasive? Like you know, there's so much invasive species that come from people moving things around. Like,
So, yeah, 100 years ago, we were introducing invasives. No one was thinking about it. So we actually have a list of things we can say, yay, we introduced this. But we're very careful. We do know about things that have invasive tendencies. If they're dioecious, we can grow one sex. We can keep them away from our perimeters. So we have a variety of policies, and we don't release anything that we think could have those tendencies. But, you know, we grow honeysuckle. We've been growing it. It's already too late for most of the things. So, but we are hyper aware of that. That's for sure. USDA would not let those things in, most likely either.
okay.
Yeah.
Once you plant something, you sort of monitor it, and if it's getting out of control,
Yeah.
you don't have mind for that.
Yeah. Well, I mean, like calopanis is something that we shipped off for research in New Hampshire. And guess what? It started running, so they have to be a little bit more careful. Yeah.
I really respect this place's desire to bring resources here and to preserve them. With so many botanical gardens and collections closing around the U.S., has there been thought to taking some of the resources at Harvard and keep them going?
It wouldn't be legal. This runs on, people give money to the Arnold Arboretum. It says it has to be used for the Arnold Arboretum. Which is really great. We run on endowment here. Pay out a year $53 billion. We have a chunk of that. But it's all legally required to be spent in service of an Arnold Arboretum. So, yeah, okay.
Thank you, Ned.
You bet.
Okay, hope everyone had an excellent lunch and enjoyed all of the fabulous posters. You should feel free to keep those up until the end of the day so that we can continue walking around. For this next session, we're going to continue somewhat on our theme of ecology and evolution. We have 3 talks by some really amazing ecologists and evolutionary biologists. But this time we're going to dive a little bit more into microbial interactions. Up first, we have Rachel Vanette, who is a professor at the University of California, Davis. Her work uses lots of diverse techniques to understand how microorganisms affect really important processes like pollination and herbivory. And today we're going to hear a talk titled Home Sweet Home, Microbial Growth in Flowers is Shaped by Nectar Chemistry.
Thank you. I will also add arboretums are places where science thrives. And in this talk, I will basically be highlighting some work that we've done in the Arboretum at UC Davis. So arboretums are amazing resources. First of all, before I dive in there, I'd like to, again, thank the organizers for the invitation to participate in this symposium. It's been amazing, and I've really enjoyed the diversity of talks and look forward to catching up with many of you and hearing your thoughts on our work. Before I dive into the science, I'd like to acknowledge my collaborators on this particular project. So the work I'm going to talk about today has been about 2 years in the making. So it's not necessarily what everybody in my lab works on. I'll take a short detour later on to tell you what most folks in my lab work on. But we're very interested in sort of nectar processes. And here with this particular talk, I'd like to acknowledge the chemist behind all of the data that I'll present, Caitlin Rearing. She's a research chemist at USDA. Jake Sakala was a postdoc in my lab who helped with a lot of the field work, and Lita Landucci was a postbac in my lab funded by NSF and is now a PhD student in Minnesota. And I'd also like to acknowledge our collaborator, Rachel Davis at the UC Davis Arboretum. She's a horticulturalist and sort of essential to getting a lot of the plants that we use and facilitating our work there. Okay. So with that, I'll show you my flower pictures. So as we learned this morning, or in the recent publication, about 75% of animal pollinated plant species produce nectar. And that could be in various flower shapes or sizes or modes of attracting pollinators. And we know that floral nectar is primarily thought to be an attractant for these pollinators that visit plants and disperse pollen from one plant individual to the next. But there may be a hidden side of floral nectar, right? So floral nectar may also be an Achilles heel, or a place where plants might suffer negative effects by plant pathogens. For example, here's a blighted tree limb caused by Erwinia amylivora, a bacterial pathogen of many plant species. Nectar can also be colonized by yeasts that metabolize the sugars and potentially detract from the sugar or the caloric amount, the carbohydrates available for pollinators. And flowers can also be colonized by pollinator pathogens. These pollinator pathogens, either fungi or bacteria, or other organisms that we'll hear about later today, I'm sure, can colonize flowers and replicate in nectar as well. So is floral nectar all good, or what's happening there? So there have been a few hypotheses that nectar chemistry should potentially be multifunctional, right? Where its primary function should be attracting pollinators, but it should also have some chemical properties that reduce microbial growth, potentially. And so in this study, we were very curious to try and understand, are there chemical strategies to maintain nectar quality in the face of microbial introduction? And we had some ideas for target chemistries. So previous work, this is led or written up by Evan Palmer Young, had shown that flowers, including nectar, often produce high abundances or contain high abundances of secondary metabolites that we might hypothesize to potentially have some antimicrobial function. And here within nectar, we see it likely contains alkaloids, amino acids, flavonoids, terpenoids, and this varies a lot depending on the plant species that you sample, right? And so we had this hypothesis that maybe variation in some of these compounds could affect the microbial growth in floral nectar. However, at the same time, I'm going to draw on a lot of Lynn's work here, there may be costs to defending your nectar. So in this case, there may be an ecological cost. So in this nice study, they sampled different species of Nicotiana with various pollinator reliance and found that those plants that rely on pollinators for outcrossing, or that were obligate outcrossers, had lower levels of alkaloids, specifically nicotine, both in their nectar, but that this effect expands to basically be present in leaves, where the leaves of plants that are obligate outcrossers also contain fewer defenses. So selection on nectar has broader consequences for plant chemistry. Okay, so clearly if you're making very toxic compounds, you don't really want to poison your pollinators. So there may potentially be some trade-offs in the evolution of nectar chemistry. Okay, so we wanted to take a comparative approach and really zoom out and ask, does nectar chemistry predict microbial growth? And we did this across many different plant species that were found on our UC Davis campus. And we wanted to ask a few different questions. And in this study, I'll present data from a couple published papers and then one paper that's not yet published, hoping to get your feedback and ideas on how we can integrate these data sets and hopefully so I can interpret these data a little bit better. So thank you all in advance for your wisdom. So first, I'm going to tell you how we assayed the growth of a synthetic microbial community across many different plant species, how we quantified floral nectar chemistry, also across those same plant species, and how we're attempting to link those two. And to give you a little bit of background on the microbes that we used, we inoculated plant species with a microbial syncom, or synthetic community, containing both yeast and bacteria. And you've been introduced to a few of these players already, so thank you, Tad. To give a little bit more detail on each one, we used Metschnickovia ricofii, the nectar yeast that's very commonly found in many different plant species, sort of throughout the world, especially in temperate regions. This yeast metabolizes nectar sugars and is also potentially attractive to many bumblebee species under certain contexts. We also included the more generalist yeast Oreobasidium pullulans, which colonizes many different plant surfaces. And in a cool application of this fungi, it can be applied to flowers as a bioprotectant, right? So it can actually reduce the growth of plant pathogens. So it's often used in that context to reduce the growth of plant pathogens. We also looked at the growth of Apilactobacillus mitchneri, and this is where I tell you most of the people in my lab are currently working on how some of these microbes that are gained from floral resources like nectar and pollen actually inhabit or colonize stored bee products, like the provisions that bees make for their developing offspring. Right? When they collect nectar and pollen, they often put this in a little hole in the ground or a hole in wood, and the baby bee or the bee egg takes a few weeks oftentimes to hatch. So there's ample time for that stored nectar and pollen to potentially mold over. So we are trying to understand what these floral microbes actually do in stored bee food. And one of the microbes that seems to do particularly well in that habitat is Apilactobacillus mitchneri, which also seems to be acquired from flowers. Okay, our second to last member of our synthetic community is Acinetobacter pollinis, another bacteria. This one has a cool trick of being able to germinate pollen, lyse the growing pollen tube, and steal nutrients from that pollen tube. So it's good at creating its own resources or releasing resources that were unavailable. And finally, we have Neocomagetia, an acetic acid bacteria. that really acidifies floral nectar. Okay, so what determines the growth of these microbes in floral nectar? There's my one animation. All right, so we added all these species at one time to floral nectar and basically saw what happened. So before we added these microbes to nectar, we went out to the plant species in the arboretum. We bagged flowers that had not yet opened, and we've got our sample sizes here. We came back the next day, took off the bag, and inoculated flowers that had opened with our mixture here, with 1 microliter of nectar that contained the same relative concentration of each species. We waited 24 hours, or we re-bagged the plants, waited 24 hours, harvested those flowers, collected nectar, and plated them onto specific media so we could estimate microbial growth or the reduction in microbial growth using 3 different plate types. And luckily for us, all of our microbes are distinguishable on plates. So we could actually count them, distinguish them. And so, yeah, that's what we did across 31 different plant species. And what did we find? Well, we found that microbial density, this is the colony-forming units that we got on plates per microliter of nectar, varies by orders of magnitude among plant species. And remember, this is just across 24 hours of growth in a given plant, ranging from about 9 colony-forming units per microliter, so very low, to over 32,000. Whoops. And we have our different plant species here. So we've got a pretty good range. We also found that the composition of microbes that formed in the floral nectar also varied quite dramatically between plant species, where some plants were extremely dominated by a single species, Acinetobacter in this case, whereas others were highly dominated by Apiolactobacillus, yet others were basically maintained all of the species. And what we found was that this was sort of weakly associated with plant relatedness, where about 16% of the variation in microbial species composition was explained by phylogeny, and a smaller portion was explained by sort of the floral traits, what we're calling the pollination syndrome here, multiple plant traits that we sort of boiled down to pollination syndrome. Okay, so there's a lot of variation here. Can we predict some of this with nectar chemistry? So we set out trying to characterize nectar chemistry of uninoculated flowers. So basically, we took those bagged flowers, and when we would have inoculated them with microbes, we simply collected the nectar and used it for a few different types of analyses. One of them was one group was sort of these targeted analyses where we know what we're looking for. We can use standards and quantify these things. And so we specifically looked at amino acids and sugars using LC-MS methods. We also quantified known antimicrobial compounds in floral nectar, like peroxide. And no, those are not my nails. I have field ecologist nails. They're not that beautiful. But we did actually use these hydrogen peroxide pool strips, which I'm happy to talk to you about, but maybe if you have a pool, you know what these are. But they test the concentration of peroxide in a sample. And in tandem, we took some of the collected floral nectar and used it for untargeted metabolomics, again, using LC-MS and processing and downstream annotation with MZ-Mine, Sirius, and GNPS. I'm happy to talk more about that with any of you later, as that was a whole adventure in and of itself. So combining these multiple data sources, what can they tell us? So our main questions for this part of the talk are, first of all, do plant species or clades differ in nectar chemistry, sort of on the whole? Are known antimicrobial chemistries sort of widespread across these sampled species, particularly focusing on peroxide here as part of the nectar redox cycle that we think might be particularly active against in reducing microbial growth? And finally, are there groups of metabolites that predict variation in microbial growth? These could be nutrients, right? Microbes might be nutrient limited in floral nectars. On the other hand, there may be other antimicrobial compounds that we haven't yet described or that might be active across these plants. Okay, so what did we find? Well, first of all, something that we probably knew already, plant species differ in nectar chemistry. And this is particularly in sugars and amino acids. And you can see clusters representing different species. In this case, they're almost right on top of each other. Some are a little bit more variable. But we can explain 76% of the variation, almost all of the variation in nectar chemistry with plant species identity. Interestingly enough, we also see that plant clades do differ quite broadly in sugars and amino acids. So I just took the points and recolored them by these large clade groupings. And you can see there's actually some differentiation here in this NMDS plot. And what we found that explained most of this variation are, okay, it's a little crazy, but I'll walk you through it. So here we've got arrows representing the different compounds that we detected, with blue compounds representing non-protein forming amino acids, red representing protein forming amino acids, and these light blue are representing sugars, right? And what you can see is that they're sort of pointing within these compound groups, they're kind of pointing in similar directions, right? So the nectars that are particularly high in amino acids are over here. The nectars that are particularly high in some of these minor sugars are over here. And I'll show you a few representative plots for those individual compounds. We find that the asters are particularly high in some of these more minor sugars. Not necessarily the big three, are sucrose, glucose, and fructose, but some of these minor sugar constituents like melanocytose, this kind of interesting sugar that seems to be attractive to ants, the sugar alcohol, myo-inositol. What are they doing here? I don't know, but we should talk. We've got some ideas. And so you can see they're pretty well separated. On the other hand, we found that amino acids seem to be more abundant in some of the other plant groups. So we only sampled 3 different species in the monocots, and they're all quite closely related, but they had very high amounts of many amino acids. And the rosids that we sampled also, on the whole, had higher amino acid content than many of the asterids. Okay, so we have some both differentiation by plant species as well as plant clades. One of the more striking differences that we found was variation in amino acid concentration and composition across plant species. So here I'm showing you a slightly different view, but we have our plant species here, and we have the relative abundance of the different amino acids found in nectar. Some of our favorites here, proline, right, this darker blue, but we also find pretty high concentrations of glutamine, glutamic acid, as well as some more minor constituents, including neurotransmitters, things like that. So nectar is pretty complex. But what I wanted to point out here is this enormous range of magnitude of variation in the concentration of amino acids across different plant species, setting up some potential interesting hypotheses, which we'll return to that in a second. But this is sort of remarkable in the range of concentrations that we see. We also found from our untargeted analysis that the presence of secondary metabolites in nectar is also quite variable among plant species. And I'm showing here just the compounds that were annotated as terpenoids, although I have the same plot for carbohydrates, fatty acids, free amino acids and small proteins, shikimic acids, and phenylpropanoids, and a couple other different classes. So if you want to see anything, let me know. But what we found here were some sort of expected things and some interesting things I just wanted to point out. So here we have our phylogenetic hypothesis for the relatedness of these plants. And across many of them, we find some metabolites to be very common. Perhaps not surprisingly, we find abscison, or a plant hormone, to be very commonly occurring in floral nectar. On the other hand, some individual compounds are restricted to single plant species sampled here. This benzenoid, bisabolane, seems like it potentially is interesting. It also occurs in our Arbutus unido. If you remember, that was the plant species that had the lowest concentration of microbes, so maybe there's something going on there. We also found some known or well-characterized metabolites, like uridoglycosides, that occur in only some plant clades, and particularly clustered in some plant clades where we expect them. We may hear more about this kind of work later, but some of these compounds have been shown to reduce the growth of bee parasites, so they may be sort of active in these plant-pollinator interactions. Okay, so we've found that yes, indeed, plant species and clades seem to differ in nectar chemistry. Our known antimicrobial chemistry is widespread. So what did we find in terms of peroxide? Well, from both our sampling of the literature, or our sampling of floral nectar, or as well as our scanning of the literature, we found that plant species vary a lot in the amount of peroxide that's detectable in nectar. And this has been characterized mostly by Clay and some of his collaborators as a particularly antimicrobial compound in floral nectar. And especially within the Nicotiana, the genus Nicotiana, we find many plant species that are very high in peroxides. And we also found a few other plant species that have high or very antimicrobial levels of peroxide. However, on the whole, most of the plant species that we sampled have pretty low concentrations of peroxide. This was not what we expected. So what's going on with that? I'll circle us back to that in a little bit. I did want to point out the few plant species where we detected very high concentrations of peroxides were this Arbutus, as well as Ruellia. And from our lab experiments, we've found that some of the really strong antimicrobial effects happen basically above 1,000 micromolar. So below that, we're not seeing a super strong effect of peroxides on the growth of at least some microbes. Okay, so finally, do we find that this nectar composition is associated with variation in microbial growth? So here we used a few different approaches, including leveraging mixed models, where we examined if microbial density in floral nectar was a response of the abundance of particular compound groups, as well as while we controlled for phylogeny. We also used sort of this multiple regression with chemical modules defined by co-expression patterns, where we can take expression or co-expression patterns of individual metabolites, group them together, and then use those groupings as predictors in a model. But in either case, we get just about the same answer, and that is that resource limitation is driving microbial growth in nectar. And specifically what I mean by that is the amino acid concentration in floral nectar seems to be the main driver of variation in microbial density within floral nectar. If a flower contains more amino acids, the microbes grow much better. And we can see the different clades sort of clustering a little bit here. But this seems to be our take home. Interestingly enough, we found that alkaloid concentration was very strongly related to total amino acid concentration. And so even if we look at alkaloids as a predictor, they also predict variation in microbial growth in the exact same way. Perhaps because they're part of the same pathways and there are many amino acid derivatives that are alkaloids. Okay. We also find a weak but significant effect of variation in nectar peroxide, where plants with very high levels of peroxide tend to have lower microbial density in floral nectar. However, even though this is significant, you can see there's a lot of scatter all around this. Peroxide does not predict it super well, and we think that that's probably because some of these microbes are very adapted to dealing with peroxides in floral nectar. So here you can see this is microbial growth, or the OD of this particular microbe grown in a plate assay. And this is the response of Oreobasidium pullulans to different peroxide concentrations. And it suffers very little negative effect until we get to high concentrations, and then it really doesn't do well. In contrast, our nectar friend, Mechnicovia ricofii, seems to tolerate even very high levels of peroxide pretty well, potentially by ramping up its catalase activity, and it can deal with it pretty well. We also have some evidence that the growth of this, this yeast, Mechnicovia ricofii, may actually enhance the growth of other microbes in nectar. And so here we have the final peroxides. We have nectar that we've spiked with very high peroxide levels, and this is our control condition, or our control nectar after 24 hours. And we added different microbes to this solution and basically looked to see what was the peroxide concentration after we had inoculated those microbes. And here is the nectar that we spiked with Mechnicovia ricofii. No detectable peroxide after 24 hours of growth. So this thing is probably detoxifying nectar pretty efficiently. And if we looked at the community response, we find the same thing. So it's likely reducing concentrations to low enough values, potentially even facilitating other organisms that couldn't grow in those high nectar, those high peroxide concentrations. Okay, let's see. Yes, getting there. I just have a few more data slides, maybe one or two. So finally, to return to this, this correlating variation in microbial growth and variation with nectar chemistry. Our approach basically yielded one result, that amino acids in nectar predict variation in microbial growth. We found no overall compound classes, at least using this approach, that seem to negatively affect microbial growth. However, I still don't think that this was, necessarily a bad approach, because what we did find was a few different plant species that seem to be fairly antimicrobial, right? And so these give us a few different targets for looking at the nectar chemistry of particularly antimicrobial nectars. These are one species of penstemon centranthifolius, Arbutus unido, and even this cotyledon. Each of them is characterized by particular chemistries that we don't find outside of that plant species. And so this comparative approach doesn't really allow us to pick out those particular compound types as antimicrobial, since they would have a data point of 1. So follow-up approaches could potentially dig in and try to understand if these compound classes are actually antimicrobial. So it may be that those compounds specifically, or potentially unmeasured floral or nectar traits, including potentially other proteins, enzymes, or other conditions within floral nectar. Okay, so to wrap up here, I think what we found is that there are potentially trade-offs in function in floral nectar chemistry. And particularly with amino acids, we find, if we look at the previous literature, that many plant species seem to have increased pollinator attraction when they put more amino acids in their nectar. But this doesn't really come without a cost. These amino acids can also really enhance microbial growth. So, so perhaps that it offers a cost. Secondly, peroxides. These can be really potent antimicrobial chemistries produced, interestingly, both by plants and by bees. But why don't we see it more commonly across plants? I don't really know, but we do in part of this study that I didn't talk about, we actually artificially enhanced peroxide levels using a glucose oxidase enzyme, and we noticed evidence of bleaching. So basically, we produced way too much peroxide. The plant couldn't quench it, and it suffered tissue damage as a response. So potentially, there's costs to avoiding autotoxicity. We see we expect to see that in high peroxide-producing plants, they have some sort of quenching mechanism so that they can avoid that autotoxicity. So perhaps that represents a cost to the plant. Finally, we found no widespread class of secondary metabolite with links to antimicrobial activity, but certainly we found a lot of interesting chemistry here to follow up on, and potentially some leads for maybe antimicrobial products. So with that, I'd like to acknowledge my collaborators, the land and facilities, our UC Davis grounds crew that maintain the plants, my funding sources, including NSF, USDA, as well as UC Davis support. And I just also wanted to extend a big thanks to the Nectar community. I know we've exchanged a lot of ideas with many of you folks, and so I appreciate that. And let's keep it going. So with that, I'd love any questions or suggestions you might have. Thanks.
I think we have time for a few questions.
Excellent. Yeah, go ahead.
Yeah, I'm curious if you have any data on how long individual flowers leave and produce nectar, and if that explains maybe some of the diversity that you're saying. Like if very long-lived flowers probably have to actually make a huge effort to keep the microbes out or something like that.
Yeah, I would love to put that in our model. We haven't measured that trait for all of the plant species, but we have some idea, and there is a lot of variation across these plant species and how long the individual flowers are how long they live. So arbutus is a short-lived flower. So I think that, we've broken that hypothesis already, but it may be that other plant, there are more plants that would follow a prediction where longer-lived individual flowers might exhibit more antimicrobial chemistry. I don't really know. I also want to give the caveat that these are sort of Mediterranean kind of plants. We might expect very different patterns if we look at plants sampled from tropical environments or other types of habitats. So yeah, I bet there's a range of strategies out there that we have not sampled yet. Yeah, Colin.
So I guess this is a question, because over time, a lot of times pollinators are active in the morning because that's when there's lots of nectar. And then it dries out during the day.
Yep.
During that dry down period, is the concentration of secondary metabolites much higher? Because you measure the concentrations at full nectar capacity. So my question is would you expect the same result if you sampled late in the day when nectar levels are really low?
Yeah, this is a super interesting question. So this all assumes basically that nectar is a static sort of chemical mixture. I expect that that these metabolites would probably be at a much higher concentration, but also that nectar might be totally absent. And so just the absence of that sort of aqueous solution might be really stressful to microbial growth. We find that a lot of these microbes actually form biofilms that might be in part to stick to pollinators' mouths. It might be in part to protect them against this dry down period. We don't really know. So yeah, I think that could be really important to consider, but we haven't done it yet. Yeah, thanks.
Okay, I think
Okay.
we'll move on. Thank you so much. Up next, we have Marjorie Weber, who is joining us from the University of Michigan. Her work focuses on spanning and integrating across lots of evolutionary scales to understand between-species interactions. And today we'll get a small slice of that in her talk titled Plant Rewards for Tiny Mutualists, the Hidden World of Plant Mite Mutualism.
Thank you. It's been really, really fun to be here. There's so many people in this room whose work I admire, and it's been really fun to be a part of the conversation. So thank you so much for inviting me. Okay, so my research lab, we study a bunch of different plant-animal interactions, and the goal of our research is to sort of connect ecological processes with macroevolutionary patterns. So we ping-pong between the ecology experiments and the macroevolutionary patterns. And to do that, to link these two, we focus on studying adaptations where the main, mechanism or the main function of those adaptations is to mediate a plant-insect interaction. So we look at how it evolves and how that adaptation operates in ecology. And so we study a lot of these different types of ecological adaptations that mediate plant-insect interactions. So I study extrafloral nectaries, I study plant defenses, and I study sticky plants that capture insects and sand and present carrion to different insects, and I sometimes study flowers too. So I was like, what do I talk about? But I decided today to not talk about any of that, and instead to talk about a particularly tiny, particularly interesting trait that's found on, it's a plant reward trait that's found on many native trees around here, and I'm particularly obsessed with it right now, so that's the story today. Okay, so once upon a time, there was this man. His name was Axel Lundström. He was a Swedish botanist in the 1840s. So in 1887, he was 40 years old. So he'd been looking at plants for a while by this time. And he was looking at a leaf, and he noticed something. And this is translated from Swedish, but he said, in this are ordinary limes. Like I was looking at this boring lime tree. And by lime, he meant a tilia, a European tilia. It's like a basswood. And he said, I was looking at the nerve axils, so the veins on the lower surface of the leaf. I was checking those out, and then this is a direct translation. He said, there are beards. And lo and behold, if you look, there are tiny beards there on the underside of the leaves, and there's a drawing of the beard, and there's Darwin's beard, so you can see they are tiny beards. And if you look at those beards, those beards are sort of often covering a little cave or like a little indentation in the leaf surface there. And he sort of said, there are these little beards, and they're covering a little indentation. And he said in this paper that he wrote about these little beards, that when he looked inside the beards, they were full of mites. So it's kind of a fuzzy picture, but you can see they're full of mites. Will my video work? Let's see. Oh, yay. Okay, so here's a video I took of a dogwood tree. You can see these little beards. We're just going to zoom in. I took this with my iPhone, so I apologize. That's why it's really bad. And that's a paintbrush, and I'm just agitating them, and you can see the mites. They're like, ah! You can see one's going to I'm like, hey, what's going on in there? Okay, here's an SEM we took in the lab recently. You can see a mite tucked in the little beard hairs. Not only are mites in there, he said, I'm noticing a lot of mites in there of all different stages, of all different ages, from nymphs to adult, but also eggs are in there. So if you cut away the leaf surface, you see that tucked in the hairs, it's like a nest of little mite eggs. And so in this paper, he coined the term domatia. He said, I shall call them domatia, from the Greek word domus, that means the diminutive of home, a little tiny domicile. And he said, lots of plants seem to have them. I'm looking around, and I'm noticing lots of them. And he's like, they're not galls. They're there whether or not the mites are there. And he presented a theory in this paper also. And his theory was that these beards mediate a mutualism. And he said in the paper, I think the plants are offering the mites a home, so protection via home. And he thought the mites were helping the plant in some way, but he wasn't sure how. He put out some ideas. He thought maybe they were cleaning the leaf surface, or maybe they were providing nutrients from their mite poop. And so he put out this paper, and he said, domatia mediated mutualism between plants and mites. That's his hypothesis. And he was basically laughed at by the scientific community. That's not the actual scientist. I Googled scientists from that era, and that's the picture. Anyway, so people responded in the literature with a series of attacks. At the time, mutualism wasn't a well-accepted default explanation for interactions. It was all about competition, survival of the fittest, nature is red in tooth and claw. And he was mocked in the literature. He was even called Panglossian for thinking that these things mediated a mutualism. And people came back with other ideas. Somebody said, I think the mites are just trapped in there. And he wrote back in another paper and said, they're moving like freely in and out. I can see them walking in and out. And then there were some other publications. There was one that said, sort of the take home was like, mites are bad, maybe the mites are herbivorous and they're eating the plant. And the plant has created a structure that like concentrates the damage. So like if you're gonna eat me, come right here. And there's which I think doesn't really make adaptive sense, but there's an analogous structure supposedly like this in lizards called a nuchal pocket that's behind their ear, where I guess lizards are like, I don't know, I don't study lizards, but I guess they're like concentrating damage by ticks in this one area, and so they thought maybe it's a place to concentrate the damage. And Lundstrom wrote back and said, I think that the mites in there aren't eating the plant. They don't seem to be herbivorous mites. They seem to be predaceous mites or fungivorous mites, he thought. So then that kind of like, the back and forth sort of stopped, and time went on, and they all died. And it wasn't like settled. And then nobody really studied these things because you don't want to be mocked. Until like 100 years later in the 1990s, when a group of biologists, some faces you might recognize here, started to do experiments on these things. So they started doing experiments where they would remove or block the domatia on plants and see like, well, what happens? And so like here you can see a domatium and then it's shaved, and they said like, what happens if you shave them? And what happens when you shave them is that you have like 36 times more mites on your leaves, and these are, again, beneficial mites. And by beneficial mites, I mean different sort of generalist mite taxa that are either predators or fungivores, almost exclusively living in these domatia. They also would sometimes block them with tar instead of shaving them, and then have little tar controls. And when they did that, they found similar results. You'd have a lot more mites, and you'd also see these explosions of things like powdery mildew, these outbreaks of things like powdery mildew, and spider mites, the bad ones. And the results were so strong that domatia were proposed as being potential alternatives to fungicide in grapes because they were such good defense against powdery mildew. So from that research in the 90s, what we know is that plants with domatia have more beneficial mites from the plant perspective. Increased mite abundance decreases pest and pathogen abundance for these few pathogens and pests that have been studied. And this can increase both plant and mite fitness. So in other words, Lundstrom was vindicated 100 years later. Domatia can mediate a mutualism between plants and mites. And that's really the only viable hypothesis for what these things are doing. Yay. Yeah. But what else do we know about domatia? And the answer to that is not a lot, like almost very little, very little. And that's partly because they're easy to miss. Here's a picture that we took, and you can see, okay, like we love extrafloral nectaries. There's also a mite domatium in there with some mites, but we're like, zoom in on the ant. So in defense mutualisms, ants have gotten most of the attention. I love ants, but we know relatively little about this cool, widespread mutualism. So I've been lucky to have a series of NSF grants that have sort of been aimed at understanding what we can about mite domatia in an evolutionary and ecological context. So today, I wanted to sort of give you a little taster of some of the things we've learned over the last few years about mite domatia so that you love them too and go out and study them. Okay. So some of the questions that we didn't know when we started the grants were, how many plant species have mite domatia? Where are they found geographically? Do they interact with other plant traits, like other trade-offs or synergisms with other plant traits? How do they function, really, to impact leaf ecology beyond just these pests and pathogens that have been studied? And what are their genetic underpinnings? So I'm going to go through these and just give you a little taste of what we've learned about each of these questions. Okay. How many plant species have mite domatia? A postdoc in the lab, Andrew Myers, went through, what we did was we went through the literature, we went through herbarium specimens, they're preserved in herbarium specimens, and we went through species descriptions. We gathered as much information as we could on both the presence and absence of mite domatia. We confirmed mite domatia reported so far in over 2,000 species, in over 500 genera, and 105 families. So you can see, one mystery is that they're almost, well, they really are only in woody plants, pretty much. There's a few, very few. That's a mystery. So I'm really, that's something. Tell me why. I don't know. When we look at how those species are distributed across the plant phylogeny, we see interesting patterns. So here's the vascular plant phylogeny. I didn't label the tips, but each tip is a family, so you can see. And if we paint the branches with mite domatia on here, we see that mite domatia pop up across the plant phylogeny. They're in the families aren't like all lumped together. These have been gained and lost many times. We don't see them in the early branches because they're not in gymnosperms because you need a broad leaf. They're not really in monocots because you need vein axils, and those have parallel leaves. But once vein axils pop up, they start popping up over and over. Using this phylogenetic information and the information on sampling and gain and loss, we estimated, a very conservative estimate, is that mite domatia are present in about 10% of broadleaf woody species. So like 1 in 10 globally, if you go out, you could find mite domatia, including lots of really ecologically important and beloved species. So oaks, members of the red oak clade, beeches, many maples, tilias, as I noted before, ironwood. I'm just kind of like highlighting some that are around here for fun. Black walnut, many dogwoods, and many other trees and shrubs that are in your backyard, maybe. And then also many agriculturally important species. Did you drink coffee today? They have mite domatia. Do you enjoy wine or cherries? So they have agricultural relevance. We also know now that they come in different shapes and sizes. They're not just beards. They can take on interesting shapes. But the most common morphologies are these 3 morphologies. So the beard on the far left, left. Then these caves, which are sort of like like a flap of laminar tissue. And then these pits, which is sort of like an igloo but like the hole is in the top. Here's some pictures of the actual ones. So there's some tough domatia, SEMs that we took in the lab. There's some cave domatia. And here's some pit domatia. And you can see that's like the top of the leaf, the bottom of the leaf has the holes. And if you flip it over, you can see the little bubble. And here's just a cool SEM from a paper of the opening to the mite domatia. You can see like the mite palace and its egg. Okay, so how many plant species have mite domatia? They're very common, we estimate about 10% of broadleaf woody species. Where are they found geographically? Okay, so if we think about where plant rewards are found, plant defenses are found, indirect defenses, or plant mutualistic traits are found, we can generate some hypotheses about where these might be found globally, right? So some of these hypotheses that we thought about and that had been in the literature were maybe they're found in dry climates more, maybe they're adaptations to dry climates where domatia protect mites from desiccating. Or maybe they're found in humid climates. That was also in literature where there's higher fungal spore abundances on leaves. There also were some latitudinal hypotheses. So they were hypothesized to be more commonly evolved in high latitude places where they protect mites from cold, but also in low latitude places where where traits related to species interactions and defense may be more likely to evolve. Or maybe another hypothesis that's out there is that maybe mites are just like aerial plankton. That's a real phrase about mites. And that this is just a generally good defense, and that maybe they're just kind of like ubiquitously popping up in lineages anywhere. So we mapped the distribution of plants with mite domatia on the map. We mapped it on the map. And what we see is that species with mite domatia are distributed globally. There's in many, many places in the world where if you go there, you will find species with mite domatia. If we account for the sampling and the phylogenetic relatedness of these species, we see that one of these hypotheses does pop out overall as being supported above the others, which is that mite domatia are evolutionarily associated with temperate climates. So there's lots of distantly related lineages in the temperate region with evolving mite domatia, having mite domatia. There's lots of species in the tropics with mite domatia too, but they tend to be clustered in a few clades, and I'll talk about them in a minute. We also see that if we look at plants that invest in mutualistic defensive reward traits, and we split up, are those mite domatia or are those traits that are more focused on ants, like ant domatia or extrafloral nectaries? We see that shifts from the temperate zone to the tropics. So here you can see, sorry it's a little fuzzy, I don't know why. But you can see where we increase latitude, the proportion of species that go in for these defensive traits shifts from being more primarily predominantly ant-based in the tropics to predominantly mite-based in temperate regions. So that's kind of cool. We also saw this really neat pattern that when we see mite domatia in the tropics, they tend to be repeatedly evolutionarily going towards these pit forms. So in other words, we have these beards in the temperate regions, and then we have these pits in the tropical regions. Sorry, I should have put beards again at the bottom. I don't know why they didn't pop up. Okay, yeah, so we see this convergence. And so we're here. So if you went outside and looked for mite domatia now, you would find the beards, and I hope you do. So what we're seeing is this large-scale latitudinal convergence in this widespread but understudied mutualistic reward trait. And I don't know why, but it is very, very, very repeatable across the phylogeny. Suggesting, given that, that convergence is a signature of adaptation, suggesting there's some sort of cool adaptive story there. Okay, so they're globally distributed, but often in temperate regions. Do they interact with other plant traits? Okay, so we've had a lot of interesting, adventures into thinking about what plant traits these might interact with. I'm going to highlight one, or maybe two. Okay, so the one I'm going to tell you about is we were curious whether plants with these mite domatia, if they invest in mite domatia, if they don't invest as much in other defenses, like direct defenses. So there's this hypothesis that direct defenses and indirect defenses might sort of evolutionarily trade off and ecologically conflict with one another. This hypothesis comes, I think, originally from Dan Jansen, who was like going around tasting acacia leaves, and he was like, "Those ones are like spicy, and they have ants." I think that, so the idea here is that plants that invest a lot in direct defenses, so these are traits that directly repel harmful arthropods, things like high trichome density, lots of metabolites, that those direct defenses could deter or poison their bodyguards, sort of like what Rachel was talking about with poisoning your mutualist, but poisoning your bodyguard mutualist here. And that would interfere with the indirect defenses. So it'd be costly to have both of those traits. You wouldn't do that. This hypothesis hasn't been super well tested using a sort of a large-scale phylogenetic perspective. So we were interested in doing this with mite domatia. So we did this in the wild grape genus Vitis because there's a lot of variation in mite domatia presence and investment in size across the phylogeny. And they're easy to grow in the lab in a common garden. So what we did was we looked at, we quantified the direct and indirect defenses of 23 species of wild grape in a common garden in the lab. So we looked at defensive chemistry, investment in domatia, and we also did a palatability assay with a generalist herbivore to see how tasty they were, just the leaf material was. And what we found was that across the phylogeny, species, over evolutionary time, species that evolve heavy in investment in domatia lose their direct defenses. So they've lost their chemical diversity. So when domatia size goes up, the phytochemical diversity of the leaf goes down. And each dot there is a different species in the common garden. And that chemical diversity, phytochemical diversity, relates to how palatable they are. So species with lots of phytochemical diversity are more less palatable to a generalist herbivore than the ones that don't have that phytochemical diversity. So this trade-off, I think, is really exciting because it supports this hypothesis, which I think is cool, but also because plant defense trade-offs are hypothesized to play a role in generating and maintaining plant diversity. And so understanding mechanisms that facilitate these drivers of diverse defensives could be tied to the evolution of hypotheses about the evolution of plant diversity. But but mite domatia might not just trade off. They might, with other plant traits, they might act synergistically with other traits. And indeed, we have found that some other defensive traits, or some other traits in general, act synergistically with mite domatia. So in particular, we have research showing that plants that have extrafloral nectaries and mite domatia have more mites. I wrote a paper calling this the bed and breakfast hypothesis, which the editor told me not to call it, but then I fought for it. I think I was still a graduate student when I did that. So. But what we find here is that if your mites have food, then there are more of them. We don't know if the mites are eating the nectar or if they're eating the fungi that grows on the nectaries, and so that's something I'm really interested in trying to find out. But we also see that pearl bodies, which are small plant structures that have lipids and fatty acids in them, that they can be associated with mite abundance on leaves associated with Domatia. And then pollen is the other really big one. So basically these food sources, bed and breakfast. So do mite Domatia interact with other plant traits? Yes, we see trade-offs across at the genus scale with plant chemistry, plant-direct defensive metabolites. And then we see synergisms with food rewards, both in ecological experiments and, I didn't show you this, but also in a phylogenetic sense across genera. At the genus scale, we see that Domatia and extrafloral nectaries are evolutionarily correlated in viburnum, one of the groups we study. Okay. How do they function to alter leaf ecology? Okay, so as I mentioned, there was this sort of set of studies that looked at how Domatia impact mite numbers and how that impacts sort of these specific pests on leaves, powdery mildew and spider mites. But we haven't really branched out to understand what's going on in sort of the general leaf ecology beyond those target pest species. Our goal with this is just to sort of link leaf trait evolution, so the repeated evolution of these mite Domatia, to the structure and function of the phylosphere via mite Domatia. Like we see this repeated evolution of this convergent trait, mite Domatia. What is that doing to the phylosphere ecology? And this work is being led by a postdoc in my lab, Julia Boyle. So we've done this, 2 types of experiments that I'll show you results from. Both of these were common garden experiments where we took a bunch of species in a genus and that vary naturally in their traits and put them together and looked at their leaf microbiomes. We did this for Vitis and Viburnum. We had in those common garden species that naturally lack Domatia, species that naturally have Domatia, and then for those species that have Domatia, we blocked them so that we could create experimentally species that lack Domatia as well. And then we sequenced the leaf phylosphere, the fungi and bacteria to try to understand, and we have the phylogeny to try to understand how this repeated evolution of the trait impacts the phylosphere community. Okay, so I'm going to show you some of our preliminary results. This stuff is unpublished, so I'm really curious about feedback. Okay, so the first thing is we do see this pattern where mite abundance is negatively correlated with fungal, is negatively correlated with fungal abundance. So this really strong pattern is what other people have seen too, where if you have very few mites on a leaf, you can have these fungal outbreaks. But once you get above a certain number of mites on the leaf, you have very low fungal abundance. This particular graph I'm showing you is from plating out the fungi in the leaf, so to try to really get a good idea of the fungal abundance. So as mite abundance goes up, when Domatia are there, fungal abundance goes way down on the leaf. But if we sequence what's there, we see that although fungal abundance goes down, the diversity of the fungi goes up. So in other words, Domatia, when they're present, I'm not showing you the bar graphs, but when Domatia are present, it increases the number of mites on the leaf by a lot. And that creates a low abundance, but high diversity microbial community on the leaf. So it's like little sheep grazing the leaf surface and creating this grazed down microbial community, fungal community, but it's a higher diversity community. But does that matter for the plant? Does that matter for the plant? So here are some data from the Vitis, the grape common garden, where I'm just showing you the top fungal OTUs. So I'm showing you the fungal stuff because most of the action was in the fungi, which isn't surprising because the mites are fungi eaters. But here are some of the top OTUs associated with mite abundance on the leaf. And the change with mites, so that you have the warm colors, the red colors are increases. So you see a lot of species are increasing with mites, but then you see one species that's really going down with mites. And does anyone know what that species is? Can you see it? It's powdery mildew. Yeah, so powdery mildew was the most abundant pathogen in the study. It was really high abundance, and it was really common across the species in the common garden. And it was the species, the top OTU that really went down with the mites, which was really cool. So what we think is happening, so what's happening is, is when you have domatia, the abundance of powdery mildew goes down. But what's really cool is when the abundance of powdery mildew goes down, which is on the y-axis there, the fungal OTU evenness goes up. So I think that's super cool. So our working hypothesis is that domatia, they create this low abundance, high diversity microbial community in the leaf by suppressing the competitive dominant. In other words, mites are like the pie zest or sea star of the leaf. Thank you. Yeah, thank you. They're suppressing the competitive dominant, which is allowing for higher evenness. And because that competitive dominant is an outbreak pathogen, that can increase plant fitness, thereby selecting for these mitomatia. So that's my working hypothesis for how the community ecology of the leaf surface is selecting for, or these interactions on with the community ecology of the phylosphere are selecting for the repeated convergent evolution of this simple trait on the plant leaves. That's how that's how we currently think they function to alter leaf ecology. Okay, and then finally, I just want to tell you briefly about our work on the genetic underpinnings. So we were interested in understanding, can we uncover the genes that are underlying mite domatia evolution? So the goal here was like, okay, we have this phenotype, this convergent phenotype. We know where it's evolving, when, and in what habitats. We can now link it to the community ecology of the phylosphere. Can we link that now to the genotype, getting sort of this like large-scale macroevolution, global macroecology perspective down to this repeated genetic adaptation, I guess. So we did a transcriptomics study. I'm like a little shocked I'm standing up here telling some of you about a transcriptomics study with a collaborator, Eleanor Ritter, who was a PhD student and is now postdoc at Georgia. And Eleanor was in Chad Niederhuth's lab at Michigan State. And what we did was we had Vitis riparia, which is the riverside grape. We took a genotype that consistently has very, very diminutive or almost no domatia. And we took a genotype that has really big kind of hunk in domatia. And we sampled the domatia tissue, the leaf laminar tissue right where the domatia were while the leaf was expanding. And then we also, at the same time, sampled just laminar tissue from both, just like a little bit away from the domatia. So we could isolate candidate genes that were being differentially regulated in the big domatia species in the domatia. So we weren't just capturing stuff that's like different between the genotypes. Does that make sense? Okay, so what we found was a set of candidate genes that sort of point to some interesting things happening. So we found these trichome-related genes that make sense because they're making these beards, right? They're also like changing the structure of the leaf because they're creating, remember, that cave that's underneath the beard. So we think that has to do with these leaf development-related genes at the bottom. We also found defense-related genes that were candidate genes. So powdery mildew-resistant genes, which is really interesting. And I don't know if that's because maybe the genotype that isn't doing the mite domatia is doing something else to defend against powdery mildew, or if it's because the expression of domatia is tied to something that people have been thinking is a direct defense when it's an indirect defense. Anyway, I don't know, but that's interesting. And then some JA-related stuff. What really was cool for me was that I was like, so I'm an evolutionary ecologist, and I always like it when people find like the gene, but I'm always like, that's cool because they get like the gold star. And I was like, I want to find the gene, and then I can give a talk like this where I'm like, we found the gene. But what I didn't realize was that it generated some really interesting new hypotheses about the ecology that we didn't have before. So I'm going to tell you two of them. One is that genes were lighting up that are involved in volatile synthesis. And I think they maybe smell, like that generates the hypothesis that maybe they smell good to the mites. And so now we're trying to think like, how do we sample a tiny headspace and do like little Y-tube experiments with the mites? And then the other thing is it lit up, some genes lit up having to do with sugar transport. And so now I also wonder if there are tiny nectaries in there, or so there might be a breakfast in bed situation. Yeah. Thank you. And we did a GWAS of Domatia and wine and table grapes because in a lot of these cultivated species, mite Domatia have been bred out of the species relative to the wild relatives, and we spray for powdery mildew pretty aggressively. So we're interested in trying to know, are Domatia viable tools in sustainable agriculture, and can we think about that? In conclusion, some of the things I've told you are that mite Domatia are super cool. They're widespread across the plant tree of life. They're estimated to occur in about 10% of broadleaf woody plants. They're found globally, but they're most common in temperate environments, yet their morphology we're finding really widespread convergent morphology across latitude. I also told you that mite Domatia evolution trades off with investment in direct chemical defense at the genus scale. And I told you about the bed and breakfast hypothesis. And I told you that the evolution of Domatia alters the phylosphere community dynamics, so it lowers microbial abundance and increases diversity, the Pisaster sea stars of the leaf. And finally, I told you that mite Domatia genomics can offer an opportunity to link genotype to phenotype and to ecology. So with that, I hope I've done Lundstrom proud 100 years later, and I would like to thank you all for listening.
Okay, we're doing great on time, so questions.
Yeah, go ahead.
The winter, like they shed all the leaves, like they regain every season.
Yeah, okay, so the mites overwinter in the bark and the bud scales, supposedly. You might think maybe that's the reason why they're only on woody plants, because they need the bark to overwinter. But they're also only on woody plants in the tropics, where they don't need to overwinter, so explain that. One thing we're doing now is actually we're going out in the winter, we've been going out in the winter and like spraying the bark and like getting all the mites out that we can out, and then sequencing the leaves in the spring to see if the mite communities are the same in the bark. And we're doing that with trees with and without Domatia to see like what's happening in the winter. But I don't know. I mean, we know so little about the mites, like in general, and what they're doing. And, yeah.
Are there cheaters in the system? Like, do herbivores move into the Domatia? And if so, are there controls? Do you know what's known about it?
There's so few cheaters. It's really remarkable. Sometimes you're looking at a leaf and there's like a big old rip in there or something. But it's really rare to see cheaters. And I don't know why, because it kind of seems like they're open for business. But it's rare. Yeah. No.
What kind of diversity is there in the mites?
Oh, this is so fun. Okay, so that was your question?
What was the question?
Oh, so I'm sorry. The question was, what sort of diversity is there in the mites? Yeah, so we know very little about the mites. We know that the mites come from about 10 families of generalist mites that are predaceous or fungivorous. There's some like specialist things, like there is one mite that we just found in like Iran that is a specialist, it's a predaceous mite that's a specialist on the fungivorous mite. So there's like some cool stuff. I recently was talking to an acarologist, Barry O'Connor, who is retired, and he told me, I was like, okay, we've gotten these into morphospecies, we know they're in this family, like we have like 5 morphospecies in each family, we are sequencing them and we're getting them down to this. How much cryptic diversity do you think there is? Like how many species of mite do you think is in each morphospecies? And he was like, maybe 45. So like I think there's phenomenal opportunity to study community ecology of the mites, and now we've just sort of started. We have a new method where we're taking forensic tape, DNA-free tape, and sticking it on leaves. And then when you pull it off, you can sequence the mites and the fungi on that leaf. And so our next push, hopefully starting this summer, is to really get into the sequencing and description of the mites and to start doing some experiments with mite feeding to figure out what's going on. But it's a big open area. Yeah. Yeah.
Do you ever see?
Some of our species have extrafloral nectaries that are really targeted towards ants and mite Domatia, like that one I showed you. The ants are really big compared to the size of the Domatia. I've never seen an ant eat a mite out of a Domatian, but I wonder if they do. That's like a little snack. But I don't know if they interact. I wonder also if the mites clean the extra floral nectaries or keep kind of clean extra floral nectaries, like if they benefit each other. But we don't know that either. Yeah, we have no evidence of them interacting, but I have suspicions. Yeah. Oh, sorry.
Last question. We'll do one more.
So it's really weird that your domatia don't have other denizens moving into them, typically other mites. Are there any volatiles that are being produced specifically in the domatia, or other like waxes or secondary defenses that are only found in those areas?
A lot of species, if you look at the domatia under a microscope, you see like weird stuff happening. Like the trichomes aren't normal leaf trichomes. They're like gunky. They're like yellow. They're I'm sure there's interesting chemistry and waxes and stuff happening, but none of that has been studied. Yeah.
Thank you so much.
Yeah.
And then our last speaker before our short break is Lynn Adler, who is joining us from the University of Massachusetts Amherst. Her work simultaneously investigates antagonistic and mutualistic interactions, and so I suspect we'll get to hear a little bit about that today in her talk titled Floral Rewards Mediating Pollinator Pathogen Dynamics.
Thank you. I'm going to make sure I've got this right before I blind anybody. Okay, good. Hi, everybody. Thank you for the introduction. Thank you to the organizers for inviting me. I have learned so much today and feeling so inspired. It's really wonderful to be part of this. Thank you to all of you for still being here. I know we're getting close to a break, and I appreciate that you're all still in the room. I started out my graduate career in an herbivory lab, and I still sometimes like to frame things in those contexts. So thinking about tritrophic interactions, for example. We think about a plant. In some audiences, I'll say, what plant is this? But I know you guys know clays here. This is tobacco. So we think about plants. Plants make lots of different kinds of chemical defenses to protect themselves from getting eaten, to reduce herbivory. But if herbivores can tolerate those defenses or even sequester them for their own use, then that can help protect those herbivores against their own natural enemies, their own predators. And given how hard it is to come up with any kind of generality in ecology, it's sort of amazing for how many different plants, chemicals, herbivores, natural enemies, we see these relationships. So that's nice, but here we are in a plant reward seminar. And yeah, so we saw people talking about pollinators today, plant rewards, and what's a pollinator? Right? I mean, from a plant point of view, interacting with a pollinator has a very different outcome than interacting with an herbivore. But from an animal point of view, a pollinator is a really very specialized kind of herbivore, right? It eats nectar and pollen. It specializes on those tissues. And as you've learned a lot about today, the more we look, the more that we find that nectar and pollen contain plant defenses. This becomes maybe particularly interesting these days as people are concerned about pollinator decline, and while that is a multi-factorial issue, one of the stressors that has been implicated in declines is parasites and pathogens of pollinators, which leads us to wonder, can we take what we've learned from decades of studying plant-herbivore natural enemy interactions and apply that usefully to try to understand something about plant-pollinator pathogen dynamics? Another way of asking this question is, how do plant communities affect pollinator health? And if we have a bee and she's foraging in a field and she's visiting a bunch of flowers, she's getting different things from those interactions, right? I mean, the obvious one that she's getting is food, and when I first made this slide, it was late and I was punchy, so I got a little silly, but she's getting sugar, she's getting carbs, that's the lollipop, from the nectar that she's consuming. She's also getting proteins and lipids, that's the hot dog, from the pollen that she's consuming. And we know that, and I do a little bit of work on nutritional ecology, but not a ton. But what if she's also getting medicines from the plant rewards, the floral rewards that she's consuming? And a third possibility to consider is, I'm thinking of flowers might also be a little bit of nature's toilet bowl. Think about the rest stops, you stop, you get your snacks, but you also go to the bathroom. Pollinators are flying, they're foraging, they're also defecating. There's a bunch of different pathogens that are transmitted fecal orally. Flowers are a hot spot. They can have 10,000 times the number of the density of insects as leaves do. So these are also high traffic areas that have the potential to share germs. In my lab, we actually study the toilet bowl quite a bit, but that's not what I'm going to talk about today. I only have so much time, and I'm going to focus on some of the medicine stuff. But feel free to ask me about the other things. So today, in my limited time, I'm going to tell a very particular story to you guys. I have to start by convincing you of something, which is that sunflower pollen is amazing. So we have to start there. And once I hope I've convinced you of that, I've made my case, I would like to think a little bit bigger and think about the landscape consequences of sunflower pollen for bee health and reproduction. And then once we talk about that, then I want to get a little bit smaller and address the obvious question of what is it that makes sunflower pollen so amazing, and then end with telling like a little bit of, sort of giving you some hints of where we're headed, some things that aren't quite ready for primetime sharing, but where we're going. So I'll start a little bit by just introducing the system that I do a lot of my work in. So I've been working largely with Bombus impatiens, which is the common eastern bumblebee. It is very common. It is by far the most abundant bumblebee species in eastern North America. It's really easy to work with because it's commercially reared. We can order colonies whenever we want. We can do work with them all winter. And I like to acknowledge that this species is not in decline. It's actually doing quite well. Some other bumblebee species are in decline. Bombus impatiens is sort of filling those niches. So we don't study it as a threatened species because it's not, but it makes a nice model system. I also work primarily with the pathogen Cryptidia bombi. I'm sorry, it doesn't have a common name. I'm going to just call it Cryptidia. It is a gut parasite. It is a trypanosomatid. It's related then to parasites that cause leishmaniasis, African sleeping sickness, Chagas disease in humans, but this is one that infects bees. It is transmitted fecal-orally, so if a bee defecates on a surface and another bee encounters that and consumes it, then she can get infected. It's not the baddest of the bad as pathogens go for bees. There are others that are more tightly associated with declines, but it does have a range of negative effects. It reduces lifespan, it reduces colony reproduction, it impairs learning and foraging, particularly if bees are stressed in other ways, like nutritionally stressed. What it is by far the most common pathogen that we see, certainly in our area, and most studies find as well. We easily find it in 50% of the bumblebees that we sample, and in some sites up to 80%. So yeah, there's a lot of it around. So that's the system that we work with the most. So to start with, this whole sunflower pollen is amazing. We do a lot of bioassays in the lab, and just to sort of orient you to kind of what those look like for us. We'll start by sort of isolating bees in individual containers. We don't use those little vials anymore. We use bigger deli cups. They like those better, but this is the picture I have. So lots of individual bees in individual containers. And we'll start out by dosing them all with the pathogen. So we start out by exposing uninfected bees to a known amount of the pathogen so that they're all sort of starting out with that same baseline exposure. And then we can randomly assign them to whatever our treatments are of interest. In this particular example, pollen diet. So maybe we give some sunflower pollen, give some rape pollen, we give some buckwheat pollen. This is not a hypothetical, this is what we did. Or we try to treatment with a mix of all three. I'm going to take one step back and say we didn't start out thinking about pollen. Like many of you in this room, I came from a background of thinking about chemistry and thinking about nectar chemistry. We started out working with nectar secondary compounds and found that, yay, some really reduced infection. And then we got grants, and then we did it again, and oh, now they're not reducing infection. It turns out that it's really variable, and we started to feel like maybe I was missing the boat by not thinking about pollen, this other really important floral award. So this was our first experiment, kind of saying, maybe there's something interesting with pollen. Maybe we shouldn't ignore that. So we had these 4 treatments: sunflower, rape, buckwheat, mix of all 3. So we infect them, we assign them to their diets, we feed them their diets for a week, and then we dissect out the gut of the bees and look at how intense the infections are that are developed in those bees. We do a lot of assays like this. So far so good? This is making sense? Great. Okay, awesome. This was actually an undergraduate honors thesis in my lab, Jonathan Giacomini. He then went on to do a PhD in this area, but I was like, Jonathan, how's your experiment going? And he said, it's so weird. It's like, this is really weird. When we feed the bees sunflower, they're just not infected anymore. So you're going to see graphs like this, where the y-axis is the Cryptidia count per sort of a known volume of gut solution. And here's our buckwheat pollen, results in pretty high infection, rates a little bit lower, the mix is even lower, but sunflower, most of the bees just don't have a detectable infection after a week of eating sunflower pollen, when we've infected them all at the beginning. Yeah, we did that too. Whoa. Having learned our lesson from nectar chemistry, we've then repeated this many, many, many times. It's remarkably consistent, and in fact, this was a crazy master's thesis, but here's another one of those. So here's our buckwheat pollen, high infection, a wildflower mix, just like what honeybees collect, sort of mixed pollen, and then here's 17 different Paleanthus annuus, either domesticated cultivars or wild accessions. They all reduce infection, and even there's two congeners, Helianthus argophilus, Helianthus pilaris, so two different sunflower species. And we fortuitously managed to get two different kinds of goldenrod pollen that summer from beekeepers, and which is in the same family as sunflower, but not that closely related. They all do it. They all do it. They all reduce infection. So I hope this, I saw a few wows, so maybe you're on board. So we found now that many different kinds of wild and cultivated sunflowers reduce infection, goldenrod too. Due to time, I'm not going to show you the data, but we've also, wondered, is this like an Asteraceae-wide thing? We've gotten some other Asteraceae pollens from an allergen company. 4 of the 7 reduced infection. A couple of others looked to me like they did, but the statistics weren't quite there. We have some new data, not quite ready for prime time, but we're getting more Asteraceae. We don't even know what they all are yet, but alone in mixtures, a lot of them seem to reduce infection. This is maybe not universal in the Asteraceae, but pretty widespread. This is Sonia Glasser is my graduate student. April Sharp is Becky Irwin's graduate student. So, hope you're on board. Sunflower pollen is amazing. That's cool. All of those things I just described were done in the lab with individual bees. And it's kind of nice to know whether the stuff you find in the lab actually has any consequences out in the field. I would say this is particularly interesting for a couple of reasons. So the lab to the field, that was the first reason. And the second reason is that sunflower pollen is kind of notorious for being low in protein and low in some amino acids that are important for honeybees. So you talk to a beekeeper, they're like, sunflower pollen, stuff's garbage. It's no good curing the common cold if you're going to starve in the process. So just because something reduces infection in the lab doesn't necessarily mean that it's good for bees in the field. Wrong button. So what did we do? We, where I live in western Massachusetts, there's a lot of sort of small, diversified farms, but we contacted growers in the area and identified 20 different farms that were growing varying amounts of sunflower. Where we are, it's not a huge crop, so 3 acres might be like the most that we would get, down to like a few rows at a small farm. And we put bumblebee colonies, commercial bumblebee colonies, out at each of these farms. So we ascertained that those were sort of uninfected colonies to begin with. Then established them out at the farm for the sunflower flowering season. We would collect workers coming back to the colonies at sort of every couple of weeks, all of them, and assess them for pathogens. Some of them we did visually, the way that I know how to do it, under a microscope, and half of them we sent to my molecular collaborator, Quinn McFrederick, who did a qPCR assay. The results are kind of the same. I'm going to show you the visual results. But then we can also measure the weight gain in the colony and queen production, so production of reproductive offspring in that colony too, to look at how do the colonies do. And we also measured all the flowering resources at those farms that bees would use, everything that was flowering. That was a lot of work. The interesting story came out with sunflowers, so that's what I'm going to tell you. But we looked at floral diversity and other flowering groups and things like that too. Yeah, we also looked with the qPCR, we also looked at other pathogens, but the interesting story was with Chlamydia. Yeah, so we were so excited, and this was work of former postdoc, Rosemary Malfi. So this one is now probability of infection. Did the bee have any detectable Chlamydia or not? Yes or no? And our x-axis here is the log of the number of sunflower heads. We did that instead of area because sunflowers, different varieties have different numbers of heads, different densities. But the salient point here is that as the amount of sunflower on the farm goes up, the odds that individual bees are infected goes And furthermore, of the bees that are infected, this is now that sort of cell count. So this is infection intensity. So of infected bees, the more sunflower on the farm, the lower the infection of bees on average. So like farm level sunflowers in the field reduce infection of foraging bees, which we were really excited about, but we were even more excited to look at queen production. So now we have the number of daughter queens, and as sunflower goes up, queen production in those colonies goes up. So not only is sunflower reducing infection, it's increasing reproduction at the colony level. Yeah, so what are the landscape consequences of sunflowers for bee health and reproduction? Farms with more sunflowers have bee colonies with less infection and greater reproduction, suggesting that like this one species, this sunflowers, the amount of it that you have on the landscape has these sort of measurable impacts on at least, it says pollinators, but really farmers and patients at least in these agroecosystems. And then everybody's always like, what's the magical thing? What is it about sunflower that makes it work so well? And you guys are probably a lot like me. The background that I came from, I assumed that there was some magical chemical that we would find in that pollen that would be the thing that would reduce infection. I have a biochemist collaborator, Phil Stevenson, who's at Kew Gardens in University of Greenwich. So he started extracting things from the sunflower pollen and the buckwheat pollen and looking at what was different and giving me chemicals. And then we would mix those chemicals in with just wildflower pollen as like a control at different concentrations, infect bees, see what happens. You maybe guess where this is going from my tone of voice. So each of these graphs is an undergraduate honors thesis. Okay? Yay. Yeah, and these ones are log counts now, which is why the sunflower doesn't look quite as dramatic as it did on the others. It's because we log count actually. So each year is a tenfold increase. So here's like buckwheat control. If we mix it with low root and medium root and high root and very high root, it doesn't matter. Sunflower still does its sunflower thing. These were spermidine coumarole derivatives. They'll synthesize some for us. Low, medium, high, doesn't matter. Sunflower does its sunflower thing. Okay, maybe it's not like defensive chemistry. What about all those fatty acids that can be antimicrobial that sunflower is kind of famous for? So like, yeah, there's a thesis, there's a thesis. Yeah, so 9 different fatty acids from sunflower. None of them did anything. And you know, there's only so long I can hit my head against the same wall before I start thinking that maybe I need to think about things some different ways. So 4 honors theses later, it's like, you know, maybe it's not chemistry. Maybe there's something else going on. If you look at a sunflower pollen rate, it looks downright dangerous. Buckwheat has a nice smooth exine, and sunflower has this really spiky exine. So how do we test the morphology? Nanoparticle printing is too expensive. We looked into that. Alka Koch was a research associate with Phil Stevenson at the time. So yeah, did a couple things. One is that we tried just extracting metabolites, so like the chemistry fraction, so just like a methanolic extract from either sunflower or buckwheat pollen. So we can use that for chemistry. But then there's also methods to basically extract all the innards of pollen and leave the shells. It's a kind of rigorous process, but it can be done. So you basically just get left with those exines, the outer surfaces. So we can have shells of sunflower or buckwheat pollen too. And then we mixed each of those with wildflower pollen. So we can have the metabolites or the shells of sunflower or buckwheat mixed in with wildflower pollen as just a base. And then feed it to bees and see what happens. Yeah, so then we can compare the effects of each of those mixtures to just straight up wildflower pollen, straight up buckwheat pollen, straight up sunflower pollen as just sort of negative and positive controls. All right, so I'm going to walk you through this because it's a little bit complicated, but here is just wildflower pollen on its own and how much infection it results in. With buckwheat, so control means just whole buckwheat pollen. Metabolite means buckwheat metabolites added to wildflower pollen. Shells means buckwheat shells added to wildflower pollen. none of these things are significantly different from each other or the control, or wildflower pollen on its own. But when we look at sunflower, okay, so here's sunflower metabolites added to wildflower pollen. That doesn't reduce infection, but here's sunflower pollen, and here's sunflower enzymes mixed in with wildflower pollen. They looked indistinguishable from sunflower pollen by itself. We kind of couldn't believe it, so we had Halka make a whole second batch of metabolites and shells and sent it to my collaborator, Becky Irwin, and she repeated it at Laura Figueroa. This came out a couple years ago. She repeated it at NC State, and we got the same results. Yay! So yeah, there we are. Something about the enzymes. Something about those shells. So how does it work? We tried. We see very little evidence that it's any kind of chemical mechanism. It seems like there's something about the exons or the spines that are the key to interfering with this infection. This means we can look at other families that have spiny pollen too. Asteraceae are not alone in that. The hibiscus Malvaceae. Thank you. I have a moment. The Malvaceae family, the Cucurbitaceae. There's a few others out there that also have spiny exines. And actually, yeah. If you're interested in that, Becky's grad student April Sharp I just learned about some things last week, but I didn't put it in here. It's not my data to share. So conclusions and some things that I didn't get to talk about because I was worried about time, but then I'm going to have a what next, so we're not quite done. But like conclusions from the stuff I showed you so far, sunflower pollen consistently and dramatically reduces Cryptidia and common eastern bumblebee. I never see things that we always get the same result. And with sunflower pollen, you always get the same result. I didn't show you this, but it reduces infection in queens as well as workers. All the studies I showed you with workers, but it works for queens as well. People ask you, like, what kind of sunflower should I plant? I tell them any kind of sunflower. It works with many different kinds of sunflower. Many other kinds of Asteraceae pollen seem to be effective too, which is exciting because Asteraceae are abundant in almost every ecosystem there is. If we have, it works in the field, so more sunflowers on farms result in lower colony infection and higher queen production, so actually higher reproduction of those bumblebee colonies. I didn't show you this because I was worried about time, but we have done some work trying to figure out what it is that's going on in the bee, and sunflower seems to induce a laxative effect that might perhaps flushes out these, but there's a little bit more on this. It also seems to upregulate some immune genes, although our evidence for that is kind of mixed. It looks like it might be something to do with the spines and not the chemistry. And it suggests that this might be, sunflowers may be one key species that is important for pollinator, at least fungus and patient's health. Sunflowers are native to North America. Helianthus is native. So that might be important in native as well as agricultural habitats. But we're not done. I want to tell you kind of some of the stuff that I'm excited about that we're doing now. I lead a 5-institution NSF integrative biology grant. The goal of integrative biology is to span across scales, and so I'm super excited to find Megan Povolones, a molecular biologist who studies trypanosomes because they affect humans. Because I have zero pathology background, okay? I'm like decent at plants, at insects, and I know like pathogens are not really my area. So I always feel like basically I'm sort of like taking a sledgehammer, smashing stuff up, and then trying to figure out like how it broke. So in this we have, I'm a molecular biologist, a mathematical modeler, Chris Myers from Cornell, a biochemist, Phil Stevenson, and several ecologists, Laura Figueroa from UMass, Charlene Jha from UT Austin, Becky Irwin from NC State, with a goal of understanding the molecular mechanisms, assessing impacts on pathogens and bees in the field, and using models to integrate across scales. So we're really excited about this. It's moving along, but I want to give a particular shout out to Megan and the things that she's able to do that I would never be able to do. So she comes from a background of working with Cryptidia fasciculata, a Cryptidia that infects mosquitoes that's used as a model system for understanding the trypanosomes that infect humans. And within weeks, she was able to get our Cryptidia from our bees into culture and start working with it in culture. And the cool thing is that the Cryptidia will actually, in a petri dish, so they infect the gut by attaching to the and grow in these rosettes. So what you're seeing are these rosettes attached to the petri dish, and these are rosettes of Cryptidia grown with buckwheat exines for 48 hours, and here's sunflower exines for 48 hours. And here's some plots of like the average number of rosettes, buckwheat versus sunflower, 24 hours, 48 hours, 72 hours. They don't attach. So sunflower pollen is interfering with the attachment. And they even grow rosettes just in media and let them develop and then rinse them with sunflower exines. That seems to scrape away those pathogens, whereas buckwheat exines don't. So this is interfering with attachment in some way. Megan was also able to genetically transform the Cryptidia with GFP or RFP, so we can have pathogen cells, which is, like, to me, absolutely mind-blowing. But then we can look at we can track infection in the bee gut, which my graduate student Sonia Glasser is doing now in collaboration with Megan. So this is the ileum, the hind gut of the bumblebee, where you can see all of this red is Cryptidia cells that have colonized the hind gut. And so now we can, like, look at actually how diets affect the progression of that disease. That sounded so simple, and it really hasn't been, but Sonia has been doing amazing things with that to help us get a better sense of what's going on. Also, like, whenever I get a talk, people are like, okay, what do I plant to save the bees? Should I tell them to plant sunflowers or not? I'm not sure. Because, it really benefits the bombers and patients. That bee's not having any problems anyway. That's a competitive dominant. We have looked at some other bumblebees, more on that in a minute, and like impacts on other bumblebees, some of them, and also resistance infection, but some not quite so much. So, but like if you knock down the pathogen in the dominant bumblebee, is that good for the community because then there's less pathogen to spread around? Or are you helping the species that didn't need help anyway? I don't know. Can we figure this out with models? Maybe, maybe not, but we're playing with it. So Chris, our modeler, Amelia Kusi is a graduate student of mine, and they've been working on developing some models, but we're also trying to get some data. So we have a postdoc, Lauda is my collaborator, Kate is the postdoc. We've worked with people creating pollinator habitat, some who were just creating the habitat they created anyway, some we designed a seed mix with a lot of Asteraceae, and they grew that instead. Some people just managed pollinator habitat by mowing and not planting anything. And then Kate had a grueling and impressive field season last summer, quantifying floral resources, bee communities, infection in bumblebees, pollinator networks, to try to look at the role of Asteraceae in larger community dynamics. Not ready to show you guys yet, but that's where we're headed. Also, I cut out of this talk because I didn't have enough time, but we're also thinking about how broad are the impacts of sunflower pollen in different bee species. And maybe even using a comparative approach to try to help us figure out what it is that's going on. We've done about 5 different bumblebee species now. The ones that are more closely related to bombers and patients, we see that sunflower reduces infection. Ones that are more distantly related, seems a little bit less that way. Graduate student Elise McCormick is working on that, and is sort of an ongoing project as well. So those are some of the things that we're working on that I would love to chat more with folks about. Overall, we're trying to elucidate both the mechanistic basis and the ecological consequences to understand how plant communities can structure pollinator pathogen dynamics and affect bee health. So now I'm really done. Lots of collaborators, lots of lab members, lots of funding sources to thank, and thank you all for your attention. Do I have time for
You have plenty of time for questions.
I really didn't want to run over.
No,
Okay.
you did great.
I did good? Okay, awesome.
Very interesting informative talk.
Thank you.
I have studied the pollen of more than 300 species of Asteraceae.
Oh.
I wrote a book on sunflowers of Louisiana. Many species do not have spiny pollen.
Yeah.
So did you make a comparison?
Yes.
Of,
We have.
What was your?
It doesn't really seem so in the paper that I didn't show you guys because I was going to run out of time, that has the 7 different Asteraceae, we regressed the length of the spines against the effect, and don't really see much of a relationship. I mean, sunflower kind of skews it a bit because its spines are so long and its effect is so strong. But another one that's really effective is dandelion. And the spines are kind of, so no, it doesn't seem like it's spine length per se. We just had our big integrative biology all-team meeting last week, so I'm kind of bursting with lots of ideas. But April Sharp actually found that newflower pollen and hibiscus pollen, which are both spiny, do reduce infection relative to buckwheat, but not as much as sunflower does. So there's something else going, like there seems to be an Asteraceae effect, and it doesn't seem like it's all about spines. We're starting to talk about like enzyme biochemistry and charges on particles and like negative or positive charges and whether the pollen's just like sticking to the pathogen and then the spines are a distraction. But certainly something about Asteraceae, although mostly about Asteraceae and some other things. Yes?
Well, along those lines, I mean, sporopollen is very resilient. It's difficult to hydrolyze it.
Right.
Is there a difference in the composition in Asteraceae and other steroids?
We just started peeking around at that literature last week, and it does seem like the composition of spore pollen is very diverse, and that's something that we need to start looking into next. I think that's our new thing that we're excited about pursuing. Awesome. Yeah, I hope so. Yes?
Great talk.
Thank you.
I was wondering from the bee perspective, since there's such a strong effect of sunflower pollen on bee health, do bees preferentially visit sunflowers? Do we know?
Thank you for asking. I always, do bees self-medicate? That is the question, always. It has been hard, so yeah, it's been hard to do that in sunflowers because there's such weird flowers anyway. They're so huge, so like, I've done other stuff with sort of preferences for different plant species, and we've had to take sunflowers out because it's such an outlier. The bee spends 30 seconds on this flower, 10 seconds on this flower, and 20 minutes on the sunflower head. So sunflowers are a little bit weird. I do have some other work with a postdoc, Gordon Fitch, who's now at York University, looking at self-medication, actually using plants in the mint family. So you work with Monarda fistulosa. So mints are really interesting because they have a lot of them have chemotypes, so really strong chemical variation that can be controlled by pretty simple genetics. So Monarda fistulosa plants, each individual plant will basically make one monoterpene. So there's like thymol plants, and then there's carbachol plants, and linalool plants, and eugenol plants, and we have a nice self-medication story from that. Yeah, it's not quite out there yet, but we are seeing some evidence that infected bees and uninfected bees are making different choices that prefer the plants that will, the infected bees have a stronger preference for the plants that will reduce their infection more, with consequences for the plants. So I'm excited about it, but it's not quite out there yet. Yeah.
So coming back to the exile in your protocol for getting rid of all the stuff
Yeah.
inside, how sure are you that non-sporopollen constituents within the exile are definitely gone?
We asked Hauke about this, and I reviewed the protocol just over lunch just to make sure. It's a pretty rigorous protocol. It's lots and lots of washes with lots and lots of different kinds of solvents, and then hot water and boiling and tests with hydroxide. Like it is a pretty intense protocol. We've asked Hauke about it. He's like, I really don't think there's anything else left. It's pretty. I mean, I can show you. I've got a protocol I can show you, but it's pretty intensive.
Wow. Have you done any transmission lifetime microscopy? I'm just curious. Look to see what the exiles look like.
Yeah, we did. Yeah, it's in that paper actually, because we also wanted to make sure that we hadn't disrupted them too much. So we have some nice pictures. Like we sort of mixed the exines with our wildflower pollen, and then took some pictures to go to. This doesn't still look like sunflower pollen grain or buckwheat pollen grain, and you can pick them out in there. Yeah, they're still there. Thank you.
Trying to get into something mechanical, like a radiolarian skeleton or something, something that might be shaped that might mechanically.
That'd be fun. Have we tried feeding them something mechanical, like a radiolarian?
You said it was too expensive to.
Yeah, we really love to print like down
But
particles.
there could be some natural.
Like some sort of diagram or something.
Oh,
okay. The next honors thesis. Yeah. All right, that sounds like fun. Thank you. Good idea.
Okay.
What
Let's see. I think back here at 405, is that right? Everyone check the back of your, yes, 405. Okay. That's the new one, or hide the old one?
I think they all work.
They both work?
Yeah, yeah.
Checking the animations on my slides.
I have it pulled up for you.
Oh, I'm not Evan. I'm the next one
Oh,
after this. I
sorry.
just want to test it before. I mean, Evan will do it, but
Evan will do it.
I just want to make sure that like if you.
Sorry,
if you go
I
into
know who you are.
Yeah. Oh, fantastic. Okay. Oh yeah, it seems to be moving.
It's
well.
good? Okay.
Yeah. And then this is the. Well, we'll figure that out.
Do you want to do more? Do you want to go through it all? You're welcome to go through it all.
I just want to.
You can
I don't
also
want to.
unplug it if you want everyone to
Oh no,
see it on
people
the
can
screen.
see a
Okay.
little bit. I just want to do some of the more annoying.
Yeah, I know exactly what you
Because
mean.
there's some of them that tend to be.
The animations that you start and you're like.
Yeah, the ones where it's like I'm
Yeah,
blocking like stuff and
yeah,
things.
yeah.
Okay.
No, it looks great.
Thank you.
Yeah.
I'm just gonna close this first then, or.
Yeah, just close that.
Okay. Thank you.
And collects everything. It's like a little Roomba.
Oh, sure. I just finished. I feel like I'm not already forgot.
It's all right. No pressure.
Yours I read many times. You're talking about the.
I'm doing a mix. I'm talking a little bit about capsicum.
Okay.
I'm talking about Melianthus as well. Yeah. So a little bit about
my favorite.
a little bit of
Melianthus is my favorite.
I wanted to talk a little bit more about it, but per suggestion, I needed to cut it back. So
Also, we have 15 minutes to
yeah.
start.
Yep.
I don't want it to go too sick, so.
Yep.
Great. Go eat something.
I'm afraid if I do, I'll just get hungrier, or I'll have a sugar crash 10 minutes in.
How's this snack?
But my sister, for example, that's how I started. If I show you a Yeah, and those trainers, I can probably, I'm sure that they're. And then I put one another, I can do system, yeah, but it does most of the dispersion. Huge brand. I do my copy, but very easy to only one by India or fruits. His wife named my wife, both are I was affected in India. I'm a very I'm very sunflower oil, people ask me, whatever I bought. Otherwise, they do not know that they think it's fun. I said, why? It's structure. I said, unless you doesn't want, then you compensate. You will not understand what I see.
One of my rules, which I had,
What was doing? What was driving you?
One of my rules was I don't work Friday night, I don't work Saturday night, and I don't work Sunday morning. Other than that, I still work. And that's still the case. I've done that my whole life. But now with my children, so I mean, I have children are grown up, but when our children were small, then like if it was something that my children wanted to do and my wife wanted to do, then that's the priority. Or if my friends called up and said, let's go to tennis, like, it was something to do. I always did that. That was my priority. I said to my children, let's go for a walk. My children say they want to go home, and I would work. What?
But I would medical doctors, yeah, because there is a reason, you know, there's oh, I was saying, very much.
He's a good friend. And I respect him. You know that they moved to California. I am still in communication. Other people was a full set. He was a full set. I said, one thing, you know, my daughter was common with all this.
and then you can just turn it on. That'd be great. You won't hear much because it's mostly for the online people. Yeah. The battery might die that one. Let's try it. Oh no, it's the same. Okay, that's fine. Just use that. Yep.
Test, test. Okay.
And then this should work. Have you tried it yet?
Have not.
Oh. Oh, I was like, did I kill the computer? Please
No.
don't.
There we go.
Okay.
Perfect.
All right. Okay.
Thank you.
Yeah. Okay.
Appreciate it. Yeah, no rush. That's fine. Take. All good.
All right, everyone. Thank you very much for staying until the last. Welcome to the last session. In my mind, it's the best session because we're going to have two early career scientists presenting in this session. It's also relatively short. And the topic, we are having a very difficult time putting the topic for the session, but we decided it's going to be a little bit more molecular chemistry. And then the first speaker is Evan Magner from University of Minnesota in Clay Carter's lab. And then he has done a lot of amazing work on color nectary and then very productive grad student. And then he's going to tell us some of the colorful nectar today. So let's welcome Evan.
Thank you for that introduction, and thank you to the committee for allowing me to share some of my work. This is such an amazing honor, and I'm so excited. I kind of got caught up in everybody else's amazing talks today. I kind of forgot that I was talking too at the end of this. But it's been so exciting. So, I was going to kind of start this off with saying, trying to convince you that nectar is complex. But, I think throughout the day we've seen over and over again, yes, indeed, nectar is complex. It's not just a simple sugar solution. There's so much more that's going on in there. And so I'm going to be talking about just one particular aspect of nectar that is kind of understudied, but I have fallen in love with it, and that is with colored nectars, with like capsicum pubescens here with that beautiful yellow coloration to it there. Now I'm going to back up a little bit. Now when we think about plants and plant pigments, they're ubiquitous. And they serve all these different types of facets of the plants. You know, they're involved in light harvesting and protection and also attraction, like in flowers, as you can think. But one interesting thing about this is that most of the time when we think about pigments that are in things like petals and fruit and root and leaf tissues, these pigments are actually internal. Now, not always, but sometimes these pigments are also found within the cell themselves, and not only within the cell, but also within particular organelles within that cell. So things like carotenoids being in the chloroplast and the chromoplast, and betalins in the vacuoles and the cytoplasm. Except for the rare instance in which you get pigments that are not in that environment and instead are exposed to everything out here, to light, to temperature, to microbes that we've seen, to other pollinators. This is a very not controlled environment compared to what the other pigments are used to. They're used to being in an environment that's controlled pH, a controlled temperature, a nice habitat for them to exist. Here, it's completely different. So I think for me, it's really interesting to think about how this environment actually may be driving some unique chemistries that are occurring. And also, I think looking at these colored nectars, I think it's also a great opportunity to also just help to explain some of the diversity that we see generally in nectar as well. And I think you'll see that I've kind of picked up from a couple of the other presentations, and so it might connect here and there with that. So capsicum pubescens is kind of the one that I just always gravitate towards. But when I think about colored nectars, what I'm really doing is I'm thinking about two different questions. One is what makes colored nectars possible? So what is the pigment and how is that pigment made? But also, what are the functions of those nectar pigments? What are their roles? Are there multiple roles? When I first started this, I was very naive, so I thought it had to be a visual system, and in the story there. But as I'll show you a little bit later, I think there's a lot more going on. And so for this presentation, I'm basically going to break it down. I'm going to start and kind of give you a little brief history of colored nectars. It's a short history, so definitely won't take long on that. I'll kind of talk about a little bit about our methodologies. I know Clay had kind of touched upon that some of the early work with colored nectars, there was some misidentifications. And so we've come up with the methodology that I think really allows us to really pinpoint the pigment that is responsible for the color that we see in the nectar. And I'm going to be doing that through the use of two different case studies that I worked on. The first one is the intensely black nectar of Melianthus, and then the second one is this multifaceted yellow nectar of capsicum. So to get started, we'll get into kind of just a brief history. The first time that colored nectars were attempted to be cataloged or kind of put together into one source was back in 2007 when Hansen and his group actually went through. They went through the literature, they used gray literature, they used word of mouth, they used anecdotal things, they used anything they possibly could to surmise how many and the diversity of colored nectars out there. So they came up with about 70 so species that produce colored nectars. And since that time, less than 10% of those have really even been looked at to even be attempted to characterize them. As Clay had mentioned too, some of the earlier work also misidentified some of these compounds. So like with Nesicodon mauritianus here, the original authors actually identified it as this arone here. Our lab, though, working with this, actually discovered that it was this novel pigment, Nesicodin, which was really interesting. Also interesting is that there is a reoccurring theme that I'm going to keep pointing out through these stories, and that is the presence of ROSs like hydrogen peroxide. There seems to be this co-occurrence that we keep seeing ROSs and pigments having some sort of intimate relationship with each other, which I think is very interesting. We're still trying to figure that out and understand that a little bit better. But in this system here, you can see where we have a synapyl alcohol, a nectar protein, actually then converts that to the synapaldehyde, which can go on to combine with the proline to actually form the Nesicodin itself. Now, as I mentioned, there are challenges, and it's not just the challenges that misidentifying a pigment. As I've learned, the chemistry of this is very, very complex, And sometimes it's not just one pigment that you're actually looking at. So while you're looking for a really, really red pigment, you might be looking through your mass spec data and you might see an orange pigment or a yellow pigment and these types of things. So what we have tried to do is to streamline this in a way to identify the colors that we are most interested in. Unfortunately, those are kind of biased because we're using our eyes and we're not using a pollinator's eyes, so that would be really cool if we could. But what we do, just to break it down really quick, we first take an absorbance spectra of just the raw nectar itself. Then we use zip tips, those are C18, and we basically are just trying to clean up that, so remove sugars, remove salts, those types of things. And then we get another absorbance pattern. We take those clean samples and we run them through an HPLC tandem mass spec then. And of course, we start our mass spec analysis. But this is kind of where the important part happens. And that is when we start looking at the PDA from the mass spec, what I am trying to do is I'm trying to take the absorbance spectrum that I found from my clean sample, and I'm trying to match it up to a particular metabolite. So that way I know the color that I'm chasing is this color, and this metabolite is the only thing in that suite of compounds that could produce that. We then, of course, go on to try to recapitulate the nectar pigments as well, and then, of course, different functional analyses, whether they be effects on pollinators or perhaps maybe microbiome effects as well. So to start, I'm going to start with the Melianthus story here. It has this intensely dark black nectar. You can kind of see up at the top there. It's actually, when we were using a photo card next to it, we do this to kind of standardize the images and the colors. It was more intensely black than the black standard of the photo card, which was really, really interesting. Really cool aspect of that. But here's Melianthus here. It's a small family. There's only 8 in the genus. We have the bush over here, inflorescence, and then the flowers here. And again, like I said, we start out with our absorbance pattern. So here I'm showing a non-anthesis nectar and an anthesis nectar. Non-anthesis can be either before or after that anthesis time point. But the interesting thing that we see is that it is gray. It's slightly acidic, while the dark black actually gets up into the alkaline area. And you can see from the absorbance pattern, with black, we would expect it to be absorbing all the way across the spectrum, and of course it is. So this is the thing that we're going to be chasing now on this. So my first question was, what compounds are actually found in the black nectar? So first, starting with that mass spec data, I'm not going to go through all of these, but 2 of the ones that we were really interested in was gallic acid, and then the second one was ellagic acid. We were interested in these for a couple different reasons. They were, compared to the other ones, you can see the masses are a little bit larger. These are smaller, so we thought maybe these could be precursor molecules. And also there was work done previously that showed that a peroxidase can actually convert gallic acid into its dimer, which is ellagic acid. Now, once we go past those, it's really interesting that the other ones that I've highlighted here, these are also tannins as well. They are just glycosylated mostly. So we've got this suite of tannins that are involved in here. And so really, where do you begin from that? Well, the first thing I really wanted to do was basically, let's quantify those 2 major components, the ellagic acid and the gallic acid. And so we did that here, and the thing that we saw was that from non-anthesis to anthesis nectar, you have the gallic acid is decreasing, okay? During that time, the ellagic acid, which is in the yellow here, is actually increasing in that time. So this is kind of giving me notes here that maybe ellagic acid is important for that black pigment formation. Maybe the gallic acid is more for the lighter gray coloration. So playing around in the lab with just gallic acid and ellagic acid does not give you any coloration really, other than like maybe a pale yellow coloration to it. So how do those compounds relate to actually making a black pigment? That got us thinking about a historical thing, which was iron gall ink. So over here we have an oak gall that was drawn using iron gall ink. And so to make iron gall ink, you take oak galls, pulverize them down, put them in water. You take that water extraction and you add iron salts to it and gum arabica. The iron immediately gives you that black coloration, and even there are notes on past medieval texts that, you know, if you needed to go back and you needed to touch up your work, you could go back with more iron onto your wet ink there, and it would actually intensify that, so it wouldn't go towards that brown coloration that you see on some manuscripts. So with that in mind, we decided, okay, we need to look for iron in here. Is there iron in the actual nectar? And so sure enough, we did see that there is iron in the nectar. And we saw that there is a striking difference in the amount of iron in the nectar. So the gray bar there is that gray non-anthesis nectar, and you can see lower concentration there. And then you get to the anthesis nectar, and you can see that iron concentration has shot up dramatically. So super exciting stuff. But I wanted to make sure that that was actually the effect that was producing this black coloration. So I did a few things here. So what I've done here is I've actually taken the gray colored nectar here, and I've successively added iron at varying concentrations to see if it would actually intensify the black coloration of the nectar. And sure enough, here's some images across the top there with their corresponding concentrations of iron, and you can see that indeed it does continually get darker and darker and darker. I also put this here just to show kind of where the anthesis nectar point is, where the concentration is, and where it is on things. Yes, the question probably will be, are those darker than the ones that we had from the nectar? Hard for our eyes to tell, but absorbance would tell us yes, that those are actually getting more intense as well. So the next thing that I really wanted to figure out is how this is being done post-secretorily. Because as I mentioned, it starts as a gray-colored nectar and then makes its way towards this black coloration. So to do that, I went back to nectar proteins and actually found that NEC3 was being identified as a peroxidase. And as I mentioned earlier, there's already been a paper that discussed how a horseradish peroxidase, or excuse me, a turnip peroxidase was actually able to convert gallic acid to ellagic acid. So with that in mind, I went ahead and basically set up a stepwise progression here. So I'll start over here on the left. We started with active protein that was isolated from Melianthus. I added a comparable amount of glucose to simulate the nectar sugars. I then added the gallic acid at biologically relevant concentrations, and then the hydrogen peroxide. And even at this point here, you can start to see that we're starting to get towards that gray coloration now at that. Finally, add the iron, and it's instantaneous. You get that dark black coloration almost immediately. Now, to compare that, I did the same thing with inactivated proteins and went down the line, and as you can see, you get some gray coloration here, but it is not intense like the black one there as well. So I wanted to, again, match that back up to the absorbance spectra that I was using in the beginning. And so I actually did it a couple different ways. So the black line is the actual anthesis black nectar there. The green line, instead of using the nectar protein, I actually used HRP in this instance just to see if a peroxidase could, in fact, also convert the gallic acid to the ellagic acid. And you can see with the orange line, we're pretty much there as well. And then I did use the nectar protein here with the solid orange line. And then, of course, with the inactivated nectar proteins, it is down here on the bottom with pretty much no absorbance there. So pretty confident then at that point that we were able to recapitulate and truly understand the chemistry that forms this really intensely black colored nectar. Now, moving to my second story, which is about the multifunctional role of riboflavin in the yellow nectar here. So capsicum pubescens over here again, and we have a slightly acidic nectar here. That's kind of unusual for colored nectars. They seem to be alkaline more often than not. And they have these two distinct peaks at 372 and 446, which is important, and you might want to consider keeping that in your head for a minute. So my first question again, just like the last one, what is the actual pigment that's responsible for this yellow coloration? Again, we started with our UHPLC tandem mass spec. The sample came out pretty clean. So the first peak that we have here, coming out at about 7 minutes, we identified as riboflavin. It's the major component of that. Super exciting. At the same time, too, just like Melianthus, I was interested to see what these other components were, or these precursor molecules, or what are they related to those molecules. And what's important to note is that riboflavin, in the presence of light, will actually produce singlet oxygen, as well as these two compounds, cyclodehydroriboflavin and lumochrome. And when I look at my mass spec data, those are the two things that I see in there. So again, just trying to point out that once again we have a colored nectar, and once again we've got ROS being generated in this instance here. So again, an interesting co-occurrence there. So having that identified, I wanted to see, was the riboflavin sufficient enough to actually produce the yellow coloration, or again, is this a suite of compounds that may be contributing in different ways? And so this is actually from the PDA itself here. The yellow line itself is our riboflavin standard that we ran through, and then our nectar sample is right behind it with that black line there. So slight variations in that, but overall, the peaks match up almost perfectly, identically. So it's a great way to fingerprint, basically, what compounds you have. And here is just another look at that, where we took the biologically relevant concentration of riboflavin and compared that to our actual raw nectar samples. Not the clean nectar samples, but the raw nectar samples. And that is also why you see this discrepancy here. Again, the sample was not clean, so therefore you have things like proteins and things like that that are going to be absorbing at that wavelength. The last thing I wanted to do, really, is to see the effects of riboflavin. And first, what I wanted to do is to see, basically, is it a visual cue? The pollinators are not necessarily known for this species. However, it has been shown within papers that Apis mellifera honeybees are effective pollinators and do visit quite often. So kind of using them as a basis, I decided to see if riboflavin could, in fact, be a signal to them. So I did work with Dr. Emily Snell-Rood and Dr. Amod Zombre at the University of Minnesota. And what we did was we actually took the reflectance data of the petals, the leaf, and the nectar itself, and we mapped them inside of the visual space of a bee. So we have long wave, medium wave, and short wave. And so basically what we can see here is that the nectar is here, the leaf is here, the petal is here, and since it is inside that triangle, that means they can see it. If it was outside of that triangle, they were not able to see it. Okay, so now we know they can see these colors, but can they tell the difference between those? So Dr. Zombre actually came up with a really cool way to do that in these just noticeably different calculations that he has. And basically, we saw that indeed the nectar is conspicuous. So here we have the petal to the background and the nectar to the petal, showing that basically bees can see this yellow and it's differentiated. I wanted to take that out into the field. So first round, these are bee water feeders. So we just took those and we found these really cool IR sensors that we just put at the mouth of those. So as a bee enters, it crosses that beam, it gives us a count, it comes back out that same way, gives us another count. We divide everything by 2 just to see. It was really interesting, too, to see the fluorescence that is in that yellow artificial nectar that we created. Right at the interface is just brilliantly green. It's really, really interesting. I wish I could capture it in an image. But here's an image, here's a video rather of that setup. You can see I was at first very hesitant and worried that the bees may not feel comfortable enough to go into the system, but obviously you can see that they were right at home in that quite quickly. And so this is with the nectar observable. We're just showing ratios here. So if there was any favorable days, you would see bars coming down here. Anything above would be favoring the sugar with the riboflavin. And you can see over a 5 trial period, they absolutely chose it over and over and over again, which I thought was cool. I then wanted to see if it could be a nutritional reward as well. And the idea came from a couple different papers here. So the first thing was we found multiple instances in which riboflavin was found to be essential nutrient for bees, namely in brood rearing. Without this, they cannot raise their brood. a more recent paper also showed that riboflavin increased not only the lifespan, but the memory of the bees as well, which was really interesting. So I wanted to test that. And so to do that, I over-engineered a different apparatus to measure this. So to break it down very quickly, we have the exact same feeder that I showed you earlier, same sensor here, but now I have built a wall to where they cannot see the nectar at all. Even when they get on the inside here, they cannot see what the color of the nectar is. And how this works is the bee enters the apparatus. It trips the IR motion sensor, which turns on the camera up here. We get a count from the IR sensor here. We get a count here, and we get video of the action as well. We also added in a weight sensor here in the back, so that way we could also measure the consumption as the bees fed. And there was also an environmental sensor as well. But again, I was worried that they would not want to go into this tube. It's very unnatural for them. But as you can see here, again, it only took them a day to really start feeling comfortable with this. And I think the data you'll see that they got really comfortable. It definitely was amplified throughout the colony because by day 7, you know, we were pretty consistent, even on the last one where it was visible, pretty consistent about ratio of about 1.7 or so. But day 7, at first I thought maybe there was an error, maybe things didn't work, but we tested the apparatus over and over and over again, and it was working just fine. I think it truly was just a sense of having that many visitations. And so I kind of want to just summarize all this up for you all here. Again, I wanted to kind of convince you that nectar was a complex chemical thing, but everyone else did that heavy lifting for me previously. But I think I do really want to highlight that colored nectars are diverse and they're very, very understudied. There are blue nectars, green nectars, there's so many out there. I think that there's a lot of capacity there. And also, again, that interesting connection that we see with these ROSs. Again, I'm not really certain how to understand this because it's a complicated thing. But we attempted to kind of capture this in a recent Tansley insight that we wrote. And so just kind of talking about the species that I talked about today, the Nesicodon, which takes advantage of or producing the hydrogen peroxide, riboflavin, producing a singlet oxygen in this case here, which I didn't show the data on it, but it actually, that singlet oxygen we actually showed actually works as an antimicrobial agent to it as well. And then finally, where hydrogen peroxide is actually being utilized here in this to catalyze the reaction of gallic acid to ellagic acid. So I have a ton of people to thank that have helped me on all these projects and have gotten me to where I am today. That is just a touch of some of those people, but I certainly appreciate them. And with that, I can take any questions.
One quick question.
What kind of pollinators are getting attracted to the black pollen or the black nectar from the melianthus?
Sure. So there was a paper that was done that looked at bird species that were there. So passerine pollinators, sunbirds, they absolutely love it. I think they captured like 22 on their first visitation run of things. I will say that even my experience of collecting on melianthus, bees will avoid it at all costs. They will fly near it, observe it, and get out of there as quick as possible.
Great, thank you. Thank you, Evan. And our next speaker, Galen Tiong, is from Harvard, and he's a grad student in both Naomi Pierce's lab and Ryan Natt's lab. And he's going to take us a little away from the flower and look at interaction between caterpillars and leaves. Thank you.
Thank you. Hi, everyone. Thank you for the introduction. And I mean, today has been such an amazing, everyone has been such amazing speakers, and there's just so many questions I want to ask, and it's been absolutely intriguing. So I will have to start off this talk by making the convincing argument to a group of people, to a group of scientists, plant scientists, why you should care about insects. I'm sure a lot of you are plant insect
scientist, but today I'm actually going to be talking about an insect-insect interaction. I titled my talk Exploring Nectar from a Caterpillar, and hopefully you will see why this might be relevant to a lot of the work that you are doing right now. So, I thought what better way to start a talk about caterpillars than to start talking about cats. So cats get high on catnip, I'm sure, as you can see from this picture. That's a cat really getting into it with catnip right there. And the active compound in catnip is this compound known as napilolactol. This is one of the active compounds that sort of drive this sort of high that they get from this catnip. Now, napilolactol is an iridoid, and to most of you, I mean, with Rachel's talk, she talked about iridoids and iridoid glycosides. You may think of this as a plant molecule, but actually iridoids are named after the ant genus Iridomyrmex, from which iridoids were first isolated. So it may not be completely just a plant molecule. You might also think about it, at least in this context, as sort of an insect molecule. Iridoids are a broad class of these C10 molecules. There are 10 carbons in them, found in several insect orders. You have, is this the light? Is this the laser? Oh, fantastic. You have them in napilolactol in aphids. You have them in iridomyrmecin. This is ants. I mean, beetles has chrysomelidiol, and this is a stick insect, devil stick insect, anisomorphol. So as you can see, there's a lot of convergent evolution from at least a molecular level between plants and between insects in this case. I also want to emphasize that it's a 10 carbon structure, and we'll get to why that is going to be important later. So plants and insects also have convergent solutions for rewarding neutralists. As you can see, with flower nectar, everyone has given fantastic talks about flower nectar and pollen too, of course. But when we think about insects, I think one of the common organisms we think of is honeydew from aphids. But today I'm going to talk about this nectar from a caterpillar as well. So these caterpillars are from the family Lycaenidae. They secrete nectar from a specialized organ on their 7th abdominal segment right here. That's a dorsal nectary organ. And ants consume them. This is a brilliant documentary by David Attenborough. I would recommend you check it out. It's secreting this droplet of nectar right there that contains sugars and amino acids. Now, we often compare this caterpillar nectar to aphid honeydew. But I would like to emphasize that there is a very fine difference between the two here. With aphids, this sugary secretion that we know of honeydew is a byproduct. It's eating the sap, it's stripping it of all the proteins and extracting all the proteins, and what comes out is basically waste. It's a waste product that, if the ants don't get rid of it or if they don't flick it away, it will start to mold, you'll see mildew, and these aphids will die. It's an excretory product. What is different in this case, however, this is not an excretory product whatsoever. This is not the anus of the caterpillar. This is a secretory product. This is actually a specialized gland that's developed by the caterpillar, similar to nectaries in plants, to secrete this sugary nectar solution here. You might almost want to think of it almost like a venom. This is like the glands that you can see in all these different animals. It's really secreting something that's a lot more specialized, and you would expect here a much more specialized physiology and a much more specialized, potentially, metabolome in this case. And now, if you think about venoms, you can think about, well, it's an injection, and I think about drugs. That's, sort of the connections that I'm making here. And so it might be interesting to think, it made me think, is this caterpillar potentially secreting this solution, and is it doping it with some kind of other molecule that might be influencing ant behavior? Now, you may ask, what evidence do we have of this nectar manipulating ant behavior? So this was a study done by a postdoc, I believe, or a visiting scientist, Masaru Hojo, in 2015 in Naomi Pierce's lab, looking at how the DNO secretions of a different of a Lycaenic caterpillar actually affect the ant behavior. And what he found was that ants that feed on these DNO secretions exhibit both decreased locomotor activity and increased aggression. They also have changes in their brain dopamine levels. I can go into that, into more detail if you'd like after this talk. But from this, we can actually reason that something else must be in the nectar. It's not just a simple story of sugars and amino acids going on. Now, Lycaenids are a really broad family. You have close to 5,000, 5,500, 6,000 even, depending on how many they discover in the next few years, of species of these Lycaenids, and about 70% of them are actually associated with ants. But as you can see from all these pictures, the degree to which they're associated with the ants really vary quite a lot, and they associate with different kinds of ants. And in some cases, you might think, well, there are some exemplars. This to you might look like just a tree branch with a whole ton of ants. what are you talking about? What do you mean lysine? I mean, it's right there. If you couldn't see it, the caterpillar is buried under those ants. And those ants aren't eating the caterpillar at all. They're very friendly. They're just swarming the ants and going about their business. And you know, it's right there. It's just an incredible phenotype that we see, that sort of the end, you know, the real end of like how associated with ants, you know, the caterpillar can be. And so my work focuses on this very intensely ant-associated caterpillar. It's Jalminus evagoras. You find it in the coast of eastern Australia. That's the butterfly, and that's the caterpillar right there with its dorsal nectary organ. And if you recall, I was talking about the name iridoids. You might be very interested to know that it is actually very closely associated with the same ant genera that is actually, that the name iridoid comes from. It's very closely associated with Iridomyrmex ants, and you know, to re-emphasize the strong association they have with each other, there's the caterpillar. I'm not sure if you saw it in the earlier picture, but it's just surrounded again by these ants. And so, you know, this is a very well-studied system. This was studied by my advisor, you know, Nomi Pierce, for many years, and many years ago, several years of this study, and you know, many students have sort of looked at this system. And we know it's a mutualism because we know that caterpillar nectar and the secretion that it's making, it enhances colony growth. If you compare these two graphs without caterpillars and with caterpillars, there's a much higher colony size after like 18 weeks of growth. And in return, these ants actually protect the caterpillars. Similarly, if you know, with ants, you don't have that much egg parasitization in the wild, but without ants, you almost have 50% of the eggs being parasitized by these parasitoids. So there is, you know, there's a give and take here, there is a mutualism. Now, this is context dependent, right? This mutualism can actually become a parasitism with, you know, caterpillars manipulating ant workers. I'm going to briefly explain this setup right here. You know, the ants here are allowed to freely associate it. They can either choose to tend the caterpillar over here, if there is a caterpillar, or they can tend the brood in the ant nest over here. Just for the next figure, I'm going to show you these two. You can think about for the next figure, you'll see that there's going to be a shaded shape which is associated with the caterpillar. The unfilled shape is going to be the negative control where there's no caterpillar present. Now, when you compare a colony of about 80 workers right here, when you compare having a caterpillar to tend and no caterpillar to tend, there are a lot fewer ant workers that sort of end up in the nest chamber tending the brood. But you don't really see a large drop in that survival rate if you look at brood survival rate and the y-axis. But what happens is that when the colony size actually gets really small, and you start going to 40 or 20 workers, you have that same decrease actually in the number of workers that are tending the brood, and instead they'll choose to tend the workers, which results in a massive decrease in brood survival rate in those situations. So there's some level of manipulation here where the ants are no longer, you know, ants are no longer sort of in control. There's a lot more, you know, there's a drop in survival, especially once they get at these lower levels, because there has to be something potentially that might be drawing them to tend these caterpillars. And so the question I'm going to talk about today, about my work, actually is looking at what is in the secretion of the Jalminus evagoras caterpillar. You know, besides sugars, besides amino acids, are there other interesting molecules that we can look at? So what I did was, you have this caterpillar with its dorsal nectary organs. I collected secretion, I did LCMS analysis, and I got a few molecules out of it. I'm going to skip over a lot of the technical details and just tell you that I found two molecules of interest. Those are its formulas. We don't exactly know, you know, this is not really a molecule where we have like a direct match, but I'm going to kind of run through the details of like what I think it could be. Based on fragmentation patterns, we can say that it has a C10 scaffold decorated by respectively 3 sugar units and 2 sugar units. And as I mentioned previously, you know, one thing that we know is C10 is a C10 structure, are iridoids. So immediately in my head, I was thinking, well, you know, could this be an iridoid glycoside? And so, you know, the immediate question that went to mind, you know, where is this iridoid glycoside coming? How potentially does a caterpillar make this compound in this gland that we were talking about, this DNO gland? And so what I did was, we did RNA-seq on these caterpillar segments. As I've mentioned, you know, you have the gland is right here, and this is sort of a control area. This is the abdominal first segment. And we were looking for genes that were highly expressed in this DNO segment and not in this control abdominal segment right here. And we actually found a really short, promising list of candidates right here that showed significantly increased expression. In fact, we only found about 14 genes that were differentially expressed. That, you know, this had to do with the number of samples, but it was a reasonably good short list that we had, because out of these genes, 3 genes are known to synthesize iridoid precursors. If some of you know about terpene chemistry, these are this is sort of like involved in the mevalonate pathway. And so I'm going to briefly talk about iridoid biosynthesis really quickly. It starts with, this is terpenoid synthesis as well. It starts with these two 5-carbon units. They get conjugated to form this 10-carbon unit. This is GPP. You have the dephosphorylation of this double phosphate group over here to form geraniol. This is a terpene that we associate with, the smell of flowers. And then there are several steps that occur, between geraniol to form nepentolactol that I mentioned previously, is the molecule that is involved in catnip. And now we know this pathway in aphids, actually, because of a very nice PNAS paper published in 2022 by people from Max Planck. And it's a brilliant biosynthesis paper where they managed to build out the entire pathway with individual genes to synthesize all the way from GPP all the way to nepentolactol. And what I have for you here is that we find 3 genes from this list of 14 in Jalminus oevagoras that have corresponding gene analogs by annotation in the aphid nepentolactol biosynthetic pathway. If you notice, there is there's the aphid version of GAH and the Jalminus oevagoras version of GAH. This is cytochrome P450. This is a short chain dehydrogenase, and then this is a steroid reductase. And we see that there are matching analogs in Jalminus oevagoras to these genes that we know from the aphids. And also, besides those 6 genes, there are also another 3 genes that are of likely biosynthetic interest. Now, what I presented to you here, they just seem, these are just candidates. These are helpful things to maybe like, oh, these are interesting. But I'm sure many of you might be thinking, the next logical question is, how do we actually test the function of these genes? It's fine to have candidates. You have to make something. We have to show that it works. And so we can do this actually by leveraging something known as heterologous expression. We can take the gene from this caterpillar, put into an expression vector, and transform it into various systems. I'm here talking about transforming them into yeast. We can transform it into agrobacterium, which can then infect a Nicotiana benthamiana. And then we can look at the metabolomes of these things. Besides looking at the metabolomes, we can also, they would be expressing this enzyme, and then it will be converting the substrate to the product. So either via in vitro interactions with this purified enzyme, or in vivo, not in vivo, directly extracting the metabolome, we can actually look for product through LC-MS analysis to look for whether we can find these compounds in these systems from a gene that you would never ever find in yeast, and you would never ever find in Nicotiana. And to make long story short, these enzymes do catalyze this multi-step pathway from geraniol to nepentolactol. As I mentioned, these are the genes from the aphid, and these are the genes from the caterpillar. I managed to basically go all the way from geraniol right here through these 3 different genes in different systems to finally get to nepentolactol. In this situation, just for an example, this is one of the figures that I have. If we have a system with the aphid version, we have a system with the Jalminus version, and we have the same peak right here, a nice clean peak in LC-MS. So the natural questions that arise from here, that come from here, are these genes homologous? Are they convergent? That's a very interesting question right here. I have some evidence. We can talk about this. I mean, you can ask it in a question, or we can talk about this like after my talk as well. And actually, I would argue that this is maybe the most interesting question to me. What are the remaining reactions necessary to reach our final molecules? And this is especially interesting because we don't know what these molecules are. We don't really have these real analogs. And by this method of, without knowing the final molecule, building this molecule through biosynthesis, we're really looking at potentially novel compound discovery here. We don't know what these compounds are. Can we build these compounds out with biosynthesis? And then more importantly, use them to then test function in our ants to see if there is a modification of behavior from these pure molecules right here. I'd like to quickly end this talk by talking about the potential ecological function in caterpillar nectar. As I've mentioned, as we want to test it, we want to have a hypothesis to test. And I want to ask whether plant nectar can tell us about its function. We know that in this flower, Catalpa speciosa, they have this. This is an iridoid glycoside catalpol. It's in the nectar of these flowers. It's unpalatable and toxic to nectar thieves. These ants and this skipper right here, if they feed on the nectar, they get intoxicated, and sometimes they can't fly and they'll die. But they are palatable to the right pollinators, Xylocopa, your bumblebees. They will eat the nectar and be completely fine. And so the question I'm asking here, potentially is the caterpillar potentially doing something with its nectar? making something, is it making nectar and doping it with these compounds that make it palatable to iridomyrmex and unpalatable to other ants? And potentially, we can then ask questions of whether this chemical innovation might be driving the mutualism and this extreme mutualism, this extreme association between the ant and the caterpillar here. Yeah, thank you very much. I hope I've convinced you why this might be interesting to you, and I'm happy to take any questions. We'll have time for a couple of questions.
Have you been able to look at any closely related caterpillars and see if you can identify other organisms that generate this, or if it is unique to this particular species or a small group?
So that's the next plan. So we have genomes from other lycinids that are not from this area. I mean, they're from Britain. There's like from Darwin Tree of Life. They're really good genomes. When we look at these genes specifically, there is some evidence that they are duplicated, which gives evidence of neofunctionalization. We don't see this duplication happening in more distantly related lycinids, which makes me think that this might be a lot more specialized within maybe not just this species, but potentially in this genera. But we have intentions to, at least I have intentions to go to Australia and collect these species, look at their secretions, look at their get a good genome, and actually look at, well, are these genes also present in the caterpillars, and do we see something that looks like an iridoid glycoside in these caterpillars as well? And yeah, that's my hypothesis, that there's something going on. To some extent, I think it sort of cuts off at the tribal level because this interaction is really special. Yeah.
What happens with the other life stages, like the eggs and the pupa? Are they on the plants, do the ants attack them or not?
So the ants don't attack the eggs, larvae, and the pupa. The eggs and the pupa don't have this organ that secretes nectar anymore, but they have different. I'm kind of skipping. There are a lot of details about this system which make them excitingly, very interesting, but also very complex. They actually have two other organs that have been described that are involved in this interaction between them and ants. Not even talking about like cuticular hydrocarbons here, but like full-on specialized organs. So the pupa also has a different set of organs that mediate this ant interaction. The ants will swarm the pupa. The ants will also sort of swarm the eggs. And in general, throughout all the stages, except for the butterfly stage, the ants do not attack the caterpillar. In the butterfly stage, the butterfly is flying, but they generally try to avoid. They will lay eggs where they know ants are, but they don't hang around where they're not as friendly anymore at that stage. Yeah.
One last question.
Sorry, I saw him. Yeah.
Plant nectar, the concentration of sucrose varies with species.
Mm-hmm.
How about the caterpillar nectar?
Yes. I have not looked into exact quantifications, but I think from preliminary studies, there is variation in the amount of, the kind of sugars actually. So we know that from initial looks, there is sucrose, there is fructose, there is malacitose in this caterpillar nectar as well, which malacitose and attraction to ants is one of the things. So there's a lot of potentially interesting things. I think there's like some variation across the species. Definitely, yeah.
Thank you, Gala.
Thank you so much.
All right, and our last speaker for the whole symposium is Dr. Patricia Jones from Baldwin College in Maine. And she's an assistant professor at the Baldwin College, and then she's going to tell us a little bit more about bees today and about how the dietary choices of those fascinating creatures. Thank you.
Thank you. Thanks for bearing with me to the very end. I'm really thrilled to be here. I grew up in Cambridge over in Central Square, and I stayed with my dad last night, and he said, "Why'd they invite you to this thing?" And I said, "I don't know, Dad, but I'm stoked. Don't ruin it for me." So I'm really glad to get to talk to you about some of the stuff that I've been doing, both in the lab on campus, and then I also get to run a spectacular field station, best field station in the whole world. You heard it from me. You should come visit, which I'll get to talk about a little bit at the end. But I think to tell you about this place and where I came to in studying bee decisions, I have to tell you a little bit about how I got here. In 2012, I was a grad student of Mike Ryan's at UT Austin, working on these really amazing frog-eating bats, and studying how bats are using different types of frog cues in order to locate them. So they locate frogs by calls, they're listening to frog calls, they're attacking speakers playing frog calls, which I could do in flight cages. And I was really interested in these questions about how does the cognition in this smart little animal allow them to find their food, and then therefore that's then an agent of selection on these frog sexual signals. And so cognition and behavior has this potential to be a selective force on the things that they're foraging on. And I was really inspired in thinking about that cognition and learning by research at the time that Kevin Lala was doing at St. Andrews on these social learning strategies in fishes, and when do fish use social information from other fish, whereas when do they stick with information they've acquired from their own previous experience that they've had. And I was trying to do these experiments with bats because they're really social, and they learn frog calls or novel sounds, like I was using cell phone ringtones I was training them to discover new food sources. So they can learn these novel sounds from other bats, from being exposed to other bats. And so I was doing these experiments in captivity with these bats, and it would take me 2 weeks to catch one bat, often in the field, and then I'd bring them into the lab and I'd train them to a cell phone ringtone, and I'd expose them to a demonstrator bat who'd been trained to a different ringtone, and altering reward schedules such that bats who consistently got rewards, my thought was, okay, bats who consistently get rewards when they go to a ringtone are more likely to ignore what another bat is doing and just stick with their own strategy. It's working well for them. Whereas a bat who inconsistently gets rewards should be more likely to pay attention to social information and pivot to something new. And that was generally what I was finding. Okay, if you got rewards about half the time, you're more likely to investigate these novel ringtones demonstrated by other bats than bats who were getting rewarded every time. And they'd ignore what another bat would do. And Kevin Lala invited me to St. Andrews to a conference. And I was at this conference, and Ellie Ledbeater was talking about the work that she was doing with bumblebees and social learning in bumblebees. And I thought, oh, I'm working in the wrong system. These things are diurnal. You can order them in the mail. I can experiment with them in the lab. And then even better, you just step outside, and there they are, doing the same things that you're studying in the lab. You can't do that with tropical bats. They're out there in the dark in the jungle, and you have no idea what they're doing when they're not in the lab with you. So I thought, okay, this is the system in which I really should be asking these questions about social learning strategies. So I managed to convince Lars Chitka to let me come and spend a summer in his lab, trying to test the same experiments I've been doing with bats with bumblebees. So thinking about if I vary the reward quality of a flower that a bee is going to, and then I provide them social information about an alternative flower, does the reward quality of the thing that they've been going to influence their likelihood of using social information to learn about something new? And so I had these setups where I trained bees. The first thing I did was I had bees, colonies of, this is Bombus terrestris. I gave them this initial test where I just gave them a bee who was totally naive to flower colors. They'd been foraging on clear flowers. First, I just gave them an array without any of these pinned demonstrator bees of blue and yellow flowers and said, okay, what do they choose? What's their sort of initial bias for one color or the other? And then I trained them either to high-quality flowers or low-quality flowers. And then I gave them social information about an alternative or just presented an alternative with no social information. So I had bees who went through these different treatments. And I looked at how did that affect what they do. And bats have preferences for some ringtones over others. So I shouldn't have been too surprised by this. But even within one bumblebee colony, they had these strong biases. Individuals were biased towards some of them. This is one colony of bees. Some of them really liked blue in their initial tests, and some of them liked to go to yellow. These bees were a little bit more biased towards blue, but I had this individual variation in these biases for colors. And those individual variation in biases for colors, their bias for, here I'm calling it the alternative color, so not the one that they were trained to, influence their likelihood of both, influence the effects of both nectar chemistry and their likelihood of using social information. So my bees who'd gotten no training, they'd just gone to this socially demonstrated array that I showed you before where there were pinned bees on one color. They're more likely to land on that color that's been demonstrated by another bee, but especially if that color is the one that they initially had a bias for. Bees who I trained to really high quality information, they mostly ignored social information about an alternative flower color. They stuck with the one they had experience with. Bees who were trained to really low, and by quality this is sucrose concentration, so 20% was my low and 50% was my high. Bees trained to these 20% sucrose concentrations, if there was social information about an alternative, and that alternative was the color they had an innate bias for, they were more likely to go to it. Whereas if it was a color they didn't have a bias for, even with social information, they didn't necessarily visit it. And bees without social information mostly didn't switch. They stuck with what they had some experience with. So seeing these interaction of these three things. What bias does the bee have initially? What experience are they having with different nectar qualities? And then do they have access to social information or not? And this sort of has driven general questions for me about how does behavior, how does and cognition in particular, the previous experiences a bee has had influence what flowers they're choosing to visit in these kind of complex landscapes. And at the time, I was thinking about, bumblebee learning and cognition with these really simple nectars in the lab. And then what's the role of plants in this? So how is it possible that the plant maybe could be manipulating what the bees are doing? And I was particularly fascinated by the concept of plants manipulating bee cognition. The ways that the plants could be influencing how the bees are remembering things or learning. And right at the time, Geraldine Wright came out with this paper on the impacts of caffeine on honeybee memory for floral odors. So she showed that honeybees remember floral odors better when they've been foraging on caffeinated nectar. So this idea of caffeine influencing how you remember different types of floral stimuli, and the possibility of plants manipulating bee cognition for their own benefit, in theory, that they remember their flowers better and are more likely to visit them. And I thought, gosh, that's really cool. I really wanted to think about ways that nectar chemistry could be affecting cognition. So I needed a postdoc. I found Anurag at a entomology conference, and Anurag was working in this milkweed system, thinking about mostly effects of these cardenolides, these toxic milkweed compounds on herbivores, and he'd done a little work on effects on bumblebees, and I convinced him to let me do more work on the effects of these cardenolides on bumblebees in particular, and how they might be influencing bee behavior. So milkweeds have these toxic compounds in their leaves, we know that. They also have these toxic compounds in their nectar, which creates this mystery of why do you have these toxic compounds in nectar. So my thinking on this was largely driven by this classic paper on the ecological significance of toxic nectar, in which Lynn had proposed a bunch of hypotheses about why is nectar toxic. So maybe nectar's toxic because we just heard about the pollinator fidelity hypothesis. Perhaps the toxic nectar deters, and catalpa, perhaps the toxic nectar deters bad pollinators but encourages the good pollinators, and so it's this filter for pollination. Maybe it specifically deters insects who might rob the nectar and maintains the good pollinators. Maybe, and I love this drunken pollinator hypothesis. So this is an impact on cognition, effectively, that's making them kind of intoxicated. And this was particularly proposed in terms of thinking about orchids and milkweed, which have pollinia, which that maybe these bees were not good at grooming the pollinia off themselves, and therefore they were carrying more pollinia around when they were sort of in this intoxicated state. Maybe it's antimicrobial, as we heard about this morning, or I guess that was this afternoon, and its impacts on microbes in nectar and preventing microbial degradation. Or maybe there's this sort of null hypothesis, this pleiotropy hypothesis, that it's a consequence of the defense for leaf tissues. So to my advantage, Jessamine Manson had looked at this already a little bit in milkweeds, and she had looked at concentrations of cardenolides in leaves and in milkweed flowers and in nectar. So she had found that there were qualitative and quantitative differences in cardenolides between leaves and nectar. So some of the plant species with the highest leaf concentrations, like Asclepias curassavica, have very low nectar concentrations. And vice versa, Pumila, which has the highest nectar concentrations of cardenolides, has very low leaf concentrations. So this seemed evidence of, maybe it's not the pleiotropy hypothesis. Perhaps we have these differences. You'd expect them to be correlated with each other if circulating nectar cardenolides was a product of leaf defense. And it's not just quantitative, but like Pumila has 11 different cardenolides in their nectar. So, and those are different ones than occur in the leaves. So there's this variation qualitatively in the compounds that are present. Jessamine had worked a little bit with, none of these cardenolides that occur in milkweeds are commercially available. So we'd worked with some ones that are commercially available so that we could actually do experiments with lots of bumblebees. So Jessamine had worked a bit with digoxin, looking at the effects of digoxin on bumblebees, and found really mixed results. Even at 10 times the highest natural concentration, which is 100 nanograms per microliter, at 10 times that, she'd found some bees are attracted to the presence of digoxin in nectar. And so I messed around a little bit with this, with bumblebees, thinking about, okay, I'm going to train bees to one color associated with just sucrose. one color with sucrose and cardenolides added to it, and then let them forage in a mixed array and say, if you've had these experiences, how is that influencing the choices that you're going to make when you have options? And I did this with a mixture of 2 different commercially available cardenolides because we wanted to sort of mimic the mixtures that occur in milkweeds, but we didn't have access to any of the actual milkweed compounds that sum up to the highest natural concentration that had been reported for that pumula. And there was no effect on individual bumblebees. Individual bumblebees didn't really care. They care about color. They care about the order that they got the stimuli in, but they didn't really care about the presence of those cardenolides in nectar. I thought, okay, this was just one bee and one foraging bout. Maybe they need more exposure. It must take more time, perhaps, to build up this effect. So I tried this other experiment where I had a colony of bumblebees. I attached the whole colony to a foraging arena with this array of flowers, one paired with cardenolide nectar and one with just sucrose. And then I came back in regular intervals and said how many bees are on each of my 2 different flower types to get a sense of what they were visiting. And I let them do that for 2 weeks to record the sort of perhaps a buildup over time that might be happening of an effect. And there's a slight effect of cardenolides over time that at these high natural concentration, they make slightly fewer visits to them. Color matters still. But in general, the effect is a little bit stronger in the first week and weaker in the second week. But okay, maybe at these high natural concentrations, there might be some effect on bees. But I still felt kind of frustrated by this. So I started my lab at Bowdoin. Anurag and I had an NSF grant together. And I started I really wanted to figure out what was happening with these concentrations within the natural range of cardenolides. And so I looked at, okay, from 10 nanograms per microliter wabane, which is a sort of what would be a common milkweed, a common concentration up to 100, what's the impact on bees? These high natural concentrations are toxic. Bees die sooner. They consume less of them over time, but they like to consume actually the low concentration. So they have some preference for these low concentrations of milkweeds and some deterrence within the natural range of high ones. But I'm a behavior person, so I also wanted to see the effects on behavior. And I had an undergraduate honor student who we set up these flight arenas again where we could look at bee choices. And we found when you look at bee choices in these kind of arenas, they preferentially land on flowers that have these low natural concentrations of cardenolides. And they're not really particularly deterred by the high concentrations. Reed is also a computer science major, so we used a deep learning model to track these bees, which was just a cool side note, but if anyone wants to talk about that, it was a fun thing to do. And so even though these nectars are having, at the highest natural recorded concentration, are having toxic effects on bees over time, at our low natural concentrations, they don't seem to be deterrent, and even the bumblebees will prefer them and preferentially land on flowers associated with them in captivity. So those are commercially available cardenolides. What about the ones that actually are in the milkweed nectar? And Anurag has a talented chemist who's able to isolate from milkweeds the actual milkweed cardenolides and ship them to me in a tiny little Ziploc baggie of white powder, which I then can use to do much more realistic experiments with these bumblebees. So we used glycosylated speciescide, which is from common milkweed, it's Scutis syriaca, and we looked at its effect on bumblebees in comparison to wabane, the commercially available cardenolide. So wabane, at the highest natural concentration, bees in these little vials, like Lynn showed earlier, they'll stop consuming it as much over about 4 days of time. Low wabane, similarly, they start to drop off. But the glycosylated specieside, they keep consuming that at fairly high rates across this time period, and they're not different from sucrose. So it doesn't seem to be deterrent for consumption the same way that wabane is. And then really interestingly, Amy Hastings, Anurag's really amazing lab technician, looked at, okay, in terms of the effects on enzyme function. So cardenolides inhibit the function of the sodium-potassium ATPase. Amy isolated sodium-potassium ATPases from the brains of bombus impatiens and of fruit flies, Drosophila melanogaster as our control. And then this is looking at how much cardenolide do you have to add in order to inhibit enzyme activity at 50%. So if a point is higher on this line, or higher on this graph, on this y-axis, that's showing you that the insect is resistant, because you have to add more of it to have the same amount of inhibition. So Drosophila is equally, their enzymes are equally inhibited by this milkweed cardenolide and by wabane. But Bombus and patiens are much more resistant to the effects of this milkweed cardenolide than they are to the commercially available wabane. So whether this is plants evolving compounds that are less toxic to their pollinators, or whether this is pollinators evolving to be more resistant to the compounds that are present in a abundant nectar source, I don't know. But it seems like there's real specialization at the structural level for these particular compounds. I had an undergraduate student who worked on a little bit of behavior with these. We couldn't do a lot because we don't have a lot of the compound, but generally finding that even though it seems like it's not as toxic to their sodium-potassium ATPases, we are at higher concentrations of them seeing some reduction in how much activity they do when they're sort of wandering around in a petri dish. So they seem a little bit lethargic compared to our sucrose control. So we're still maybe seeing some toxic effects in terms of how they're acting. So thinking about cardenolides in nectar in general, so they can be toxic within the natural range. It depends on the compound, very specifically. They can be attractive to bees at low concentrations, which leaves me wondering why. Maybe they're medicinal. Maybe it's the antimicrobial compounds of them. Maybe they just taste good. I don't know what that, it'd be interesting to think more about why they're attracted to them at these low concentrations. And they can be deterrent at our high natural concentrations. I also sort of wonder maybe it's not about the bees. And this is kind of a far-fetched hypothesis, but I'm sort of curious if it's possible for some of these nectar compounds that are consumed by lepidopterans when they're nectaring as adults and then making oviposition decisions about whether the presence in nectar might actually be influencing the oviposition decisions of the herbivores. And so I did this experiment with monarchs where I gave monarchs experience foraging on these artificial flowers with two different colors, one paired with a high cardenolide concentration and one with just sucrose. And then I paired the flowers with plants. And I could take, the wonderful thing about milkweeds is all the ones in a pod are full sibs, so I could take these full sibling milkweeds, pair them with artificial plants. It's artificial in all kinds of ways, so I think somebody needs to work on this a whole bunch in some other ways, because I think it's a cool idea. But I could look at, all right, they had multiple days of experience foraging on these flowers. I'd shown in other things that they can learn to associate, they can learn these associations between cardenolides and flowers, and then I had them lay eggs in these experiments. And we do see some reduced oviposition on plants that are paired with flowers that had cardenolides added to nectar. So maybe some of this, even though we think about nectar as being about the pollinators, maybe for some of these herbivores that are feeding on nectar, there's some role that the nectar is actually signaling to herbivores. So in thinking about Lynn's hypotheses from this 2000 paper, and why is nectar toxic? And for milkweeds, I sort of said, okay, it's not the, Jessamine had showed it's not really the pleiotropy hypothesis. Maybe we are seeing some filtering happening here. There's still a lot of unknown questions about it. But I was really interested in the sort of two of these hypotheses, this drunken pollinator and antimicrobial hypothesis, and how the two of those things might be interacting. Because it seemed to me that if you're antimicrobial, then you're antifungal, probably, which might also mean that you're preventing the production of ethanol in these flowers. And so one of the things that I was sort of curious and thinking about in leaving cardenolides behind and thinking about nectar chemistry and cognition is what are the impacts of some of these compounds, and could they be having interesting sort of contrasting effects? So I had these two thoughts. Maybe secondary metabolites in nectar could directly increase preference or constancy, similarly to Geraldine Wright's proposal that evidence that caffeine enhances floral memory for odors. Or maybe the presence of these secondary metabolites might be preventing microbial growth, and maybe they're doing that because the ethanol is costly. If you build up some aspect of ethanol, does the ethanol itself decrease preference or constancy? So I was doing this experiment with behavioral experiment with bees in captivity and thinking about giving individual bees experiences, either with just sucrose or with sucrose and caffeine or sucrose and ethanol added to them, and then allowing them to forage in an array. And I wanted to look at constancy and preference, so two slightly different things. So in constancy, I'm thinking about the order of visits. And do you consistently visit the same flower type, or how much do you switch to a novel flower type? Whereas preference, I'm thinking about the total number of visits to each different flower type. And how do these different chemistries impact what the bees are doing? I also wanted to look at social behavior. So do these compounds, which we know have social influences in mammals, are they having social effects in bumblebees? And if you provide bumblebees with an array of flowers with other bees added to them, how do they respond to the presence of those other bees? So in terms of constancy, when bees are trained to blue, they show constancy for blue. When they're trained to white, they don't. They forage randomly, effectively in our arena. They still have a preference, which I'll show you in a minute, but their 0 would be like a random choice between, in terms of visit order, whereas 1 would be really constant, visiting the same flower type over and over again. When you give them caffeine, on blue, similarly, they're pretty constant. On white, they're not constant. With ethanol, they're slightly less constant, but it's not different. And they still are more constant on blue and not very constant on yellow. So color matters more. Color always matters. Color matters more in terms of how these guys are responding to these compounds. And then when I look at preference, so this is the total number of visits to white flowers. So if it's 1, then they're visiting white flowers. If it's -1, they're visiting blue flowers. They do have a preference for white, the ones who are trained to white, even though they're not very constant. It's not as strong as the preference that they have for blue. And it doesn't really matter between these different compounds, so not strong effects of these compounds on what they're doing, except that the caffeine and ethanol had sort of reduced preference for this white, this less preferred flower type. Whereas for the more preferred flower type, it didn't affect them. They were still preferentially going to it. When I looked at social behavior, all of these bees, for the most part, were choosing to land on unoccupied flowers, with the exception of when bees who had experience with white and some of the caffeinated bees was marginal, that they were a little more likely to join other bees at flowers or perhaps not care, not preferentially avoid other bees at flowers in this case. So this response to chemistry depends on the colors that it's paired with. And this has been like such a repeated theme for me. Whenever I'm trying to do something where I want to set up a choice, and pairing a color makes so much sense to me in thinking about a bee foraging out in the field and choosing between colors, but the color really matters in terms of influencing the choices that the bees are making. I have the privilege in this job at a tiny liberal arts college with just undergrads to run a field station. And this field station is in Canada. So Bowdoin is here in Brunswick, Maine, just north of Portland. The field station is up here, just off the island of Grand Manan. So we cross the border into Canada. We take a big car ferry, and then we take a lobster boat. And this is my field station on Kent Island. We actually own these 3 islands, and the field station is located here on Kent Island. Bowdoin acquired it in 1936. So we've had it a long time. It's a spectacular place to get to work. We've got little family cabins. We've got a dorm. We've got lab space. It's a great place to be with kids. We've got fireweed. We've got goldenrod. Please come work with me on insects. Because it's traditionally been an ornithological research station. My current NSF grant is on storm petrels. If anyone talks about pelagic seabirds, I'm happy to talk about that too. But it's a great place to get to do pollination stuff. Because it's a boreal island with reduced plant diversity compared to other places, the boreal zone has fairly low plant diversity, and it's surrounded by very cold ocean. And so it's a wonderful place to be able to think about what is the plant network that's available here, because it's very bounded compared to the mainland. We have no honeybees that have been introduced, no bombus impatiens, and we can think about, within this really bounded area, can we know who all the plants are and then who the pollinators are visiting those plants? So I've been working since I became director on creating a plant pollinator network for the island, thinking about, on the green here, my plant species, and my yellow are my insects, thinking about what are the plants that these insects are going to. And then in doing this work, I wanted to think about, well, what are the stimuli that are shaping this kind of network? Like, how are bees making decisions within this network context? And I was really inspired by Aphrodite Kansa's work, looking at how floral stimuli structure pollination networks. And she's worked with scent and flower, color, and phenology and other things, and how they're shaping the ways that networks are structured and the bee choices. So the first thing we did was we get a spectrometer, and we go out. This is one of my Bowdoin students, measuring the color of flowers on Kent Island. And then we can take the Kent Island flowers, and here they are colored for your human color vision space, looking at ones that are purple are reflecting more blue and more red. We've got some UV reflectance, especially in our yellow ones. And I can plot them in, this is honeybee color vision space, just because I figure it's sort of a general hymenopteron. And we can say, okay, colors that are more distinct in this B color vision space are more distinct to B, should be more distinct to B vision in general. And colors that are more similar should be more similar, are less distinct to the B's visual systems. These are stimuli that I then used in learning experiments. So I was curious about these insects that are going to plants in the field on Kent Island. They're within this plant community. They're making decisions about what to visit. And one of the things that I wondered about is species that are more specialized, are visiting a fewer number of plant species. Are those plant species they're going to more similar in color? Are they specializing on color effectively? Or are they specializing on other plant traits? And it may be that those plants are going to are actually quite distinct in color. And perhaps generalist species are visiting plants that are more similar in color, whereas more, instead of ones that are more spread in their color space. So I used some of these common pollinators that we have, some wasps and a megachilid bee, some bumblebees, including a cuckoo bumblebee species. And this is what I found, that the more specialized these bees are, and they're not all that specialized, they're all fairly generalist species in our little island network, but the more specialized they are, this is a measure from network analysis, the more distinct, the more distance there are in the colors of plants that they're going to. So these fairly specialized insects are visiting colors, plants that are more distinct in flower color. And so one of the things that I wondered about is what is driving that? And could it be that they're better at color learning? And if the more specialized insects are better at learning colors, does that allow them then to focus on plants that are quite distinct in color from each other in terms of what they're visiting, whereas more generalized insects perhaps are visiting plants that are more similar in color? And so we did the learning trial methods that I'd mooched from Felicity Muth at UC Davis, where you can wild catch a bee and test their behavior in captivity, because none of these species are commercially available to me. I'm just catching wild bees out at a field station and looking at how well do they learn these different color associations. So what we find is that that's the case, that our bees who are more specialized, or they're not all bees, wasps, are insects who are more specialized, make more correct choices in these training trials. So they seem to be better, they perform better in this color learning test. And maybe that performance in that color learning test is indicative of in the field that's allowing them to specialize on particular plants that are more distinct in flower color. We're expanding this because one of our other common pollinators up there are syrphid flies. So the other things we can do is we can take all those plant colors and we can look at them in fly color vision space. And then we can do these same learning experiments. The wonderful thing about this protocol is we can put these syrphid flies in little vials and test their color learning abilities. Syrphid flies really like yellow, universal concept that the color really matters. But we can do these experiments. This is my honors student, Whit, who's just about to defend his honors thesis, thinking about how syrphid flies are learning colors within this kind of network context. So I just want to conclude by saying thank you for coming, thank you for staying past 5 o'clock. Color really matters. Any kind of response that bees are having to any kind of stimulus, it seems like it always matters what color the flower is, because if it's a color they like, they don't care if the nectar is toxic or if it doesn't taste good. And I wonder about the possibilities of that for plants to manipulate that and overcome distasteful, having defensive compounds in your nectar. If flower color is really attractive, does that allow you to get away with maybe having less good quality nectar or toxic compounds in nectar that might be deterrent to bees? And then I just want to say thank you mostly to all my undergraduate students who've helped with all this research, to my NSF grants that have funded it, to Bowdoin College, who's funded a lot of this with endowments for our field station. And I'll need a postdoc to come and do plant insect stuff with me. We have an endowed postdoc. So come work at this really spectacular little field station in the Bay of Fundy. And I'm happy to take questions.
Time for a couple questions. I guess people are getting tired.
Yeah, fair enough. Yeah, Lynn.
Thank you, that was so much fun. I'm just curious, there's this paper about milkweed, cardinalides, and that Bombus physioculus.
Yes.
Yeah. So this other bumblebee species that like an awful lot of species of bumblebees on milkweeds are Bombus
Yeah.
physioculus.
Yeah.
And it seems to be able to tolerate those compounds.
Yeah.
A lot better. So I was curious, I don't know if this question is anything better than it looks like you found that the Bombus implications was kind of tolerant, which I was surprised because the sort of message of that paper was that Bombus physioculus was much more tolerant of these compounds, and that maybe those compounds are sort of filtering to this
Yeah, it's such a cool paper because he used Jared Lee's paper from Penn State, where he used, right, Penn State, or is he at Michigan State? Penn State, where he used iNaturalist records of photos of bees on flowers, which I just think is such a cool way to study pollination biology. Yeah, and he has access to Penn State's entomology, where they're rearing all these bees in captivity. So they took all these species and looked at the effects of cardenolides on them. So I could imagine that impatiens is more sensitive than Gracioculus, for sure. And I think that there's species variation in that, I would suspect. There's also no milkweeds, which Anurag says I should just bring them there. So this is my pivot of yeah, there are no milkweeds, but I would love, there's no Gracioculus, and there's no impatiens. Sandersonis are common bumblebee. But yeah, so yeah. Was that a question? Yeah.
Previously, one of the speaker mentioned about the pollen count accuracy, how it could be kind of medicinal effect on that. So, do you have any idea whether bumblebees learn that aspect of foraging on the pollen, this kind of pollen would be helpful to its health?
Yeah, so milkweed is locked in these pollinia. So the bumblebees don't have access to the pollen from milkweed as a food source. The impacts of cardenolides in milkweed nectar and whether the cardenolides are toxic to Crithidia, I don't know if anybody's looked at. Have you looked at it, Lynn?
I think we need to.
Yeah. But that's a great question.
This concludes the whole symposium, but the conversation doesn't have to end here. We have a symposium. We have a reception for an hour with food and drinks outside, and then also you're welcome to walk around the arboretum to hear Matt talking about all the great plants here. And then just a couple of advertisements. We have the special issue accompanying the symposium on new phytologists coming out in a couple of weeks. Many of the stories we talk about here actually have been published on this virtual issue. So if you're interested in that, check it out. And not to proclaim the spot, but he did mention he's interested in thinking about in the next 5 to 10 years organizing another meeting. So give him pressure. Yeah. With that, let's thank all the speakers again. Yeah. And then the shuttle go back to Cambridge leave at 6:30. And then you have a time, please thank Wendy and Nicole, which is outside, for helping putting this together, and also Faye for organizing the event.